Good morning, everyone. I'm Nihal Altan-Bonet, and I'm the chair of this year's Salzman Award and Symposium Committee. Along with my co-chair, Paolo Lusso, and committee members Alan Ryan, Joe Marco Trigiano, Gabriel Parra, and the founder of this annual symposium, Mrs. Lenore Salzman, I'd like to welcome all of you to the 22nd Norman P. Salzman Memorial Awards and Symposium in Basic and Clinical Virology. This symposium was established by Mrs. Salzman, the Salzman family, and the Foundation for the National Institutes of Health to remember the legacy of Dr. Norman Salzman. Dr. Salzman spent much of his scientific career here at the NIH and was a true pioneer in molecular biology. Can I have the next slide, Katie, please? Thank you. Dr. Salzman spent much of his scientific career here at the NIH and was a true pioneer in molecular biology and biochemistry, and whose investigations and discoveries into the mechanisms behind viral RNA gene expression and replication led to the founding of what is known today as the field of molecular virology. Dr. Salzman, in addition to being a pioneering scientist, dedicated himself to training and mentoring students and postdocs in virology, and his lab alumni include many scientific luminaries like Michael Bishop, Bernie Moss, Aaron Shatkin, Marshall Bloom, who will be speaking today, Kenneth Burns, George Curry, Peter Howley, John Mendelsohn, and many, many more. In this spirit, this year, for the first time, we will be bestowing 2 awards, one for an outstanding postdoc and the other for an outstanding graduate student whose investigations have contributed to moving the field of virology forward. Next slide, please. In addition to the 2 awards, this year's symposium is unusual in that it will be all virtual due to the COVID-19 pandemic. While this isn't ideal, it allowed us to extend the reach of this symposium and invite incredible scientists from around the world to speak and share their research, in particular on the biology of coronaviruses, on current vaccine efforts, and on emerging new viruses that may be the cause of the next pandemic. Next slide. This symposium will be split into a morning and afternoon session, separated by a 1-hour lunch break. The program is available on the website. I would like to ask all of you to use your chat boxes to write your questions down for each speaker. At the end of each speaker's talk, we will select some of the questions, ask them to the speakers on your behalf. I'd like to start our morning session off by introducing a man who needs no introduction, Dr. Harvey Alter. Dr. Alter, as you all know, just won the Nobel Prize in Medicine and Physiology, sharing the prize with Drs. Michael Houghton and Charlie Weiss. Dr. Alter's entire career has been spent at NIH, arriving first at NIH in 1961. In 1969, he became the chief of the Infectious Diseases section and associate director of research in the Department of Transfusion Medicine at the NIH Clinical Center. Dr. Alter's investigations have bridged molecular virology and medicine throughout his career. His first contribution to virology and medicine was as the co-discoverer of the so-called Australia antigen, a viral surface antigen that was key to detecting and screening blood supplies for hepatitis B virus. His second major contribution was after he created at the Clinical Center a blood bank, and through his investigations with the stored blood samples in this bank, realized that even though the blood transfused to patients was being screened for hepatitis A and B viruses, the patients were still coming down with a form of hepatitis. He conjectured that there must be another virus in the blood supply, which he called non-A, non-B hepatitis virus, and which as we all now know, was the hepatitis C virus. It was this seminal work, along with the pioneering investigations by Dr. Houghton and Rice, including sequencing and culturing the hepatitis C virus, and demonstrating that it alone was sufficient to be the causative agent for the non-A, non-B hepatitis, that was honored last month by the Nobel Academy. But much more important than this honor, Dr. Alter's astute observations and conjecture of the existence of hepatitis C contributed to the development of new blood screening tools and therapeutic agents that has saved and continues to save countless number of lives around the world. Without further ado, I'd like all of us to give a warm virtual welcome to Dr. Alter. Thank you, Dr. Alter, for being with us this morning.
Thank you for that. I give you a virtual thank you. I did not have the privilege of working with Dr. Salzman, but I knew him and Lenore very well, and they are both very special people. And it's wonderful that Dr. Salzman is re-honored every year with this symposium. And young scientists are given recognition, giving us some hope for the future of science in these days when it is under severe attack. I think I'm here because I won the Nobel Prize, and you all came to see how was that possible for somebody who just made it out of PS 77 in Queens. I really don't know how to respond to the Nobel. Truthfully, I've never won one before. And I have, however, learned some things in the aftermath. One is that the elation is very short-lived, but emails go on forever. Secondly, I found that on October 4th, I was just another ex-Yellow Beret who'd survived several Board of Scientific Council's reviews, and had probably outlived his time at NIH. But on October 5th, I was suddenly a stable genius, a font of wisdom, and the savior of the world as we know it. But I want to assure you now that I am the October 4th version of myself. Today, I'm supposed to give an inspirational message to young scientists to provide a roadmap to the path for success. One of my congratulatory cards summed up success, I thought, wonderfully. It said, "There is no elevator to success. You have to take the stairs." This is profoundly true for my research. It was a step-by-step process with an overarching goal, but not the end point of discovering a virus or of eradicating a disease. The process of research is slow and incremental gains. It's filled with incremental gains rather than eureka moments, and often many missteps along the way. You have to remember that stairs go down as well as up. So what are the elements of the climb to research? The first point really is that hard work is a given. You have to work hard as if your career depended on it, because it does. I don't know of any NIH investigator, and probably the same in academia, who doesn't work more than the given hours, and then doesn't take additional work home. So if you're not willing to work hard, then research is not the career for you. There's a scurrilous rumor about that government workers are unmotivated and lazy. I just want you to know that I have always given 100% at work. It's just that it has this peculiar distribution. And today happens to be one of my worst days. So the next point really is that you have to find a mentor, preferably a great mentor, someone who wants your mind as well as your hands, and who seeks your advancement. My first mentor, by chance, was Baruch Blumberg, a Nobelist in the making. And Barry imparted a research philosophy to me that has served me well throughout my career. I'll get back to that later. But my greatest mentor was Bob Purcell. Bob and I were a relatively similar age, but Bob was way ahead of me in his experience, in his knowledge of virology, and in his research savvy. He taught me more than anyone else in my career. He was both a mentor and a collaborator and a friend, and he should have shared this Nobel Prize. Here's a picture of Bob with John Jaron, another early mentor of mine. John went on to work for many decades with Norm Salzman and has a brilliant career. Lastly, in the recent decades, a clone of Dr. Purcell, another mentor collaborator is Patricia Farsi. Her brilliance has greatly enhanced anything I could have done on my own. In addition, Patricia taught me to always adhere to ethical principles. Thus, my guiding principle is to steal from one is plagiarism, but to steal from many is research. Next, I want to stress collaboration. I've already alluded to this, but being collaborative is essential in today's world where there's such a massive body of knowledge and such highly advanced technology. So I'd advise you to try to become a specialist in something. Try to have a unique knowledge base, whether that be just that you've captured your field, or that you have a particular data set, or you have a particular collection of samples, or a technical skill that makes others want to collaborate with you, because you're going to find that collaboration is essential and it's to everybody's benefit. Next, I want to emphasize observation and persistence. When I first saw this Australia antigen, this unique line of a reaction between a multiply transfused patient with hemophilia and an Australian Aborigine, it was an interesting finding, but it had no clinical correlations. Blumberg, who then left NIH, continued to doggedly pursue its meaning, and often through divergent and dead-end paths, his team ultimately found that it represented the surface protein of the hepatitis B virus. This turned out to have massive importance in terms of testing, blood testing, and vaccine development. But the pursuit of its meaning, this is the point I want to make, the pursuit of its meaning could have been dropped at any time. There were so many dead ends. So what Blumberg taught me more than anything else was persistence. And that was needed in our long-term studies of post-transfusion hepatitis that was spread out over 30 years. There were a lot of incremental insights, but discovering a new virus was not in the original plan. But what I learned is one hangs in there long enough, the unexpected can become real. This is what makes research, whether it be clinical or basic, so empowering. Next, as Yogi Berra supposedly said, when you come to a fork in the road, take it. The road less traveled might be the one with the unanticipated payoff. You have to take some risks, not too many, but you have to take some risks sometime, and you have to spend time. Lastly, find a great partner, someone who will support you and forgive you for all the hours you spent away from home, for all the hours you spent at the office, and then at home. This is my wife, Diane, who was always there for me, even in the most difficult moments. So finally, I want to leave you with a poem, a definitive poem about success that I wish I had written, but Ralph Waldo Emerson beat me to it. And he said, success, to laugh often and much, to win the respect of intelligent people, to earn the appreciation of honest critics, to appreciate beauty, to find the best in others, to leave the world a bit better, to know even one life has breathed easier because you have lived. That is to have succeeded. So thank you very much. May you have success. That's it, Nihal.
Thank you, Dr. Alter. Thank you so much. Our next speaker will be Maria Freire, President and Executive Director of the Foundation for the National Institutes of Health.
Good morning. I'm Dr. Maria Freire, President and Executive Director of the Foundation for the National Institutes of Health. As many of you know, Congress created the foundation almost 25 years ago to support the mission of the NIH. We raise funds to create and manage programs, including partnerships that advance biomedical research, and we also support education and training. I'm delighted to join you today, albeit virtually, to celebrate the 22nd Norman P. Salzman Memorial Symposium and Awards in Basic and Clinical Virology. I must extend a very warm welcome to Ms. Lenore Salzman, the Awards Organizing Committee, and the NIH Virology Interest Group, all who work with the FNIH each year to make this special event possible. More than 2 decades ago, Lenore came to the FNIH to establish the Norman P. Salzman Memorial Fund in honor of her late husband. In fact, it was one of the very first awards FNIH has ever sponsored. We're very proud of that. With support from family, colleagues, and friends, the fund honors Norman's legacy as a pioneer in molecular biology and a very well-respected member of the NIH community. Apart from the 40 years of groundbreaking work, Norman's laboratory was recognized for one of the best training grounds for young scientists, and in fact, many of them today are shining stars in the field. So I want to thank you, Lenore, for your generosity and for your inspired vision. While the annual symposium highlights the current research of eminent extramural and intramural scientists, the awards in virology are presented to outstanding fellows. In fact, this is a way of continuing Norman's legacy. So let us congratulate the recipients of this year's awards and their mentors, and we very much look forward to hearing you speak about your work. Now, I will hand the podium, or actually, should I say the screen, to Dr. Nihal Altan-Bonnet, who will introduce our keynote speaker. Thank you.
Actually, it will be Dr. Paolo Lusso, my co-chair, who will be introducing our keynote speaker. Thank you. Thank you so much.
Good morning, everyone. I'm Paolo Lusso from the National Institute of Allergy and Infectious Diseases, and it is my great pleasure and honor to introduce our keynote lecturer today, who is Dr. Rino Rappuoli. Dr. Rappuoli is the chief scientist and head of the external research and development at GlaxoSmithKline Vaccines, the founder of the GSK Vaccine Institute for Global Health, and one of the true giants of modern vaccinology. His visionary work led to a series of new paradigms in the vaccine field, including the reverse vaccinology concept that exploits genomics to identify vaccine candidates, and which have truly revolutionized our approach to vaccine design and development. His major achievements are many, include the development of the recombinant pertussis vaccine, vaccines for meningococcus B and C, and many others. Rino is the recipient of several prestigious awards, including the Paul Ehrlich Prize, the Albert Sabin Gold Medal, and the Italian President Gold Medal. He's a member of many prestigious societies, including the EMBO, the National Academy of Sciences, and the Royal Society, among others. So without further ado, it's my distinct honor to introduce Dr. Rino Rappuoli, who will talk about perspectives for active and passive immunization after COVID-19 acceleration. Rino?
Today in my talk, I'm gonna try to tell you how vaccination and human monoclonal antibodies can help us to control the COVID-19 pandemic, and also how we can take advantage of the learnings we're gonna do about this to try to better plan the future. So if we move to the next slide, the next slide will bring you a little bit off the road because it brings you to my city, Siena. And the reason to tell you about my city is because in Siena, which is a small city, slightly more 50,000 people, this is not really the first pandemic. Next slide tells you, next slide shows you a beautiful picture of the 13th century, when Siena was one of the richest cities worldwide at that time. And they were very rich, they were doing trading, they were start banking, and they had lots of painters, artists, architects coming to Siena. So for a rich city like that, they made a dream. They wanted to build the largest cathedral ever made. Somehow, like today, if you want to show your power, you build the tallest building worldwide. So they started building in 1348. And then next slide will show you that basically you see the red arrow. That arrow points what they built. They built a facade, really an impressive, strong, big facade. It was supposed to face south, where people were coming from Rome, and anybody coming to the city had to be intimidated by what they were going to see. But in 1348, May, the Black Death plague came to Siena. In 3 months, 2/3 of the population died. The cathedral was never finished. You see on the back of the wall, the modern cathedral is still big, but nothing compared to what was supposed to be. Next slide shows you the same wall from a different point of view. And basically, you see that wall that is supposed to be the facade of a huge cathedral has never been finished. And today, I call that the largest monument to infectious diseases. Because it reminds us how in 3 months, from May to September 1348, a pandemic for that time was able to destroy one of the most important economies of the time, the culture, the dreams of many people. So that's what the experience we have in Siena. Next slide will tell you bring you back to 2020. Which are the big threats or the most important threats to our planet today? Well, 2 things I want to put on the table. COVID-19 pandemic, obviously, we have 45 million cases, more than 1.2 million deaths, 16 trillion, the total cost has been calculated to the US economy. The other threat I want to put on the table is antimicrobial resistance. There are many million cases per year worldwide, 700,000 deaths every year. And the cost has been calculated between now and 2050 is 100 trillion, bigger than the one of COVID-19 pandemic. Not only bigger than that, as big as the cost that's been calculated for climate change. So it's impressive, something that we don't need to forget. So next slide shows you where we stand today. Well, we stand today is the place where I think it's predictable that by 2021 we will control COVID-19 pandemic thanks to vaccines and human monoclonal antibodies, maybe antivirals. In 2020, we don't have very much. We have the same tools that they had in Siena 700 years ago: quarantine, social distancing, hygiene, what we call today non-pharmaceutical interventions. By 2021, I predict that human monoclonal antibodies and vaccines will control this pandemic. And the question is, how is it possible? Usually, I've been doing vaccines 40 years. Usually it takes 10, 15, 20 years to make one vaccine. How can we make it in 1 year? We can because of 2 things: technology, huge progress in technology, and the unprecedented investment in developing and manufacturing of vaccines. So the next slide will talk about technology. You see, this is the technology being used for vaccines. And if you go back at the bottom of the slide, you see 1930s, vaccines were very empirical. 1980, we started new technologies, recombinant DNA, then we got glycoconjugation, then we got genomics, reverse vaccinology. And today, my vision is that we have all these explosions of new technologies. And you see numbers. The 1, 2, 3, and 4 are the ones that have been think are contributing the most to the development of vaccines for COVID-19. Number one is the ability to make vaccines without ever seeing a virus, just using the genomic information downloaded from the internet. Number two is the ability to design, by structure-based design, stable antigens that are already ready to go. Number three is the use of synthetic biology, basically to do RNA vaccines, basically to do synthetic genes, all these kind of things. And number four are adjuvants. They're basically absolutely necessary to do vaccines based on proteins. Next slide will show you the first one, number one. Number one was started in 2013. At that time, up to that point, to make vaccines for viruses, you need basically the virus, you need to grow the virus, all those kind of things. But in 2013, on the Easter day, the Chinese CDC put on the internet the sequence of the genome of a potentially pandemic influenza virus, H7 and 9. And at that time, collaborating with Craig Venter, basically we were able to make in 1 week a synthetic gene that was used to make an RNA vaccine to immunize animals and a virus that could be used to make conventional vaccines in 1 week. That was the first time that basically internet was used to make vaccines. The second, the other technologies, next slide, which are being used to make vaccines are summarized here. 4 of them: reverse vaccinology, structural vaccinology, synthetic biology, and adjuvants. The orange arrows basically indicate the key things which have been important for COVID-19 vaccines. And obviously the internet-based I mentioned before is the part that the important piece for reverse vaccinology. For structural vaccinology is a milestone in 2017 when the coronavirus spike of MERS was stabilized by structure-based design. The synthetic biology allowed to make synthetic genes, but also to do develop RNA and DNA vaccines. And finally, in adjuvants, you see MF59, SO3, and CPG, which are licensed adjuvants, which are presently being used for development of COVID-19 vaccines. So those are the technologies that allow to go very fast in developing vaccines for COVID-19. What about investment? The next slide shows you that basically the vaccine development can be divided into the discovery phase, early development, late development, and delivery at the end. And the overall cost of all these phases is at least 1 billion. And you see the approximate cost and time required for the discovery, development, and early development, late development, which is 10, 20, and 70%. Usually, when we make vaccines, we never enter the early development unless the discovery has been very successful and basically well done. Then we never enter the late development unless the early development is done. We never build a manufacturing facility unless the vaccine has been successful in clinical trials. Well, this time, thanks to the big investment done by the public sector, of which the United States used more than 10 billion alone, but also Europe and the UK. Basically, all these things have been done in parallel. So companies could afford to do these things in parallel. Next slide. And so basically speed up things. Thanks to that, not previous one, please. Thanks to that, basically, there were 320 vaccines in early development. Approximately 10% of them reached clinical trials, and between 7 and 12, depending how you count, they're already in phase 3 trials. And I think today is really a special day because I just read in the news that one of the vaccines, the one Pfizer BioNTech, has reported in a press release a very remarkable success of 90% efficacy in prevention of disease. So beautiful results, and in less than 1 year, thanks to the investment and to the technology. Next slide shows, summarizes in one slide what happened. The sequence downloaded from the internet used to make a synthetic gene, and that synthetic gene has been used to make RNA vaccines, which being entirely synthetic, have been able to reach the clinical trial in a remarkable time of 66 days and phase 3 trials in less than a year. The second type of vaccines are the ones where the synthetic gene is spliced into a viral vector, and then the viral vector is used as a vaccine. That takes a couple of weeks more, but it's still very fast, and those vaccines are in clinical trials. The third one are the one which takes longer, basically where you take the synthetic gene, you put on a cell line, typically CHO, and you produce and purify the protein, and then you put the protein together with an adjuvant, and you make a protein-based vaccine. Next slide shows how many of those vaccines have been done, and you see the viral vectors the bottom. In the middle, you see the RNA vaccines, and at the top, you see the protein plus adjuvants. On the right, you see the scale of the neutralizing titers. In the blue arrow, you see where the neutralizing titers of convalescent patients are. And basically, as you see, the viral vectors usually induce neutralizing antibodies, which are basically at the bottom of the range of the convalescent patients. The RNA vaccines on the medium-high titer of convalescent patients, while the protein-based plus adjuvants basically induce level of neutralizing antibodies that exceed the neutralizing antibodies found in convalescent patients. So my conclusion up to yesterday, when I gave the talk about this slide, was that I'm very optimistic about the fact that we're going to have good vaccines for COVID-19. Today, I can say, given the press release of Pfizer BioNTech, I'm extremely more optimistic we're going to control this pandemic by vaccines. Next slide shows you what we know about safety. Well, what we know is that all the phase 3 trials at the bottom of the slide are performed in very large number of people. So overall, we should be reasonably confident about safety of these vaccines. What we know is also that while protein plus adjuvants at the top of the slide are well established, they've been used in hundreds of millions of people, so are pretty confident that those vaccines will perform well, will be safe. The other two technologies, the RNA and viral vectors, have never been in more than the people in clinical trials. So never been in millions of people. They are not really industrially mature. They're delivered at -70 or -20. So we'll need to see how these things will evolve. But basically, we need to be ready to monitor how these two vaccines work. But overall, I want to conclude that part about vaccines saying that very confident we're going to make vaccines, and they will allow us to basically control this pandemic. The next slide shows you the next thing that I want to talk about is passive immunization. As you know, this is a very old thing because Emil Wolbering described the serum against diphtheria in 1890, which is under 30 years ago. And today, the next slide, we have gone from sera to human monoclonal antibodies, which are largely used by the industry and by and they've been approved. There are more than 100 products approved by FDA and European Medicinal Agency, but they're mostly for cancer, autoimmunity, and inflammation. They have not been used very much for infectious diseases, mostly because until very recently, we were not able to have the extremely potent human monoclonal antibodies that would allow us to develop good products. In this slide, you see that in the field of HIV, the first monoclonal antibodies developed in the 90s were pretty bad, while over the years, we've been able to improve the technology, and now we have monoclonal antibodies that are at least 1,000 times better in terms of potency compared to the ones that we had in 1994. So thanks to this progress in technology, next slide, today we believe human monoclonal antibodies can be developed very quickly, and they can be an excellent tool. Proof of that is that the FDA just a few weeks ago approved the first drug for Ebola, and that happens to be a human monoclonal antibody. So we know that they're safe, they're effective, they can be developed. So our lab and many other labs started to, as soon as the pandemic came about, started to make human monoclonal antibodies. In our laboratory, basically, next slide, we got PBNCs from different convalescent patients. We sorted like 5,000 different memory B cells from different patients. Then we looked at the ones that were going, the B cells that were producing antibodies able to prevent the attachment of the virus to the receptor. And next slide, we ended up with approximately 450 neutral antibodies able to neutralize the virus. As you can see, the majority of the antibodies were potent, but not extremely potent because they were required at least 500 nanograms per ml for neutralization. A smaller portion required 100 nanograms per ml, but we were lucky to find 3 monoclonal antibodies at the bottom of the slide, the brown ones, that basically could neutralize the virus in vitro at less than 10 nanograms per ml in the very picomolar molarity. We selected those. We started to develop them for manufacturing. And the next slide shows a summary of the results in golden hamster. You see that the blue line, you see the hamster who gave the antibody that challenged with the virus, and you see they continue to grow without any problem. While the control, which either got nothing or a different antibody, they lose weight and they are they don't recover up to day 12. So we know the human monoclonal antibodies can work and they will be effective. We are not the only group working on that. Next slide summarizes the groups that are ready in phase 3, and some of these groups are slightly ahead of us, are reporting already positive results from their phase 2s. So conclusion, I think also human monoclonal antibodies will be an important tool to cure and prevent the infection by SARS-CoV-2. And the consequence of that is that, next slide, I believe that we're going to get back our liberty in 2021 thanks to vaccines and monoclonal antibodies. Why I'm talking about liberty? Next slide tells you why. Well, I think infectious diseases and this pandemic in particular took away our freedom, because during the lockdown, we were not able to go out, to walk, to work, to travel, to meet friends, to visit relatives. Grandparents could not visit the grandchildren. Really, it's terrible period that hopefully will be over next year with the thanks to antibodies and the vaccines. Now, next slide is going to the last topic I wanted to. So assuming that next year we control COVID-19 pandemic, are we going to learn something from other things like antimicrobial resistance? Going to be able to basically use all the learnings that our vaccines and monoclonal antibodies going to be the solution for that as well. Next slide shows you that vaccines are going to help or contribute to control antimicrobial resistance. And to say that, you see on the top antimalarials, antibiotics, and you see that when you have one drug, basically you start to use it and then you get resistance. Then you need a new one, a new one, a new one, a new one, because you continuously have resistance. And then you get at the end, well, at the bottom, at the right part of the slide, where basically you don't have more antibiotics, which is the situation we have today. And there we are in big trouble. On the green, below the bottom of the slide, you see vaccines are completely different, because vaccines, basically, they do control diseases. And so far, there is no resistance. When they control a disease, they control for a long time, some of them almost for a century. So next slide shows you the cover figure of a supplement to PNAS, where we concluded that vaccines need to be developed to control antimicrobial resistance, because there are no resistance pathogens to vaccines. The vaccines avoid disrupting the microbiome, and we know how important that is today. And vaccines are the best investment our society can make in terms of return for investment. And the question is, are vaccines for antimicrobial resistance possible? Next slide shows you my thinking about that. Here I've divided the vaccines in 4 different groups, A, B, C, and D. A are the easy vaccines, the ones that we have are easy to make. And the D is the red, is the ones that we are not able to make today. So as you can see, I put the COVID-19 in the green because I thought it was easy. And I put the antimicrobial resistance in the yellow because they are difficult, but technically we can make them. So I believe we have the technologies to make vaccines against bacteria which are resistant to antibiotics. And so the question is, what do we need on top of that? The next slide is something that shows you what is different. And I'm comparing making a vaccine for a virus and making a vaccine for bacteria. Well, from my point of view, viruses are simple. You have one genome, one target, the spike protein, and one assay, which is neutralization. Bacteria are complex. They have a large genome, they have hundreds of targets, one of them is never enough, and you don't know which assay you need to use to look at that. So basically, the effort to make vaccines for antimicrobial resistance needs to be much bigger and last longer than the one we have done for COVID-19. But it's doable, and I believe we should do it if we want to avoid the pandemic that is going, for sure is going to come after we take care of COVID-19 pandemic. So finally, the last slide is a conclusion of this part that basically that says that if we only use antibiotics, we are not going to be able to control the antibiotic resistance. The next slide shows you that if we use antibiotics and vaccines, we may do a better job. But next slide will show you that if you really want to control antimicrobial resistance, you will need vaccines, antibiotics, antibodies, and all the tools, diagnostics, all the tools that we can put together. With that, I think I want to thank you for your attention. I'm going to show you only a couple of slides very quickly. Next one, which is the team that made the antibodies for coronavirus, human monoclonal antibodies for coronavirus, and the next one, which are all the collaborators that helped me. With that, I thank you for your attention.
Thank you, Rino, for a really inspiring lecture. We have a few questions that came in. I'd like to start with a question. There has been a lot of talk and scare also about the possibility of inducing enhancing antibodies with the coronavirus vaccine. Wonder what is your thinking about enhancing antibodies in general as a risk for vaccines, and in particular for this pathogen?
Well, I'm aware of the big discussions and tensions and papers about antibody-dependent disease enhancement. I think while it's right to have a concern about it, I don't believe, at least with the data we have so far, that this is a real risk because usually we have seen ADE in the context of RSV and in the context of dengue, and when they were very poorly neutralizing antibodies. Here, the neutralizing antibodies we are inducing are extremely high with some of the vaccines, not with all of them. And so I'm really confident. On the other hand, being confident doesn't mean we need to control it. So I think we need to follow the evolving of the clinical trials and the implementation of the vaccines, because we really need to make sure that doesn't happen. So that's for these vaccines. While for the antibodies, we have the option to remove the Fc functions which are necessary to induce ADE, and that basically takes the risk away completely. Now, I have to say that most of the antibodies that are in phase 2 and 3 clinical trials are not doing that because they're confident that will not be a problem with ADE. So I think we need to continue to monitor. I'm optimistic, but we need to make sure that we are absolutely sure that we don't have that risk and we don't run that risk.
Thank you. There's a question from Jeff Cohen, who asked, the results announced today are based on a very short interval between vaccine and infection. What is known about the three technologies, RNA, vector, and protein, in terms of duration of protection, antibody duration?
Well, thank you for the question. It's very important. So first of all, there's been a lot of speculation out there saying that the vaccine response is going to be short-lived because the convalescent patient, in convalescent patient, the antibody response doesn't last that long. But now there are papers that say the contrary. I would say that we should not basically think that the vaccine response is going to be similar to the infection response, because the vaccine response will happen in the absence of infection, and we know that this virus can modulate some of the T helpers in the germinal centers, and so may decrease the response. Regarding the vaccines, the only ones for which we know that we are confident about duration is the protein plus adjuvant. I think the neutralizing antibodies will last for a long period of time. The other two, we don't have a lot of data on duration. So we will need to be, to learn as we go. On the other hand, even if I think even if the RNA vaccine that's been announced today is going to give a short-term protection, is very welcome, because I'm sure you can boost that protection with protein-based plus adjuvants and transform that into long-term protection. So yes, we don't know very much, but I'm confident we'll find the right solution at the end.
And we have a question from Bob Yarshan. My understanding is that some drug companies like Novartis have dropped or cut back antibiotic programs because of economic considerations. How can we induce them to increase effort against AMR?
I would say that absolutely true. After I left Novartis, they shut down the antibiotics discovery unit. Novartis was the last one to do it. Most of the other companies had done it before. GSK is the only one that's still doing that, mostly thanks to the good funding from the public sector. I think there are two things. One, as usual, one is the technical problem, is that it has been extremely difficult since 1984 to find a new class of antibiotics. So technically, I mean, you have seen the revolution done in vaccines, with the revolution done in many other technologies, but in the antibiotic field, we basically have not made much progress scientifically since the 1980s. So that's a big problem. And the other one is obviously there is not, I mean, the antibiotics are not a good business. So companies don't like to, and even startup, if they're doing antibiotics, basically the investors walk away. So I think if that's an important thing, we need to reverse that thing. The only way we can reverse is by more investment from the public sector. And I think I've shown you how the public sector can change things dramatically. The investment done in the COVID-19 vaccines basically allowed to shorten by 10 years the development of the vaccine. So I think if the antimicrobial resistance is really an important problem, which I believe it is, then the public sector has to come in and invest.
I'm sorry I have to cut short the discussion, question and answers because of time considerations. Thank you very much, Rino, for this excellent overview. And we need to move on, but thank you again.
Okay, thank you all. Thank you for giving me the honor to present today.
It's a pleasure to have Dr. Susan Weiss, Professor of Microbiology at the Perelman School of Medicine at UPenn, speak to us. Dr. Weiss began working on coronaviruses more than 30 years ago, back when very few were interested in these viruses, and those who were mostly folks who were in the veterinary field, as many of the coronaviruses were primarily causing significant disease in animal livestock. She was one of the first to develop animal models to study coronavirus pathogenesis, especially in the brain, look at coronavirus tropism, and the interplay of coronaviruses with the host immune response. Her investigations have enabled many of us today to have the model systems to study COVID-19 and the framework, more importantly, in which to understand the effects of these viruses on our bodies, in particular on our immune system. In 2020, Dr. Weiss became the co-director of the UPenn Center for Research into Coronaviruses and Other Emerging Viruses. We are delighted that she agreed to be with us today on such short notice too. Let's give a warm virtual welcome to Dr. Weiss. Thank you, Susan. Take it away.
Thank you. Thank you. Okay, so I just want to thank you so much for inviting me. And I'm just mentioned the one connection I have to the nice history I just taught was I actually did my postdoctoral studies with Mike Bishop in San Francisco and retroviruses. So I'm going to talk about coronaviruses, old and new. My talk is divided into two parts. The first part's going to be kind of a brief history and then the discussion of the biology of coronaviruses. And I think it's really important to understand the biology because it allows us to identify steps in the virus life cycle that may serve as targets for antivirals that may be effective against most, if not all, coronaviruses. The second part of my talk is going to be on the research from my own lab on antagonism of the innate immune response by coronaviruses. And I think this is important because it's become really obvious lately that that either a dysregulated interferon response or an insufficient interferon response has been associated with more severe coronavirus disease, both in animal models and in humans. So I will start by just introducing coronaviruses. They're a family within the nidovirus order. They're named for the nested subgenomic mRNAs generated during infection, and I'll show that later. Nido meaning nested, and here's the typical morphology in the electron microscope with the spike proteins protruding from the membrane shown here, and hence the name coronavirus or crown-like. Coronavirus particles are really rather simple. There's the really long, very long 30 kilobase RNA shown here, complex with the nucleocapsid protein in this helical nucleocapsid structure. Around the nucleocapsid is the membrane, which is derived from the host cell. And in the membrane, there are 3 viral proteins, the spike protein that have become quite famous lately. And the spike protein is important because it mediates viral entry by binding to the receptor and mediated fusion between the viral and host membranes, which I'll talk about in more detail later as well. It's an important determinant of tropism, immune response, and of virulence. These 2 other proteins, membrane protein and small membrane protein, are also inserted in the membrane, and they have important roles in replication. And some coronaviruses also encode a hemagglutinin esterase, kind of homologous to influenza HA. This is not encoded in the SARS-like viruses. Okay, I want to talk about the coronavirus timeline, really the timeline of modern coronavirus research. So way back in the 60s and 70s, we hear about OC43 and 229E. These are 2 human coronaviruses causing the common cold, although OC43 can occasionally infect the lower respiratory tract. And from that time on, there's been a group of people studying the marine coronavirus or MHV model that I've worked on for a long time. Other animal coronaviruses often studied from a vaccine perspective, and the human cold coronaviruses mentioned here. And so from the 80s and 90s, there was a lot of basic coronavirus biology done. We learned about viral entry. We learned about spike protein and its furin cleavage site. We learned about how the RNAs replicate, which I'll show a little bit later. And we learned a lot about host virus interactions. And most of this work, I should point out, was done with MHV and some of the animal viruses like infectious bronchitis virus of chickens, bovine coronavirus, and others, not so much with the human viruses. And then in 2002, the world was shocked, as were the coronavirologists, when SARS-CoV emerged in southern China, causing an epidemic that lasted only about 8 months, but really woke up the world to coronaviruses. And in the wake of that epidemic, that sort of began what I call the era of pathogenic human coronaviruses. And people looked for new coronaviruses and found really these 2, HKU1 and NL63, causing pneumonia and bronchiolitis in croup respectively. The other thing that happened in the wake of the first SARS epidemic was that we realized that bats were a reservoir for many coronaviruses as well as other viruses. And then so this epidemic was really controlled in a short period of time, mostly because the virus, when people were infected, they were sick and they could be identified and isolated in hospitals. And it mostly stayed within Asia and really, as I said, was gone in about 8 months. So then things were pretty quiet in the world of coronaviruses until 2012, when MERS, or Middle East Respiratory Syndrome virus, emerged in the Middle East, starting another epidemic, which is there are still infections going on with MERS, kind of slowly. MERS also had its reservoir in bats, but was transmitted to humans through camels. And then again, things were quiet, although there are still new MERS infections in 2020. And then, of course, at the end of 2019, SARS-CoV-2 emerged again in China, but in a different part of China, in Wuhan, causing the worldwide pandemic that obviously we're all dealing with right now. So all three of these viruses, SARS, MERS, and SARS-2, all cause severe acute respiratory disease. And just to be really clear, SARS-CoV-2 is the virus that causes coronavirus disease 2019 or COVID-19. It's analogous to HIV and AIDS. One's the virus, one's the disease. And just one other thing about the timeline. Back in 1980 was the first International Coronavirus Symposium in Würzburg, Germany. I had just gotten my lab and went to that meeting, and there were only about 60 people there, which was just about the whole field at the time. However, by 2003, the 10th meeting was held in the Netherlands just after the SARS emergence, and that meeting was completely mobbed with hundreds of people. And the 15th meeting was supposed to be met in May in 2020, eerily in the same spot in the Netherlands as the one after SARS, but that meeting had to be postponed, and hopefully we can have it next year. Coronavirus genome structure. So human coronaviruses are found in two different genera of coronaviruses, alpha and beta. And among the beta coronaviruses, there are three lineages or subgenera shown here. So the 2290 and NL63 are alpha coronaviruses. OC43 and HKU1, like the mouse hepatitis virus, are beta, lineage A beta coronaviruses or mBecoviruses. SARS-CoV is a lineage B or sarbecovirus, and MERS is a lineage C virus. All the viruses have very similar genome structure. This very long replicase gene encoding 16 proteins, non-structural proteins that I'll show you more about in a minute. And then the spike E, M, and N proteins all arrayed in the same order for all the viruses. The 3' end of the genomes encode these small open reading frames that are numbered here. And these are often contain antagonists of the innate immune response that I'm going to talk about in the second part of the talk. So remember some of these small numbered ORFs. So after SARS-2 emerged, it became clear quite quickly that it was within the sarbecovirus, a lineage B beta coronavirus, with a similar, but not that similar sequence to SARS-1. Here's a model of coronavirus replication. It's very simple. It's missing a lot of the details we know now, but it makes the main point. So the virus, the spike attaches to its receptor via the spike protein here. There's a fusion between the viral and host membranes, and the viral nucleocapsid is released into the cytoplasm. The RNA is then translated into the replicase proteins, which then replicate the RNA and produce these subgenomic mRNAs, which we'll give more details in a minute. The proteins are translated. This membrane proteins then are inserted into the intracellular membranes shown here. The nucleocapsid is then assembled by the nucleocapsid protein and the new genome RNA, and the nucleocapsids bud into the endoplasmic reticulum Golgi intermediate compartment shown here. They're then seen, mature virus particles are then seen in vesicles in which they're transported to the surface and released into the extracellular medium shown here. The spike protein for some viruses, well, for all viruses, it's inserted on the membrane, and for some coronaviruses, it can mediate cell-to-cell fusion. So this spike can mediate fusion with an adjacent cell. So I'm going to talk about several of these steps in a little more detail: entry, genome RNA, and translation of the conserved replicase proteins. So coronaviruses enter cells by two routes of entry. The first entry is at a direct plasma membrane route. And in this route, the spike protein recognizes its receptor, ACE2, angiotensin converting enzyme in the case of SARS-2, but it's of course different receptors for different viruses. It's cleaved by a protein. The spike protein is then cleaved by a protease. Again, in the case of SARS, in respiratory AT2 cells, it's TMPRSS2, but this, of course, also can be very variable. Once the spike is cleaved, it's activated to mediate a fusion between the viral membrane and the cell membrane, and the nucleocapsid is released into the cell. By the other method, the endosomal route, the spike protein again sees its receptor. The particle is endocytosed into an endosome shown here. And in the low pH environment, The spike protein is cleaved by cathepsin, a cathepsin-like enzyme, which then mediates, which activates the spike again to mediate fusion between the viral and host membranes, and the nucleocapsid is released again into the cell. So what determines which route is used? It's a combination of the proteases available on the host membrane shown here, or in the endosome, or even in the intracellular, in the Golgi, the furin enzyme is also partially activates the spike protein. So some virus particles are released already partially ready to mediate fusion. And I just want to give an example from MHV of also how the sequence of the spike protein also plays a major role in which of these routes is used. So these are several MHV strains I'm going to talk about. So all spike proteins, coronavirus spike proteins, have two subunits, S1 and S2, and they have to be cleaved in two places to activate fusion. The S1, S2 boundary sometimes has a cleavage site for furin, which is that intracellular enzyme, and the S2 prime site over here it also has to be cleaved in order to expose a fusion peptide to mediate this viral cell fusion. So JHM strain, for example, has a really very basic furin site that's well cleaved, and that virus goes in only by the plasma membrane route. The MHV2 strain has a site unable to be cleaved, and it goes in by the endosomal route. So both of those cleavages can occur by cathepsin in the endosome. And then A59 is partially cleaved, and it actually enters cells by both routes. So all of these strains of virus are perfectly pathogenic, but they just enter cells by different routes. And why is that important? I think it's important because it tells us something about what enzymes or what inhibitors are going to be useful. For example, chloroquine or a cathepsin inhibitor will be more useful against a virus entering by this pathway. Chloroquine raises the pH of the endosome and cathepsin inhibits the enzyme, whereas a protease inhibitor like Camostat that inhibits TMPRSS2 would only be effective against viruses entering by this pathway. And so the proteases available, it really going to depend on the cell type, and the cleavage of the spike protein is also going to depend on the virus type. So there's a lot of variables here, and it's a little bit difficult to predict which route is going to be predominantly used and therefore which inhibitors are going to be most successful. Okay, transcription of coronavirus mRNAs. So here's the genome again. It's got a 5' cap and this 100 or so nucleotide leader sequence, 3' polyadenylate. And it's got these transcriptional regulation sequences here that are kind of like promoters that denote the beginning of each mRNA. So during infection, a polymerase is first translated from the plus strand genome. It then transcribes a minus strand or negative sense antigenome, and then transcribes back more positive strand genome. And that's how the replication occurs. At the same time, a set of negative sense subgenomic RNAs are transcribed. So the transcription begins again at the 3' end of the genome. And once it gets to this first TRS, it can then jump over to this TRS here and transcribe the leader sequence. And so we get a negative strand RNA anti-messenger RNA with the leader sequence. And then at some frequency, it may skip this TRS and go to this one and jump to the 5' end again. And so we generate a whole set of these negative sense RNAs, which are then copied back into mRNAs. So the blue are the mRNAs. So then each of these mRNAs is translated usually into one protein from its 5' open reading frame shown here, occasionally two proteins. This is actually a model for MHV, but it's really similar for all the coronaviruses. So this is pretty simple. The 5' end is more complex. So there was open reading frame ORF1A and ORF1B that encodes these 16 replicase proteins. And the way this is translated is the ribosomes translate down open reading frame 1A here. And in most of the cases, they terminate translation here, but there's a pseudoknot, a structure in the genome RNA that causes a translational sort of slowdown and a slippery sequence causing a frame shift. So there's a -1 frame shift, and then at about 20% of the time, the ribosomes continue to the end of ORF1A, 1B. There are 2 proteases encoded in ORF1A, and they're as co-translationally cleaving these long polypeptides into the mature non-structural proteins. And I want to take a little time to talk about those proteins. So there are, as I said, 16 non-structural proteins in ORF1A and ORF1B. Just to mention some of them, or all of them, NSP3 encodes a papain-like protease that processes at these 2 sites. The 3C-like protease, a main protease, processes at the rest of these cleavage sites. And then the RNA to RNA polymerase is encoded in ORF1B and NSP12, and that does replication and transcription of the RNAs. And remdesivir, that we've all heard about, is directed against this RNA-dependent RNA polymerase. And there have been some proteases tried to use against the 3C-like protease. These are HIV proteases. They have not been really successful against SARS-CoV-2 because this is really a different protease from the HIV enzyme. So in addition, so in summary, proteases process replicase proteins, and the RNA-dependent RNA polymerase replicates genome and transcribes RNA. And these are all good targets, potential targets for antivirals, and they're all highly conserved among all the coronaviruses. This is just to acknowledge several people that have been working away on inhibitors way before SARS-CoV-2, and they continue to do so now. So in addition, there are all these other non-structural proteins encoding activities that are possible drug targets. We have several. The primase, the activator, and the helicase are all part of the replication machinery. There's several enzymes involved in RNA capping, which protects the viral RNA from the host innate responses. And this endo-U is an important enzyme that reduces the accumulation of double-stranded RNA that I'm going to talk about in the second part. That is again one of the which induces a host antiviral response. And there are also a couple of other host antagonist activities encoded in NSP1. This shuts down host translation, and in NSP3, a deubiquitinase and a macrodomain. So there are enzymes that promote synthesis and stability of RNA, capping of 5' ends, protection from host cell sensors, and interferon response. And again, all of these are highly conserved among the coronaviruses and potential drug targets. And just to show one example, the NSP13 helicase, another conserved protein, is shown here. This is from a study by Stefan Serfianos that we collaborated with him years ago after the MERS epidemic. So here's a structure of NSP13 helicase and a predicted structure of the MERS and MHV enzymes. And we can see that this compound fits into the same bonding pocket of all 3 structures. And this same inhibitor is effective against SARS, MERS, and MHV, shown here, replication as a function of increasing drug concentration. So in summary of this part, there are many conserved features of coronaviruses, subgenomic mRNAs, non-continuous RNA synthesis, which, by the way, allows the high rate of recombination among coronaviruses, that they already are able to jump templates, so to speak. There's this ORF1A, 1B frame shifting, the proteases, the conserved proteases, the RDRP and RNA modifying enzymes, and the further activities. So all of these are potentials for interference by drugs potentially. So I'm going to switch here and now talk about coronavirus antagonism of double-stranded induced antiviral pathways. This has been a subject of research in my lab for quite a while now. And this is, so I'm going to first describe the pathways that we focus on. Then I'm going to talk a little bit about or summarize the work we've done for MHV and on MERS, and then talk about our current work on SARS-CoV-2. So the double-strand RNA-induced pathways, so coronaviruses and other RNA viruses produce viral double-stranded RNA in replicating their genomes and their messenger RNAs that I just showed you. And the viral double-stranded RNA is sensed by several sensors in the cell, and as it is noticed as a danger signal, and it induces this antiviral pathway. So the kind of canonical interferon pathway is shown here. viral double-stranded RNA. In the case of coronaviruses, is recognized by MDA5 and not RIG-I. It signals through MAVs to induce interferons, type 1 and type 3, and then signals through STATs, phosphorylating STATs, to produce interferon-stimulated genes and cytokine responses. But at the same time, oligoadenylate synthetases synthesize 2,5-A, a small oligoadenylate that activates RNase L to an RNase L will cleave host and viral single-stranded RNA. It also leads to cell death and inflammation. And we've worked on this pathway quite a lot in our lab. And the third pathway is PKR, protein kinase R, that when sensing double-stranded RNA, autophosphorylates, then phosphorylates EIF2 alpha, which is a translation initiation factor, and that leads to translational shutdown, cell death, and inflammatory responses. And I just want to point out also that interferon, that both OASs and PKR are interferon stimulated genes. So these pathways can actually be boosted by the interferon pathway, but they can also be activated completely independently, which I'll show you later. And I think it's quite important to remember that. Okay, so beta coronaviruses so MHV encodes this NS2 protein that we've worked on quite a lot. It's a 2H phosphoesterase, and I'll show you in a minute what it does. And then MERS coronaviruses encodes an NS4A, which is a double-strand RNA binding protein, and NS4B that also is a 2H phosphoesterase, but has a nuclear localization signal on it. So it seems to have expanded properties compared to the MHV enzyme. Okay, so these viruses pretty much shut down these pathways quite effectively. So the MHV NS2 and MERS NS4B have 2' 5' phosphoesterase diesterase activity that specifically cleaves this molecule and shuts down this pathway really completely. And then NS4A is a double-strand RNA binding protein that has an effect of really reducing activation of all of these pathways. And then on top of that, NSP15, the endonuclease that's part of the ORF1, encoded in ORF1b for all of these viruses, also prevents the accumulation of double-stranded RNA by a really interesting mechanism. It actually cleaves the genome RNA, so sacrifices accumulation of more genome to presumably to reduce the accumulation of double-stranded RNA. So these pathways are very effectively shut down by these two. This is a lineage A and lineage C beta coronavirus. So we wanted to know about SARS. So we looked in several different cell types, relevant cell types. We also used MERS as a kind of control because we know that it shuts down these pathways quite effectively. And we also use Synbis virus, a completely unrelated virus, as a positive control because it activates these pathways all quite robustly. So the first cell types we looked at were nasal epithelial cells. These are primary cells recovered from patients by a collaborator. And we had cells from 4 different donors, and we can see that both SARS and MERS effectively infect these cells, the red being stained for viral nucleocapsid protein and the green for cilia. Some of these cells are ciliated, and we can see replication is pretty efficient. SARS replicates over a long period of time, shown here in the black from 4 different patients. MERS seems to clear out more quickly, shown in the green, and not to replicate quite as well. And we can just speculate that SARS may replicate longer in the nose, just from what it does in these cells. We looked at induction of interferons and several interferon genes and 1 chemokine, and we see that there's not very robust induction of any of either interferon or interferon stimulated genes in these cell types. And interestingly, we found very low levels of ACE2 receptor. The receptor needs to be there, but not at a very high level, which is something we noticed when working with MHV. We then looked in pluripotent stem cell induced derived alveolar type 2 cells, shown here. These cells are characterized by expression from the surfactant protein C promoter, shown here, the T tomato, just to define the cell type. The SARS nucleocapsid is stained here in green, and here's the merge. So again, the virus replicates quite well. It replicates to about 6 logs of virus by 2 days post-infection. That's really quite efficient. We looked at induction of interferon and ISG messenger RNA, again, compared here to SINBIS virus. And we see very weak induction of interferons compared to SINBIS, but much better induction of interferon stimulated genes. And again, the chemokine CXCL8, which is important in defense in the nose, is really barely induced. And we looked at RNA cell activation this way. We can see, we look at 28 and 18S RNA is degraded in the presence of SINBIS virus because it's activating this pathway, but we don't really see much activation for SARS-CoV-2 in this cell type. And here we saw that SARS actually, SARS-CoV-2 does activate the PKR pathway. Here's phosphorylated PKR, phosphorylated EIF2 alpha. And again, very little MDA5, which might explain the poor interferon induction, and undetectable levels of ACE2, although again, as we know, it must be there. We also looked at cardiomyocytes as there's cardiovascular complications from infections of SARS. And those cells are characterized by expression of C-troponin T, shown here. Here's the SARS-CoV and nucleocapsid staining shown here. And here's the merge. So in this cell type, we see cell-to-cell fusion. So there's definite cytopathic effect from SARS, although we didn't see that in the other two cell types. Virus again replicates to pretty high titer, 6 logs of virus, as we saw in the alveolar type 2 cells. Again, very, very weak induction of interferons compared to SINBIS virus, and really not very much interferon stimulated genes. Again, in this cell type, we saw no activation of RNA cell, even by SINBIS. And when we did a Western blot here, we can see that we have, again, activation of PKR pathway. Here's phosphorylated PKR and PIF2 alpha compared to SINBIS. And again, we saw almost no RNA cell expressed, no MDA5 and no MAV. So we speculate that this cell type has very low innate gene expression, and that may be to protect, although it didn't seem to get protected very well from cytopathic effect. Okay, so then we used 2 cell lines that we could more mechanistically look at these pathways. CalU3 cells, where we could carry out MERS and SARS infections in the same cell type, and A549 cells expressing ACE2, where we could actually genetically manipulate the cells. So virus replicates in both of these cell types quite well. This is Viro cells, which we use to propagate the virus. That's shown in the green here, nucleocapsid staining. All the virus produces double-stranded RNA, which you can see is usually it's perinuclear attached to the endoplasmic reticulum. And we see cell-to-cell, very robust cell-to-cell fusion in these 2 cell types. That's why it's difficult to really localize the double-stranded RNA. And there's less cell fusion in the Viro cells. The virus replicates quite well in A549 ACE2 cells shown here, and along with the Viro cells. And this shows the replication of SARS compared to MERS, and a MERS mutant that's lacking the 2 interferon antagonists I told you about. So in CalU3 cells, SARS replicates, MERS replicates better, and the MERS mutant is somewhat attenuated, as would be expected for a virus lacking its interferon antagonists. And both of these cell types have quite robust innate interferon responses. So we showed here that we found here comparing, we looked at SARS-CoV induction of interferon beta and lambda at 24 and 48 hours post-infection. Very poor induction compared to SINBIS. Again, poor induction of interferon stimulated genes, although it's getting better at 48 hours compared to 24, and really no induction of the chemokine. When we looked in CalU3 cells, we can compare to MERS. We were sort of surprised to see that SARS actually induces more interferon, both type 1 and type 3, than MERS, shown here, but more similar or approaching the level of the MERS mutant lacking the interferon antagonists. And when we looked at interferon stimulated genes, we saw here that in fact, SARS was quite good at inducing interferon stimulated genes compared to MERS, and even close in level in some cases to the MERS mutant that was lacking the antagonists, and very little induction of CXCL8. And when we looked at, we found that STAT1 was actually phosphorylated by SARS-CoV-2 and also by the MERS-CoV-2 mutant, but not by MERS wild type. And again, yeah, and all the viruses replicate quite well. So it looked like SARS-CoV-2 is behaving more like the MERS mutant lacking antagonists rather than MERS itself. And that was continued the pattern when we looked at RNA cell activation. We can see that SARS-CoV-2 produces degradation of 28 and 18S ribosomal RNA, as does SYNBIS here. And when we looked in the Cal-U3 cells, we could see again that SARS produces degradation, activates RNA cell, as does the MERS mutant, but not the MERS wild type. So again, it's acting like the MERS mutant, and the same thing for PKR activation. So we see that SARS activates PKR in both of these cell types, and as does the MERS mutant, but not MERS wild type, shown here. So the last thing we did was we produced a bunch of knockout cells using CRISPR-Cas9, knocked out either MAVs, RNase-L, or PKR, and this is a Western just to validate the knockout of each of these cells, of each of these proteins, each of these genes. We then looked at replication, and we found that in the absence of RNase-L, we see here increased replication of SARS-CoV-2. There was really no effect of knocking out MAVs or PKR. And this is just, here we looked at the cell death basically in these knockout cells. And we see that cells, the wild type cells shown here, that RNA cell knockout cells produce more infection, produces more cell death, which agrees with the better replication of RNA. We didn't see a whole lot of difference between MAVs, PKR, and wild type cells. And then finally, this is something that we thought is really important in our lab, which is that we can see that SARS and SYNBIS both activate RNA cell perfectly well in the absence of MAVs, which means in the presence of really minimal interferon induction. So RNA cell is a really independent pathway from interferon and can be activated even if the interferon pathways are shut down. And the same thing is true for PKR. We see that PKR is well activated. Here's PPKR and PEIF2 alpha in the absence of MAV shown here. And actually in the absence of RNase as well. And interestingly, we see PEIF2 alpha phosphorylation even in the absence of PKR. We believe this is promoted by PERK and the interferon and the unfolded protein response. That was something that we're asking about now. So in summary, it looks like SARS-CoV-2 pretty much reduces activation of this pathway, and that may be through NSP15 that I mentioned before, and through ORF6, which has been shown for SARS-1, and there's a little bit of data on SARS-2 that it prevents translocation of STAT1. And ORF3b has been promoted as an interferon antagonist, but this hasn't really been validated with virus, but it's a suggestion. So this pathway is pretty well shut down, but these two pathways are activated during SARS-CoV-2 infection. So in conclusion, coronaviruses enter cells by two pathways. The pathway used depends on spike protein cleavage sites and host surface and intracellular proteases. Coronaviruses express numerous conserved host antagonists in the replicase locus, often enzymes. These are potential targets for antivirals that may be effective against most, if not all, coronaviruses. So it's really important to keep deriving antivirals, which will be much quicker fixes than vaccines, but and lineage A and lineage C beta coronaviruses express accessory proteins that contribute to antagonism of activation of all RNA cell, PKR, and interferon signaling pathways. And finally, SARS-CoV-2, like MERS, weakly induces interferon. However, SARS lacks the phosphodiesterase and a DNA double-stranded RNA binding protein, activates RNA cell, promotes phosphorylation of PKR, EIF2 alpha, and STAT1. These data suggest that SARS is not as adept as MERS in shutting down the double-stranded induced antiviral pathways, and we wonder whether this may contribute to the unique pathogenesis of this virus. And finally, I want to acknowledge my lab shown here, and they're working really hard under very stressful conditions to work on the virus. And our collaborators, Bob Silverman's been a collaborator for years on everything to do with RNA cell, Ralph Barrick on the mutant viruses, and our colleagues at Penn providing the iPSC-derived cells and our funding. So I will stop here.
Thank you so much, Susan, for a lovely talk. It was very informative. We have a few questions, so I will relay the questions to you. The first one is, what about bromohexine as an inhibitor of TMPRSS2?
Oh, I don't know anything about that inhibitor.
Okay.
I'm sorry.
And the other question is, don't you think that the actual drug concentration in lung is limiting the efficacy of some of them? For example, lopinavir, ritonavir, that do not reach an inhibitory concentration in lungs.
Wow, I don't really know that, but I don't think they work against the virus itself, even in tissue culture, but I'm not sure about that.
Okay. The other question is, in terms of, based on your understanding and your years of experience looking at the replication of these coronaviruses, how do you compare these viruses in terms of genetic variability? You mentioned, for instance, template switching compared to other positive strand RNA viruses.
Okay,
like,
so
hepatitis C, for instance, as we were
okay,
mentioning this morning.
so the really striking thing about coronaviruses that's unique to them is the proofreading enzyme NSP14. So they really have a reduced rate of mutation, and in general they don't change that much over time. The only thing we hear about now is this D to G mutation in the spike, the only thing that seems to have been arisen. As far as recombination, they do recombine at a pretty high frequency, but I'm not clear on how closely related they have to be to recombine. Like, I think for certain, 2 SARS viruses might recombine, but I don't think a SARS and a MERS would recombine. So I would say in general, I wouldn't say they're more prone to change than other RNA viruses, probably less so because of the proofreading enzyme.
Okay, there's a question from David Thomas. Can you speculate on the pharmacologic use of type 1 or type 3 interferons?
Well, you know, there's been a lot of speculation about that. I personally, I know that there's a bunch of studies now that say that interferon antibodies that people have interferon antibodies, and that's associated with severe disease, and that inability to mount a good type 1 response is so, and the same thing with, there's also a study that associates OAS genes with a poor response. So I do think that people have certainly speculated about using type 1 interferons. So it seems reasonable to me. I think there are clinical studies going on as well.
Okay, there's a Shintao who will ask his or her question. Shintao, go ahead, you can speak. Unmute, I unmuted you. Shintao, are you able to speak? He raised his hand, but okay. Final question. In terms of what you're seeing with the innate immune response, do you see an effect of dose response of the virus?
You mean in terms of the innate immune response?
Yeah, in terms of multiplicity of infection, have you looked at that?
We have not looked at that specifically. I'm just trying to think from the other viruses what we know. I don't think it's like a linear relationship between how much you put in. Yeah, I mean, I'm sure there's some relationship, but I can't tell you exactly what it is.
Okay.
I'm sure a higher multiplicity is going to activate more.
Okay, and Malcolm Martin has a question. Malcolm?
Did you hear me?
Yes, we can hear you. We can hear you.
I was interested in the answer to the question about the multiplicity in view of what's known about wearing masks and reducing the amount of virus exhaled, by almost 100-fold.
Yeah, you sort of can look at it 2 ways. If you get more virus, you may get more innate response, but you also may get more replication. So it's kind of a tricky relationship, I think.
Okay. I don't know if there's any other questions. I think we can move on.
Hello, everyone. I'm Joe Marco Trigiano. I am a senior investigator here at NIAID, and it is my privilege to introduce Marshall Bloom as at today's symposium. Marshall is a colleague who serves as the chief of biology, vector-borne viruses section at the Laboratory of Virology at Rocky Mountain Labs in Hamilton, Montana. To those of you that don't know, Rocky Mountain Labs is part of the NIAID, National Institutes of Health. It's located in beautiful Hamilton. Marshall is a well-recognized expert in pathogenesis of tick-borne flaviviruses, persistent infections, and parvoviruses. Since the early 2000s, Dr. Bloom has studied the biology of tick-borne flaviviruses in mammalians and arthropod systems, making many considerable contributions in the area of viral pathogenesis, persistence, ultrastructural gene expression, and the interaction between the virus and the host. Marshall has been an author of numerous scientific articles, book chapters. He is a fellow of the American Academy of Microbiology, American Society of Microbiology, the American Association for the Advancement of Sciences, and the American Society of Virology. He has served on many organization committees over the years. He's also played a very major role in NIAID, especially out at Rocky Mountain Labs, where he has done a lot in terms of facility and infrastructure building, especially the integrated research facility, which houses NIAID's BSL-4 containment level laboratory. He is obviously considered an expert on biosafety and containment. And so with that, he worked with Dr. Salzman, and I will let him give an overview on that interaction. So Marshall, welcome virtually. I hope all is well out there.
Thanks, Joe. And I'm not going to use any slides for reasons that I think are becoming all too apparent. So I first want to thank the organizers for extending me the privilege of delivering a brief tribute at the 2020 Norman Salzman Memorial Symposium for Virology. And a special thanks to Lenore, with whom I've not been in touch for way too many years, and also to Joe for the kind introduction. Most of the awardees and some of the speakers are likely unfamiliar with the enormous impact that Norman had on the field of virology and molecular biology. So before contributing a few personal recollections, I want to take a minute and review his remarkable career and impact in the field, which have been alluded to already. Norman was born in New York in 1926 and attended City College before moving to earn a PhD in 1953 at the University of Illinois. He spent the next 33 years at NIH. Before becoming chief of NIAID's Laboratory of Biology of Viruses in 1967, Norman was a section chief in that lab. And his primary focus was on DNA tumor viruses like SV40. And as was noted, he contributed some of the early seminal work on SV40 DNA replication. He was the LBV lab chief until he retired from NIH in 1986, whence he moved to Georgetown to lead efforts in retrovirology. He maintained that focus when he moved to the National Cancer Institute's Frederick Cancer Research and Development Center in 1994, where he was chief of molecular retrovirology until his untimely death in 1997. Norman, along with Bob Wagner and Lloyd Kozloff, were the founding editors of the Journal of Virology in 1966. And that was while Norman was still a section chief. And a tabulation of department chairs, journal editors, NIAID lab chiefs, and other prominent virologists, both in the US and abroad, would really read like the Laboratory of Biology Viruses across from the elevator on the third floor of Building 5. And a few are Bernie Moss and Mal Martin, who were section chiefs in LBV, went on to lead trend-setting NIAID intramural labs that continue to set the standards and break new ground for a number of important virus families, notably pox viruses and HIV, and more recently, flaviviruses. Laboratory of Biology of Viruses, or LBV section chief Jim Rose, while in that lab, worked out how a short single-stranded negative sense DNA virus like adeno-associated parvovirus or AAV, used a hairpin of inverted terminal repeats to prime double-stranded templates for replication, transcription, and protein expression. Ken Burns became chair at Hopkins, NYU, and finally dean at Florida. Ken demonstrated that the benign AAV integrated into the mammalian genomes, thus setting in motion some of the most successful gene therapy strategies around. Claude Guérin did RNA DNA heteroduplex electron microscopy on that old Siemens microscope EM that was critical in confirming RNA splicing, and held oinophilia workshops in that same scope room to teach a lot of us how to discriminate good from bad vintages. Claude later moved to Rocky Mountain Labs, where he founded the microscopy program now responsible for so many of the stunning images of COVID-19 and other infectious agents. George Corey had a dazzling career looking at viral gene expression at NCI that was cut short by cancer at age 1943. Steve Strauss went on to be the first director of the National Center for Complementary and Alternative Medicine before his career was also cut short by cancer at age 60. Peter Halley went on from NCI to chair the experimental pathology department at Harvard, where he really established the groundwork for papillomavirus research. Mark Israel presented a lab meeting one week that I attended in the mid-1970s about some new indecipherable technique called gene cloning. And finally, my fellow LBV postdoc in Jim Rose's lab, Mark Buller, became a leading figure in the area of poxvirus pathogenesis and biodefense at St. Louis U until his fatal cycling accident a few years ago. What a remarkable legacy and impact from a single intramural lab under a single laboratory chief. And these are just the people I could readily occur, leaving out people like George Freed and of course, Mike Bishop. And what insight and scope Norman had to recognize that the pioneering molecular virology work in SV40 would have application to so many other virus families. And finally, I'd like to add a short reminiscence of my own, hastening to recall Mark Twain, who's reputed to have said, "As I got older, my memory improved. When I was young, I could easily remember what had happened, but as I got older, I could not only remember what had happened, I could also remember things that hadn't happened." So after, like Harvey, being a yellow beret, working on Lucian mink disease at RML from 1972 to 1975, I was sent to spend a couple of years in Bethesda to learn some real virology. I met with Wally Rowe, Bob Chanik, Shelley Wolf, Ray Dolan, Roger Cole, Norman, and a few others. I was drawn to Norman's LBV for a couple of reasons. I had a hunch that the virus I was working on was going to be a parvovirus, whatever that was. And so Jim Rose's group was the obvious place to go, but I was also attracted by Mal Martin's zany intellect and Bernie, an incredible sense of humor, Bernie Moss's quiet and fierce intellect, and the incredibly clean molecular virology that Norman's group was doing. LBV was clearly an exciting environment, and that's where I started in 1975, June. I quickly realized that I'd accidentally landed in exactly the right place at exactly the right time to learn virology and also to forge lifelong connections with people destined to be leaders in the field. Although there were electricity and even an ultracentrifuge in Montana in the 1970s, LBV was the first place I ever saw a biosafety cabinet, spinner cultures and water baths, and learned that there was a way to pipette virus without using your mouth. The cold room on the third floor of Building 5 usually held an astonishing amount of P32, but that was kept on a different shelf from where Harry Belcher put the fresh Thanksgiving turkeys that he got from a friend in Frederick. Norman, Jim Rose, and the others patiently taught me filter hybridization, cesium chloride gradient centrifugation, scintillation counting, the arcana of COT curves, plaque assays, and TCID50 measurements. And Norman also bought one of the first word processing machines, an IBM MAG Selectric typewriter that took up about a quarter of the LBV office and used floppy disks about the size of an umbrella. This was really a boon for writing papers since few of us could type. I returned to RML in early '77 and tried to make all this work in Montana. I made it a point to come back to Norman's lab every few years to learn things like M13 cloning and sequencing or vaccinia-based gene expression, and most importantly, to renew my friendship with Norman and others and get insight into the latest trends. The time I spent in Norman's LBV, the things I learned, and most importantly, the people I met, gave me the confidence to start a molecular virology program at RML, and then to apply molecular approaches to viral pathogenesis, first in parvoviruses and then in tick-borne viruses. LBV with ample justification, but he gradually accepted me into the LBV fold. In those days, back in the 70s and 80s, there was a self-service store in the basement of Building 10, and the control that Norman exerted over the charge card was far stricter than the inventory controls over the P32. And the final time I came out to LBV for an extended visit in the 1980s, Norman told me that if I needed anything from the self-service store, including a complete set of pipette man pipetters, I could use the credit card. That was when I knew that I had truly passed muster in Norman's eyes. I last saw Norman at the ASV conference at Montana State University in Bozeman in July 1997, about 6 months before his untimely death. I was on the committee for that conference, and my younger son, Seth, had tagged along to work with an ecologist there. On the final night, Mark Young had arranged a barbecue under a big tent. Norman called Seth and me over to join the minion with him, Ken Burns, Ron Luftig, and a few other AKs. We sat there swapping memories and lies while Seth basked in the accumulated erudition and Talmudic level disputation. Norman was extremely kind to Seth, as he was to all trainees, and offered him good advice about careers and schools. Seth remembers that evening 23 years later. So I want to end this brief tribute to the founding figure of molecular virology we're honoring today. And I again want to thank Joe, the organizers, and especially Lenore. It's a real pleasure to share some memories of Norman with younger scientists and trainees. As Barbara Tuchman says in the foreword to her book, The Proud Tower, we should remember those that went before. Thanks very much.
Thank you, Marshall. Mrs. Salzman is next to present the award, the first award. Mrs. Salzman.
Good morning and welcome. I am honored to be here today with this illustrious group of scientists to present the Norman P. Salzman Memorial Awards in Basic and Clinical Virology. Joining us today are more people than we have had in previous years at this event. There are 400 or more registrants and speakers. Due to the coronavirus precautions, it was necessary to shift the event to a Zoom webinar. This change allows us to reach people from all over the United States, Europe, and other countries. During these unsettling and troubling times of the coronavirus pandemic, it is so uplifting to hear about Dr. Kizzmekia Corbett at NIH, the postdoctoral award recipient today, and her career path, scholarships, and current work in developing a leading candidate vaccine we so desperately need. For me personally, this event is very special because it is both a testament to Dr. Salzman's devotion to his scientific and mentoring work and to others who worked with him to continue the highest standards for original and meritorious work in all scientific explorations. More about Dr. Corbett in a few minutes, because this is my time to say thank you. Slide, please. To the Organizing Committee, Nihal Altan-Bonet, Chair, Paolo Lusso, Co-Chair, Alan Ryan, Gabriel Para, and Joseph Marco Trigiano. Also to the Outstanding Selection Committee and other staff, Terry Burdett, NIH, Janelle Lewis and Katie Robinson, Meetings and Events Planning, Foundation for the NIH. More information about the committees can be found on the digital program. Now to Harvey Alter, this year's Nobel laureate in medicine or physics. Congratulations. Thank you for your kind words. How exciting that you are sharing your words of wisdom, your sense of humor, and your roadmap to success. You have to take the stairs. Our gratitude to Maria Freire, President and CEO of the Foundation for the NIH, the continuing sponsorship of the Foundation for NIH is paramount to this program continuing. Thank you to the NIH Office of Intramural Research and the Virology Interest Group at NIH for their support in making the symposium a success each year. To Marshall Bloom for his memories with Dr. Salzman when Norman was chief of LBV, the Laboratory of Biology of Viruses. What incredible memories you have brought back, Marshall, and also for your review of scientific discoveries at LBV and the purchase of pipettes, founding and saying that Norman was the founding father of molecular biology. To all the speakers from East to the West Coast and Siena, Italy, I thank you all. Dr. Rino Rappuoli, Dr. Susan Weiss, Dr. Rebecca Dutch, Dr. Richard Kuhn, Dr. David Wang, and Dr. Jesse Bloom. Some of the speakers will be this afternoon. Slide. It is now my honor to present the Norman P. Salzman Postdoctoral Fellow Memorial Award to Kizzmekia Corbett and her mentor, Dr. Barney Graham, Deputy Director and Chief of the Viral Pathogenesis Library, NIAID Vaccine Research Center. With this award comes a check of $2,500 to Dr. Corbett and a plaque, and also a plaque for Dr. Graham. The check and plaques will be sent from the Salzman Fund at the Foundation for the NIH. Dr. Corbett is a research fellow where she works under the direction of Dr. Barney Graham Virus Research Center. Her research activities with a variety of viruses cover a period of 15 years. A viral immunologist by training, her current work involves a leading candidate vaccine against the virus that causes COVID-19, and we've heard a lot about those vaccines today. Although I am not on the selection committee, they have asked me to say that Dr. Corbett was chosen for the Salzman Award for her seminal contributions to the development of a SARS-CoV-2 vaccine. Congratulations to both Kizzmekia Corbett and Dr. Barney Graham. I am also pleased to present the first graduate student award for the Salzman Memorial Awards to Victoria Avanzato and her mentors, Dr. Vincent Munster, Chief, Virus Ecology Section, NIAID, Rocky Mountain Laboratories, and her second mentor, Dr. Thomas Bowden, MRC, Division of Structural Biology, Wellcome Center for Human Genetics, University of Oxford, NIH Oxford Cambridge Scholars Program. With this award comes a check to Victoria Avanzato for $1,000 and a plaque, and also plaques to Dr. Munster and Dr. Bowden. The check and plaques will be sent. The selection committee asked me to say that Victoria Avanzato was chosen for her identification of a key site of Nipah virus vulnerability. Victoria is currently pursuing her MD and PhD through the NIH Oxford Cambridge Scholars Program and Emory University School of Medicine. After the completion of her MD, PhD training, Victoria plans to become an infectious disease physician while continuing her research interest in viral pathogens and global health. Congratulations to Victoria and her mentors, Dr. Vincent Munster and Dr. Thomas Bowden. I am pleased to tell you that our first Salzman Award winner in 1999 was Joanna Schisler, her Bernard Moss, her mentor, at a time when there were fewer women scientists than there are now. I am happy to report that there have been 10 women since 1999 who have received the Salzman Memorial Awards. Thanks to all of you. And now to Dr. Kizzmekia Corbett.
Thank you so much for that kind introduction. So it is an honor to get the Salzman Award because, full disclosure, I've been here at the NIH since 2014, and I have applied for the Salzman every year since then, especially after some of the seminal work that I'll be presenting to you today. So it's very exciting to be able to present this work to you and to talk to you about the work that we've been doing for the last several years around prototype pathogen preparedness. So I'm Kizzmekia Corbett, and I am a research fellow from Dr. Barney Graham's laboratory at the Vaccine Research Center. And I study coronaviruses. And coronaviruses are a large family of viruses that pose for pandemic potential. And so this family tree on the left side of your screen shows the alpha and beta coronaviruses, which are most relevant for humans. As you may well know, there are 4 coronaviruses, namely HKU1, OC43, et cetera, that can infect humans in what we call an endemic fashion. So seasonally, when one might get the mild sniffles during flu season, it is about 20% chance that it is caused by an endemic coronavirus. However, sometimes coronaviruses jump from their animal reservoirs and cause devastating disease around the world. There has been several cases of this happening, the first of which happened over a decade ago with the SARS-CoV-1 epidemic, which tallied in over 8,000 cases worldwide. The second of which started with the MERS epidemic around the time that I joined the Vaccine Research Center in 2013, which in the end tallied around 2,000 cases worldwide. So the idea that coronaviruses can jump from their animal reservoirs and pose as a human threat for emergence is something that we knew based on history, but also something that science continued to prove over and over, largely with a body of work that comes from Ralph Barrick's laboratory at the University of North Carolina, where they deemed SARS-like viruses, such as WIV1, as poised. So with that, and knowing that there was this imminence for emergence of coronaviruses into humans, we knew that we needed to have a vaccine response essentially prepared shall a pandemic occur.
A global race to find treatments for the sick and a vaccine to protect the healthy.
This ends with most or all of us being immune to this virus, and ideally that's through a vaccine.
can solutions that usually take years
We are trying to move as fast as we possibly can
be found in a matter of months?
It is an extremely revolutionary approach to get into a phase 1 clinical trial 66 days after the sequence of a virus is released is a world record.
And setting that world record, so to speak, came by way of what we call the prototype pathogen approach to pandemic preparedness. So this approach is something that my mentor, Dr. Graham, has written about over the last several years, and something that he and I summarized in a commentary for the Journal of Clinical Investigation earlier this year, which basically says that for science, one might research a known prototype virus within a given family and generate enough generalizable knowledge to that family based on that virus so that it might be applied shall a pandemic occur. Ideally, we would want to develop countermeasures for that prototype pathogen through phase 1 clinical trial. While we didn't necessarily achieve that with our work around MERS coronavirus, we did have enough knowledge generated so that we were able to utilize that knowledge and move very rapidly towards vaccine development for SARS-CoV-2. So this is a timeline that we published in one of our manuscripts this year, which outlays how we were able to move from the onset of the virus, which started in the latter parts of December last year with an outbreak in Wuhan, China, to the point of which the sequences for what was then called 2019 N coronavirus was published in January 10th of 2020. And 66 days later, where in collaboration with NIAID, DMID, and Kaiser in Seattle, we were able to go into phase 1 clinical trial, which is one of the most rapid vaccine responses to date. And how do we get there? Only by way of studying MERS coronavirus and other coronaviruses extensively from years 2013 to 2019. And actually by studying SARS coronavirus 1 before I even got to the Vaccine Research Center with a team led by Dr. Mascola and Dr. Graham. So the main vaccine target for coronaviruses are the spike protein. This protein sits on the surface of the virus and is the virus's way of attaching to the human cell receptor. In the case of SARS-CoV-2, this receptor is the ACE2 receptor. And the spike protein attaches via the receptor binding domain, or the RBD. This RBD is housed on what we call the S1 region of the spike protein, which is the cap, so to speak, of this mushroom-shaped trimer structure. And then below that, where the fusion machinery is housed, is the S2 region of the spike protein. And so by purposes of studying vaccine design, we wanted to really understand the immunologic architecture of the spike protein. So prior to me coming to the Vaccine Research Center, there was various candidate vaccine approaches that were published for MERS coronavirus coming from the VRC, largely led by Lin-Chun Wei. And although I won't go into the data from these, what I do want to point out is that in the attempt to utilize the full-length MERS spike trimer as a vaccine antigen, largely those attempts were hindered because of low-level expression and also instability of the coronavirus full-length spike trimer. And so following these data and those understanding, we sought to stabilize the coronavirus spike in its pre-fusion conformation. And the proof of concept around stabilizing fusion proteins in their pre-fusion conformation is one that comes from Dr. Graham and Dr. Wang, and also Jason McClellan, who is a previous Salzman winner to my understanding. But around the vaccine, what is namely called DSCAV1, which is a pre-fusion stabilized respiratory syncytial virus F vaccine. This vaccine immunogen, as you can see here, is the F protein stabilized in its pre-fusion conformation, which is a shorter squatted conformation as opposed to the post-fusion conformation, which happens to spontaneously flip upon itself. What I want to draw your attention to are these red surfaces on these two proteins. Notice that the pre-fusion conformation, the red surfaces are sitting atop of the pre-fusion conformation, whereas they are occluded in the post-fusion conformation. And because of this, the pre-fusion conformation has the ability to elicit more robustly neutralizing antibodies than the post-fusion conformation, as seen in this graph from non-human primates. And so we wanted to do the same kind of thing with coronaviruses. And so when I got to the lab in 2014, we started to study human coronavirus HKU1. And luckily enough for us, the HKU1 spike had the ability to be expressed very well in cell culture and led us to a collaboration with Jason McClellan's laboratory and Andrew Ward's laboratory, where we were able to solve the pre-fusion structure of the first human coronavirus spike protein. Around the same time, David Beasler's laboratory published the spike protein from MHV coronavirus. And from then, there was an onslaught of literature describing the coronavirus spike proteins in atomic level detail, whether it be the glycan shields and the way that epitopes are masked to prevent high level vaccine responses, or the way that the proteins flip into their post-fusion conformation. And then here comes the data from George Gow's laboratory, which I like to say is probably was at the time one of our leading competitors in the field, where they solved the structures for the MERS coronavirus and SARS coronavirus spike proteins, revealing dynamic receptor binding domains. Interestingly enough, we had similar data where we described the spike protein structure for MERS coronavirus, but notably, we described it by way of understanding that it might serve the purpose as a rationally designed pre-fusion spike antigen. And so how do we design this rationally designed pre-fusion MERS coronavirus spike antigen? Taking what we knew from HKU1, which is largely similar to MERS and SARS, particularly in that S2 region where the fusion peptide is housed, we were able to scan in collaboration with Jason McClellan's group, namely Ming-Chun Wang, where we were able to see that there was a region in this S2 region that served as the place in the spring where the central helix and heptad repeat apex met. By inserting 2 proline mutations in that spot, calling that the MERS S2P, we were able to show that MERS S2P expressed up to 50-fold greater than MERS S wild type in cell culture. And as you can see here with the negative stain EMs, the MERS S2P has this beautiful homogeneous trimeric mushroom-shaped structure, whereas expression of MERS S in its wild type conformation spontaneously flips into its post-fusion conformation. So we now stabilize MERS S2P in its pre-fusion conformation, but putting those 2 proline mutations into the backbone of HKU1 and SARS S2P also effectively stabilize them in their pre-fusion conformation as well. It was also noted that we were able to insert those 2 proline mutations into the backbone of several other coronavirus spike proteins, namely some of the endemic coronavirus spike proteins, such as OC43, a porcine coronavirus spike protein, and most importantly, WIV1, which was considered at the time to be a potentially emerging coronavirus and sat in the same family as SARS-CoV-1, and now what we know to be SARS-CoV-2. So as a proof of principle around how these spike proteins in their pre-fusion conformation elicit immune responses in small animal models, we tested the MERS S2P spike protein in comparison to MERS S wild type and MERS S1 in its monomeric form in mice, giving 2 doses 3 weeks apart, and then simply taking a bleed 2 weeks following that boost and showing that with delivery of the MERS S2P spike protein elicits more robust neutralizing antibodies compared to MERS S1 and MERS S wild type. Now, what was interesting from this is it wasn't just that those antibody responses were more robust against the autologous strain of MERS coronavirus pseudovirus, but also other homologous strains, which includes the Korean strain that was then circulating in 2017, around the time that we published the paper. And so when you think about pandemic preparedness, there's the obvious precision or rational design of your vaccine antigen, but then there are other things that you need to come into play as well, in order to deliver a vaccine in a very fast, reliable, and in the italicized, if there's any universal flu vaccine people here, universal fashion. And that the vaccine must be able to be produced quickly in vast quantities, that the technology upholds some level of manufacturability standard, and that it's universal enough that the idea is preemptive and you can essentially plug and play your vaccine antigen into the platform and move very rapidly into human trial. So with that, in a collaboration with Moderna, who has a mRNA lipid nanoparticle platform, we were able to insert our new therapeutic protein. In this case, it was the MERS coronavirus S2P protein, use their mRNA lipid nanoparticle to formulate the vaccine, and then show that mRNA containing the MERS S2P by proof of concept has the ability to protect mice against the MERS challenge. So the mouse model that we use is the 288-330 mouse. So this is a human DPP-4 transgenic mouse model coming from Ralph Barrick's laboratory at the University of North Carolina. These mice unfortunately succumb to a lethal dose of MERS coronavirus following challenge, as you can see by the mice that are dosed with just PBS. However, when you give 2 injections of MERS S2P in Moderna's mRNA LNP platform, even at modest doses of 1 microgram and 0.1 microgram, all of the mice survive following the challenge. Even at what is considered to be a subprotective dose of 0.01 micrograms per mouth, we do not see any breakthrough infections, and the mice actually do recover following that challenge. So it wasn't just notable from the weight loss perspective, but also from the lung disease perspective. Taking lungs at day 3 following the challenge in those animals, we saw a complete protection of viral replication against viral replication in the 1 and 0.1 microgram dose of MERS S2P mRNA. And also those mice were protected against lung hemorrhage as well. And so MERS S2P mRNA elicited a dose-dependent protection against a lethal MERS challenge in this particular mouse model. And so what that meant for us is that when 2019 and coronavirus sequences were published on January 10th, we were able to very quickly in 3 days, in collaboration with Moderna, decide on what is called the mRNA 1273 sequence, which really meant putting the 2 proline mutations into the backbone of the SARS-CoV-2 spike protein. And that led us to having immunogenicity of the mRNA 1273 just 37 days later. So I usually don't show this particular slide because this data often hides behind some of the non-human primate data. But I wanted to show this slide because I think it really does tell a story of how all of the things kind of came together for the good. We started by immunizing 3 different strains of mice, largely because in collaboration with Ralph Barrick's laboratory, we didn't know which of those strains of mice would take, so to speak, to their mouse adapted strain. And so we immunized BALB/c, C57s, and hybrid mice with 2 doses of mRNA 1273 and bled them pre-boost and also post-boost to show that we were eliciting not only robust binding antibody responses in all 3 strains of those mice, but also reputable neutralizing antibody responses as well, particularly in the BALB/c mice and the B6C3 mice. So that's after 2 doses, but also given 10 micrograms and 1 micrograms of mRNA 1273 in 1 dose, we were able to show after 4 weeks following that dose that we are eliciting 10 to the 3 IC50 titers as well. By way of protection, around the same time as we come up across that data and about 5 weeks following the boost, the Barrick lab had a mouse adapted strain of SARS-CoV-2 that, while it was not lethal, did have the ability to replicate locally in the lungs and noses of these animals. So we were able to really test our vaccine's efficacy against viral replication, both in the lower airway and the upper airway of these mice. And so you can see here in those 1 microgram immunized animals that they were completely protected from lung viral replication. And in a dose-dependent way, the vaccine response showed to be efficacious. And also, following 2 doses of mRNA 1273, 3 out of 4 of the mice that were dosed with 1 microgram were protected against viral replication in their nasal turbinates. What does that mean for the 1 dose that, again, on the previous slide I showed had 10 to the 3 neutralizing antibody responses? It means that following 1 dose as well of mRNA 1273, 7 weeks, we were able to show that 10 and 1 micrograms of this vaccine was protective in these animals, which was very, very interesting to see, particularly as you think about the utility of a vaccine in an outbreak response. We next, in collaboration with what I call the VRC's now non-human primate working group, Nancy Sullivan, Bob Cedar, Mario Roeder, and their laboratories had lots of influence on these early non-human primate studies where we immunized monkeys with mRNA 1273 at 100 micrograms or 10 micrograms, comparing that to PBS at week 0 and week 4, and then challenged with close to 10 to the 6 PFU of SARS-CoV-2, following these animals of their immunization for 4 weeks to assess immunogenicity by way of antibody and T cell responses, and also protection in the upper and lower airway. The manuscript also has pathology that was done by Ian Moore's lab, but for brevity of this presentation, I won't show that today. Looking at the antibody responses just at 4 weeks following the 2nd vaccination, so this is immediately before the animals were challenged with 10 to the 6 of SARS-CoV-2, you can see firstly with the binding IgG responses to the full-length spike protein that there's this dose-bearing response on IgG titers that is on par with the upper quartile of binding antibody responses that we see from human convalescent plasma that is collected from people who had recovered from COVID-19 disease and various severities of their diseases as well. So eliciting about 10 to the 4, 10 to the 5 with the 100 microgram dose of mRNA 1273 in the NHP model, what does that look like for the functional relevance of these antibody responses? And so we took advantage of 3 different assays, the first of which is our standard pseudovirus neutralization assay that we do at BSL-2 level here at the VRC. We also took advantage of an ACE2 binding inhibition assay that is done by Adrian McDermott's laboratory, and a live virus nanoluke assay that's done by Ralph Barrick's laboratory. And the study across all of the functional antibody assays remains the same, that mRNA 1273 at the 100 microgram dose elicits robust neutralizing antibody responses and ACE2 binding inhibition responses that is above or at the upper quartile of what you see in a large panel of 41 human convalescent sera. And it's even interesting to see, particularly with the live virus neutralizing antibody responses, that even the 10 microgram dose of mRNA 1273 fared fairly well. So we showed that we can elicit rapid clearance of viral replication in both the lower airway and the upper airway by using subgenomic RNA samples from the bowel and also from the nasal swabs. So first, looking at the bowel fluid, which is the lower airway viral replication, you can see that the PBS group on day 2 has high levels of viral replication up to 10 to the 5. But for 10 micrograms and 100 micrograms of mRNA 1273 dose NHPs, that the virus has essentially cleared from the lungs by day 2, especially by day 4 with the 100 microgram dose of mRNA 1273. So for the nasal swabs at the time, it had not been shown for any of the vaccine candidates that are in the later stage phase 3s that there was the ability to be protection in the upper airway. And so we were very excited to see that for the 100 microgram dose group of mRNA 1273, that by day 2, all of the animals had essentially cleared viral replication from their upper airway. And so the ability to limit viral replication by both the lower and upper airways has important implications for vaccine-induced prevention for both not disease, but also hypothetically for transmission as well. So as we were doing all of those animal experiments, on the other side of this rapid vaccine development timeline, obviously as things are happening simultaneously as we are moving quickly, we were also, in collaboration with Moderna, had initiated cGMP production of the phase 1 clinical drug product. So Moderna initiated that production 4 days after the sequence of the virus came out. The product was shipped 41 days later, which I think really is a testament to the ability of these gene-based delivery systems and manufacturing to be very quick. And then obviously the phase 1 clinical trial started shortly after that. I am going to just show quickly a snippet of data from the phase 1 clinical trials where mRNA 1273 was given at 25, 100, or 250 micrograms in young adults, and then in aged adults or older adults, 25 or 100 micrograms at week 0 and week 4. There's a series of blood draws that will be collected over the course of 1 year to follow these participants. But I'll just show a snippet of some of the early preliminary data. The data that you're looking at are neutralizing antibody responses from a live virus newt assay. It's a focus reduction neutralization assay done out of Emory University. In this gray section here on the far right of the graph are the responses that were published from the 100 microgram dose young adult group, where you can see that there is high levels of neutralizing antibody activity. And while I'm not showing it here on this particular graph, it is again shown to be in the higher upper quartile of what you see from in comparison to human convalescent sera. Now, when you look at people that might be the grandparents of those in the younger cohort, aged from over 56 years old, the interesting takeaway from this is that you obviously see that there are equal amounts of neutralizing antibody responses in the aged people as there are in the younger adults. And so in conclusion, we were able to stabilize the prefusion conformation of various spike proteins by including 2 P mutations in the S2 regions, vaccination with those prefusion stabilized MERS coronavirus spike trimers elicited robust neutralizing antibody responses and really laid out the framework for how we might move forward with a pandemic response, particularly as we looked at MERS S2P being delivered by mRNA, which was highly immunogenic and protective in animal models. And the same was shown in the early days of our pandemic response with SARS-CoV-2 S2P delivered by mRNA as well. And lastly, SARS-CoV-2 S2P mRNA, or mRNA 1273, elicits robust immune responses in humans and is currently in a phase 3 efficacy trial that has completed enrollment of over 30,000 participants as of, I think, now about 2.5 weeks ago. And so we look forward to those efficacy results, particularly as Pfizer showed in their interim analysis with a similar vaccine construct that they have about 90% interim efficacy rates. And lastly, I want to just thank everyone at the Vaccine Research Center, particularly all of the people that are named on this slide, which essentially started at the bottom for me, with me when I came here in 2014. Since mRNA 1273 became a thing and COVID-19 response and started at the VRC, this has been a VRC-wide effort with all hands on deck. I'm sure that this slide likely leaves off people, so I apologize if you are on this call and it does leave you off, but none of this work would be possible without everyone at the VRC's expertise and hands and prayers. And I do name a few collaborators and contributors on this particular side, mostly because I wanted to make sure that I called out the people who have been collaborating with us from essentially the beginning: Moderna, Jason McClellan's laboratory, Andrew Ward's laboratory, a lot of EM coming out of Frederick National Laboratory, and mouse studies since even all of the MERS work with Ralph Baer's laboratory at UNC. And a particular special thank you to all of the clinical trial volunteers, some of my family and friends included. And, oh, I might cry. You can just look at the picture and I know part of Dr. Salzman's legacy is around his mentorship, and part of the reason why I was given essentially the wings to fly with this project is because I had a mentor who allowed me to use his resources and sit in the back of his laboratory and ask scientific questions that a lot of other people didn't care about. And so I am completely indebted to Dr. Graham for this. And yeah, thanks.
Thank you, Kizzmekia. It's just so inspiring, your work. And it must have been an incredibly intense 9 months since this started, and not to mention before. You have quite a few questions. I'm going to take the privilege of being the chair and ask you a very basic question first, and then we'll get to the 3 or 4 questions from the panel. So my basic question is, have you looked at the expression pattern of the mRNA? I mean, what kind of tissues are they expressed in? How long are they expressed for, the mRNA vaccines that you're putting? Do you know anything about it yet?
Yeah, we don't really know. There's work on similar candidates like out of Duke. And so there's preliminary data, and only work that we've done with mRNA 1273 is in a cell culture dish, only to show that it does express and the antigenicity is proper. So there's more work that needs to be done around that.
Okay, so I'll get to the questions from our attendees. From Nazarena, it says, excellent overview of the challenge studies. First question, going back to the previous topic of VDE, have you considered immunizing humanized mice pre-challenge with a variety of other coronaviruses?
No, we have not. But the beauty of starting a phase, a phase 1 clinical trial very early is that we now have data from day 0 of the phase 1, people who we have a fairly good understanding of how they respond to the vaccine. And so we're going to try to attempt to answer some questions around pre-existing responses and how those might affect vaccine responses. But I guess what is important to know from the elderly cohort is that it looks like, if anything, preclinical responses are good. Pre-existing responses are good.
And related to that, is the prior exposure panorama of the older adults been investigated?
Not yet.
Not yet. And from Stuart Isaacs, is the Moderna RNA modified RNA like it is being developed by Pfizer BioNTech?
Yes. The vaccines from Pfizer and Moderna are essentially very much alike, inclusive of the S2P mutation, the same type of mRNA modifications, transmembrane anchored spike proteins, et cetera. So they are fairly close to each other. So you can imagine that part of my emotions today actually probably have to deal with just the excitement around the phase 3 trials that Pfizer has put out today.
And lastly, have you looked at the T-cell responses at all?
Yeah, and I left those out for brevity, but we looked at T-cell responses in mice, monkeys, and also in humans. And the short story is, for what we were interested in really mostly understanding was the type of Th1 versus Th2 responses that we were eliciting. So we were eliciting a Th1 biased response, CD4 response. We are eliciting TFH cells in the monkeys. Unfortunately, in the humans, as opposed to the mice, where we did see some spike-specific CD8 cells, we do not see that in the humans, at least by our assays. But Pfizer does report a little bit, so we are very confident in the antibody responses and the T-cell responses, T-cell phenotypes that we're seeing.
Thank you, Kizzmekia, and congratulations again on your award, and best of luck in your future studies. Thank you. All right, and our next speaker will be introduced by Alan. Alan, take it away.
Okay, so it is my great pleasure to introduce a guest, Rebecca Dutch, who, as you see on the slide, is professor and chair of her department at the University of Kentucky. She has done a lot of work over the years on a variety of RNA viruses. She trained with Robert Lehman and Bob Lamb, and has worked on pneumoviruses, paramyxoviruses, and also SARS-CoV-2. She's a former president of ASV, and I think I'll get out of the way and let her give her talk. So, Rebecca, we're glad you could make it.
Well, thank you very much. It's really a privilege to be here. Let me see if I can share. I think we need to turn off the other screen share maybe. Okay. Hopefully this will work.
Yes, we can see your screen.
You can see it. Let me hit slideshow and hopefully, all right. Are you seeing just my slide?
We're seeing just your slide.
Perfect. All right. So it's really a privilege and an honor to be here. I did not have the privilege of knowing Dr. Salzman, but I know the importance of honoring young, talented people and the importance of mentoring and the importance of using that as a way to push forward science. Actually, one of my former PhD students, Rachel Showalter, won the Norman Salzman Award a number of years ago when she was in Chris Buck's lab. And I know just how meaningful that award was to her. So I want to thank everyone who works on this program. It's so important. And then I also want to say what a privilege it is to talk with the group of amazing speakers we've heard so far. So I'm going to change gears a little bit, and I know it seems very strange in this current time to talk about anything other than SARS-CoV-2, but I'm going to shift and we're going to talk about a different respiratory virus right now, and that is the human metapneumovirus. Give you a little bit of background on it. So in some ways, this is an emerging virus in the sense that human metapneumovirus, or I'll now refer to it as HMPV, was actually first identified in 2001, so less than 20 years ago. Now, this does not mean that that is the first time it emerged into the human population. In this case, the fascinating story is that it had been there for a while and we just didn't know it was there. For many years, there had been patients who had all the symptoms and signs of what you'd expect from something called respiratory syncytial virus. But when you actually went to do the examination of the samples, you did not find evidence of an RSV infection. And finally, in 2001, a group from the Netherlands published the identification of a different respiratory virus, human metapneumovirus, that was actually the causative agent of those infections. Turns out this is a worldwide respiratory pathogen, causes wheezing, bronchiolitis, pneumonia, and is actually quite a significant cause of morbidity and mortality, particularly in children, immunocompromised patients, and the elderly. And prior to the emergence of SARS-CoV-2, this would have been the second or third leading cause of viral pneumonia in the elderly populations and a second or third in children. So this is a very important virus clinically, but it's a virus we know very little about because it's so recently come out. So what you see also here on the slide, it's a very simple virus. This is a member of the pneumovirus family. So it only has 8 genes. This is a negative strand RNA virus. So unlike SARS-CoV-2, this is not, the genome does not work as a messenger RNA. And this just gives you a general idea of the schematic of the virus. There's an envelope, just like we heard this morning about SARS-CoV-2, in this case with 3 different proteins sticking out of it, the fusion protein, the attachment protein, the small hydrophobic protein. There's a matrix protein, which seems to help with some version of assembly. And then inside is this important ribonucleotide capsid. It's the RNA coated in what's called the nucleocapsid protein. And then bound to it, we have something called the phosphoprotein, which until recently we thought of only as a cofactor for the polymerase. And then this, what we call the large protein, because RNA virologists generally don't have exciting names. That's the largest protein in the genome. What it is the RNA-dependent RNA polymerase. And together that makes up human metapneumovirus. So when my lab started to work on this, and actually that work was started by Rachel Showalter when she was a graduate student in my lab, so the person who went on to get a Salzman Award, there's very little known about HMPV, so instead we looked at what's known about related viruses, pneumoviruses, others like that, and this just shows you a general schematic of what we would believe the life cycle would be. You'd attach, and my lab has spent a lot of time working on attachment and entry, but today we're not going to talk about that. Once you get into the cell, you're going to release that genome, and you're going to have to carry out the processes of making messenger RNA, which we refer to as transcription, the process of making what we call anti-genome, that's a complementary RNA. So this is the first step of replication that goes on to make more viral RNA. These messages will make lots of little proteins. You'll then assemble this into a particle, which might happen at the plasma membrane, which is where we think most pneumoviruses would go ahead and assemble. And then the idea would be you'd bud out a single particle and it would go on to a new target cell. But honestly, there had been very, very little work done on this idea of how do these really replicate. And so a former postdoc in my lab, Nicholas Cifuentes, who now runs his own lab in Santiago, Chile, wanted to ask questions about what are the factors that are important for replication of this virus and when does it happen? So one of the things he set up were probes, in this case, FISH or fluorescence in situ hybridization probes that would allow us to just tell the difference between a positive strand RNA, so that would be either messenger RNA or what we call the anti-genome, and a negative RNA. And these FISH probes, I want to again get a shout out to NIH. When Kanta Sabaro was there, she was actually the person who helped us get this up and going and gave us some really nice advice. So in this case, what you can see is if we just put in RNA that would make messenger RNA for the nucleocapsid and phosphoprotein. So those two proteins we made and their messenger RNAs around. We'll see the formation of these little structures, which we think are the replication centers. And you'll be able to pick up the positive RNA. In this case, it's messenger RNA that's present, but there's no signal for viral RNA because it's not being made. Contrast, we have a plasmid that'll just express the full length viral RNA. And if you have that, we'll be able to pick that up, but we don't pick up the positive. So we know that we can tell the difference. So once we had these constructs set up, Nicholas went ahead and looked to ask several questions. Do we see replication centers like this form with a number of different strains of HMPV? And are they indeed the sites of replication, meaning we'd see positive and negative RNA there? And the answer is yes. So we see, and this is 3 different strains of HMPV. What we'll see is this is viral RNA present in what we think is a replication center. In this case, with the nucleocapsid protein, you can see it merges in. These are 2 different strains, and you see the same similar idea. We're forming similar structures. And these structures are generally referred to as what are called inclusion bodies. Now, this part of the project was not really a surprise to us at all because inclusion bodies are a common replicative strategy for these negative strand RNA viruses. These viruses do their replication in the cytosol, but they appear a wide group of them. This is rabies, respiratory syncytial virus. Here's our HMPV pictures, PIV3, Ebola virus, and Marburg virus. They seem to form membraneless regions of the cytosol where they coalesce to put together the critical factors that they need for replication and transcription. So what more would we find out about these replication centers for human metapneumovirus? Well, one of the things Nicholas noticed by doing a careful time course is from the beginning of the infection through to the 6 to 12 hour period, what you found were the coalescence of these. Early on, you'd see a large number of smaller inclusions, and then over time, we would coalesce so that by 24 hours, we had less than 2 per cell on average. We also found out that if you add cytoclase D, which is going to block actin polymerization, that this coalescence is significantly inhibited, suggesting that there's a dependence on actin polymerization for this process. And because sometimes visuals are even easier than graphs, this just shows you slides of a cell, an HMPV infected cell, when you've added cytoclase D and you see these large number of small inclusions, and 24 hours later, when you didn't have it, one nice large inclusion where we think the majority of the replication is going on. So is this important? Do you need this coalescence in some manner for the virus to efficiently work? And so to test this, what Nicholas did is to go in and do the same treatment, cytoclase D, but now instead he did assays to look at both the transcription and replication of this virus and to see if they were impacted. And what we find is that actually both are. This is just the controls, DMSO controls. Nicotazole, which is going to hit your microtubule polymerization, does not affect the transcription or replication really at all. But what we find is cytoclase D has a significant impact in the 6 to 12 hour period when you should be ramping up transcription and then ramping up replication. We see a significant block in both processes, suggesting that there's a correspondence between what is happening with actin polymerization, which leads where we see these nice coalescence of inclusions, and the efficiency at which this virus can carry out its transcription and replication. So if you're coalescing, how is that happening? So what we found is that these inclusion bodies are very dynamic structures. So this shows you one example. This is, in this case, the phosphoprotein, which is a key protein in those inclusions, has been tagged with mCherry. And what you see is that you'll have both fusion events. So you'll see a smaller inclusion coming in and fusing with the larger one. You'll also see sometimes events where you see one leaving. You see small bits leaving from that inclusion, suggesting they're dynamic structures. And you can see that dynamic structure potentially even better in this slide. What you'll see is, in this case, you see one pulling off. This case, you see another small, what looks like a very small inclusion. It has mCherry P present in it. And what you see is it's being essentially pulled towards this larger inclusion. And then you see this one, you saw what looks like a fusion event. So if these are really dynamic structures, and they almost seem to do fusion and fission, but I've already told you they don't have membranes around them. Why? What does that mean? So now I want to switch to what I think of as a very fascinating topic, but one that probably a lot of people haven't really run into yet, but if you haven't, you will. And that's the concept of what's called liquid-liquid phase separation, or the formation of what can also be referred to as biomolecular condensates. The idea is the following, that if you look at our cytosol that is full of all sorts of proteins, nucleic acids, metabolites, we tend to think of this as all being equally spaced out, except it really isn't. There's areas that have a much larger concentration of key things. And principles that came out of biophysics have really now been applied to the cell in about the last decade. To start understanding that in many times these proteins actually, a protein or RNA can form its own separate region regardless of whether a membrane is present. And you see these membranous organelles and things like nucleoli and stress granules. They're likely involved in lots of key processes. In fact, if you actually go into PubMed and put in liquid-liquid phase separation, you'll be amazed at what you find just in the last few years. The idea is that at certain concentrations, specialized proteins or RNA actually demix from solution, so that instead of being evenly distributed, they form their own area and they help pull in things into that area and exclude other things, even though a membrane is not there. And why is this important? Well, because if you're in a region that's liquid-liquid phase separated, it's going to be concentrating key things present, but you're going to have rapid exchange with the environment, much more rapid actually than a membrane compartment. And that's very important. So are these inclusions liquid-liquid phase separated regions? So one way to look at this is to do something called fluorescence recovery after photobleaching. So in this case, what you do is to take a cell and you go ahead and bleach the area so that now, so the cell is expressing a fluorescent protein. In our case, it's a fluorescent version of the human metapneumovirus phosphoprotein. Then you look to see if you can rapidly get back into that region your fluorescent molecules, suggesting that things can move in. You can also measure rates at which this happens. So Freire El-Najjar, who first was a very talented grad student in my lab, then went away for a postdoc and now is back, luckily for me, for a year as a visiting scholar, did this, and she looked at two things. She looked at the rate of recovery for just the P protein by itself, and this will be outside an inclusion because the P protein needs its friend, the N protein, it's another viral protein, to form an inclusion. If you put the two together in a cell, you'll see inclusions. Without them, you won't. And what you'll see is P is rapidly diffusing around cells when it's not in the inclusion. If you look at inclusion regions that have both the phospho and nucleocapsid protein, you see first two important things. You do see recovery. So that says more P can diffuse into that region. It's not membrane bound, but the rate of that is quite a bit slower than out in the cytosol, suggesting that the properties in that region are quite different than the properties of the rest of the cell, which is consistent with liquid-liquid phase separation. So liquid-liquid phase separation of viral infection. If I had put this slide up 3 years ago, there would have been very little to tell you. But it's a really exciting area where a lot of change is now coming. So inclusion bodies have been, not just our work, but inclusion bodies are proposed to be formed by phase separation for several negative strain viruses. That includes rabies, the first report, vesicular stomatitis virus, and now measles virus, which is a paramyxovirus, and very recently respiratory syncytial virus. So our work fits into that idea that somehow phase separation is critical for forming replication centers. interestingly, in the last few months, and I believe none of these papers are yet published, I think they're all up on the preprint server. There have been 8 different papers that have shown that the SARS-CoV-2 nucleocapsid protein can actually drive phase separation. So it may be, phase separation may also play a role there. There's been work to suggest that influenza genome assembly may be, that phase separation may help regulate that. And a new paper this year also suggested that the HIV-1 gag protein may promote phase separation to modulate RNA positioning. So this is a new and really important oncoming phase. But so before I go on and show you a little bit of biochemistry type experiments on phase separation, I want to tell you a couple critical concepts. When you think about what's happening in phase separation, one of the terms you can think about is something called a scaffold protein. Essentially, you're forming a region that's separate from what's around you. And a scaffold is thought to be the protein that drives this process. Think of it as the foundation or the scaffold on which this phase separation separated region rests. And then that scaffold draws into it other client proteins or other client nucleic acids, client factors, which will preferentially come into those regions and are drawn in by scaffolds. So in our work, we asked the question, the phosphoprotein, it seemed to have all the characteristics you'd expect from a scaffold protein. And that is, we know so far what we knew is critical for as a polymerase cofactor. We knew it was critical for inclusion body formation, but it had several other properties. This shows a structure from about 2013 of at least the center part of HMPVP protein. It's a tetrameric protein, and multivalency, or the ability to have multiple binding sites, is very important in the process of phase separation. It also has on both its N and C terminus extended regions of intrinsic disorder, and these are often thought to be regions that are involved in binding to other critical partners. So does this act as a scaffold protein? So one way to look at this is to purify the protein. So in our case, we purified the protein from E. coli, ran it over several different columns to get it pure. And this started as a collaboration with Trevor Creamer in our department, who's a wonderful biophysicist and continues to work with us on this. And then this is Carrie Slaughter, who's a graduate student in my lab, who's really pushed this project forward. And what you'll see is this is the phosphoprotein by itself. And what we see we see the presence of these, remember, this is purified protein put on a slide. And you'll see the presence of these nice round regions. The round shape is what you expect to see from these liquid phase separated regions. And to look more closely, there's several other things that suggest that the P protein by itself is phase separating. This just shows you a different type of assay where we see this nice formation of separate structures. But most importantly, if we look under slides, we can actually see that different droplets of P will fuse together, like you would expect to see in liquids. And that's a strong characteristic. We also see that if we put higher salt in, these droplets dissolve. So our work to date would suggest that this P protein by itself can do liquid-liquid phase separation. I will say this is different than what's been suggested for measles and respiratory syncytial virus, where they've looked only at a combination. They've seen it with the phosphoprotein and the nucleocapsid. Our work would suggest phosphoprotein alone, but what about its partner? So I told you in cells that you need the phosphoprotein and the nucleocapsid protein to form these nice inclusions. So what about the nucleocapsid protein? What role might it play in the actual process of phase separation? So there are 2 forms of the nucleocapsid protein that are important. The first is what I consider just I'm going to call it wild type because it's a wild type form, but this is a protein that binds RNA. And this is a lovely structure from Max Renner and his colleagues back from 2016, showing the nucleocapsid protein, which oligomerizes as it binds to RNA. And then the red in here is the RNA binding. And what you'll see is that nucleocapsid protein forms a tight complex with RNA and with itself, where the N and C termini of this interact. So what about this wild type protein, which would be binding RNA? Does it phase separate? Well, when you purify and you look under a scope, you see drops, but what you'll see is that they're not round in the same way. We don't see fusion with these structures, so they're not consistent with any kind of liquid state. Surprisingly, though, if we then took the nucleocapsid protein and put it together with the phosphoprotein, then we get this. We get nice round structures. And in fact, we also tested this is putting in a fluorescent RNA oligos. So not only does the phosphoprotein draw the nucleocapsid into these, but RNA is coming with it. We also looked at the other form of the nucleocapsid protein. So the nucleocapsid protein plays two roles. One is to coat that RNA, but actually prior to that step, it has to be kept monomeric and ready to coat RNA. And that's done by an interaction between the phosphoprotein and the nucleocapsid protein shown here. So we also asked the question, well, if we make a construct that has to stay monomeric and isn't going to bind to RNA, can it come in? And what we find is, again, it doesn't form round structures on its own. It seems to form these aggregated gel-like structures. They don't dissolve in high salt. So this doesn't look like classic liquid-liquid phase separation. But if we put it together with the phosphoprotein, this shows you, this is labeled, the labeled nucleocapsid protein, the labeled phosphoprotein. As you merge them together, you see they're forming really nice coincident droplets. And you, again, in this case, can nicely see fusion occurring. So our model is the following. We think that the phosphoprotein forms the scaffold for phase separation. So the first part of what's going on in replication, the scaffold is drawing things into it, and it's drawing in both the RNA-bound form of this nucleocapsid protein is going to be part of this, but also the monomeric form, and both actually would need to be present as replication were going on. We're continuing to now look at what factors might modulate the properties of these regions. We wonder about the ratio of these proteins, working on that, and also phosphorylation. So just in like one slide, I'm going to tell you a little bit about phosphorylation, and then we'll go on to how we now spread to cells. So the P protein is called the P protein, the phosphoprotein, because it's heavily phosphorylated. So we've started to examine phosphorylation and try to separate it. Can we separate its role in replication, because the phosphoprotein is a replication cofactor, from what's going on in phase separation? And so Rachel Thompson, who's another wonderful graduate student in my lab, has been doing some of these studies. This just shows you mutations of some of the key serine sites within the HMPV protein. And she's already identified several that are critical for the ability of replication, shown by what's called a mini genome assay, where we look at the ability of this RNA-dependent RNA polymerase to replicate. And so we have already identified phosphorylation sites, or putative phosphorylation sites, that knock out the replication if we take them out. We are now identifying more. I will tell you though, when we looked, we wondered, okay, as soon as we knock these out, will we get rid of the ability to form an inclusion, to phase separate? And the answer for those mutants is no. We'll still form these regions, but they can't replicate. So clearly the phosphoprotein is very complicated. It has roles in phase separation, has in forming these IVs, has roles in replication, and some of those may be tied together. And that makes it very exciting to keep examining what this is doing. So I've taken you through this first part that we're going to talk about, and that is what happens once we come into a cell. We form these structures that are involved in replication. But now in the last 10 minutes or so, what I want to talk to you about is now, how does this virus spread to new cells? The model we all think of is that you would take a single genome and you would take it to where it was assembled on the plasma membrane, you'd bud out a nice structure, and that would then spread to new cells. So a number of years ago, when Freire El-Najjar was a graduate student in my lab, she started just asking the question, if I infect BAS2B cells, which are a human respiratory line, with human metapneumovirus, what do I see? And you kind of expect nice single particles, maybe filaments, maybe spherical particles. But what she found surprised her. In much of the case, what we found is that if you infect these cells with HMPV, you start to see the formation of two things, these lots and lots of these big branched filamentous structures coming off, which we think are a form of virus, because if you look at these filaments coming off, they form branches. Over time, the branches get more, and they have genome in them, along with a lot of other things, and all the viral proteins. But you also see the formation of these large intracellular filament or extensions, two things that connect two cells. And in fact, if you look at general cells as they grow, you'll see something like this occasionally, about 10% of your cells. But the HMPV infected cells, about more than 80% of your cells have these large extensions. And when Santiago Restrepo was an undergraduate in my lab, he actually then used electron microscopy to look more carefully at these. And what he sees in mock-infected cells, these extensions are relatively clean, small, little bits of wispy filament occasionally coming off. But in HMPV infected cells, you see large numbers of these filamentous structures coming off and large numbers of vesicles on these extensions, suggesting the extension itself is very different. So what do you need to form these extensions? Just briefly, we know you need actin polymerization. So in comes actin again as a player in HMPV infection. If you use either cytoclasm D or JAS-PLAC analyte, which hit actin polymerization, we will just knock out the number of cells that have these extensions. In contrast, inhibitors of microtubule polymerization have only a modest effect. If you look with and ask what cellular factors, in addition to actin, we'd use targets of the major Rho-GTPases. And what we find is that hitting CDC42 or RAC1, both of these has a significant impact on this ability to form extensions. So what viral factors are involved? Well, one of the subtitles I probably could have given this talk is all the surprising things that HMPVP does that we didn't know about. So as I said, when we started this work, we thought P was only a polymerase cofactor. But what we find is even just expressing P in cells leads to a lot of membrane deformation and things that look a little like these extensions. And so P seems to have a membrane interaction component too. But it doesn't just do it on its own. We wondered if we knew actin was required, so maybe P needs actin. So to look at that, what Freire did was what's called a proximity ligation assay, which is an assay to allow you to look at what's within 40 nanometers of each other. What she found is that in mock-infected cells, the P plus actin, we see no interaction. In an infected cell, the P plus actin, you see a nice interaction. You don't, however, see an interaction with tubulin, so it is specific. And this would suggest the phosphoprotein has another partner, and that's actin. You can even see this is storm high-res imaging looking at an HMPV infected cell. And what you'll see in green is the phosphoprotein, and it actually lines up in what looks somewhat like striated branched actin. So you've got the phosphoprotein and actin involved in forming these structures. So why are they there? So we wondered if the purpose of these structures is to promote formation of these extensions so you could go directly from cell to cell. Maybe you don't need to always go out and in order to infect the next target. So one way you could look at this that Freire did was to actually set up what's called a co-culture experiment. In this case, we want to be thankful to particularly the herpes virologists who first pioneered a lot of this kind of work. But what you do is to take one cell that's expressing the HMPV or is infected with HMPV, and you give it a target cell, and you're looking to see if you can move the infection from one cell to another, so where the cells are co-cultured to each other without moving a dye that's also staining the initial target cell or the initial infected cell. Now, of course, what you also have to do is block the ability of released virus to be released and then infect that cell. And we did that using neutralizing antibodies that we'd gotten from John Williams at Pitt. But what you find is the following. Let's look first at this side. Neutralizing antibodies block cell-free infection right now. By cell-free, I mean if I take virus, purified virus, I add these antibodies, we block more than 95% of your infection. In contrast, if you take infected cells and put them on other target cells, even in the presence of antibody, we only block about 50% of our infection. What this suggests is that cell-cell movement is actually very important for how human metapneumovirus might move from one cell to another. So if and I also should point out, any treatment that we do, like a CDC42 inhibitor that reduces extensions, reduces the spread. So what is actually moving? If you're moving from one cell to another, what has to move? And you don't need to move an assembled virus. In fact, given this is a membrane-bound virus, it would be surprising. And these pneumoviruses don't have nice capsid structures of any kind. They have just nucleocapsid binding to RNA in these inclusions. So I want to show you one movie that we found as we were trying to understand both inclusions and how movement. So this is you'll see this is one cell, and you'll see both the GFP, which also labels our inclusions, and this mCherry protein. And what you'll see is that you see the dynamic movement of the structure. And you'll see this is an extension that's now moving towards another cell. And the inclusion is very malleable. And you'll see at one point, it actually seems to go ahead and just move, squish itself through that extension and move over. So is that a possible mechanism? Is it possible we could move that way? And I think the answer is yes. I think that movement of inclusion bodies from one cell to another, or portions of inclusions, is possible. And because these are these phase separated, very dynamic, very malleable regions, that should be possible. So I want to briefly touch on one other topic, and that is what about in the real life situation? I've just shown you cells on dishes, but what about in an airway? So that's where all these respiratory viruses are infecting. So to look at this, Tyler Kinder, who was just graduated, a wonderful graduate student in my lab, used the Epi Airway system from Matech to do HMPV infection, and you'll see this is infection of your ciliated cells. HMPV does not infect particularly well. It does infect, and over time you'll get more, but by the time we get on infection, we start to lose our HMPV infected cells. You can in fact see this. We get spread initially over the first 48 hours. By 72, we start to come back down. We get very poor release from the apical surface. And we did this side by side with respiratory syncytial virus, which releases beautifully to the apical surface, but HMPV really does not. We went ahead and then did a lot of microscopy of these sections to ask if it's getting in, but it doesn't go that well and it's not releasing. What's going on? Well, we could show that inclusions form in this. And I think this is the first time that's been shown that you can actually form inclusions in these kinds of more relevant human tissues. So this is cross-sectioning these and then looking with nucleocapsid protein, phosphoprotein, and you can see in cells that are infected, you see these nice inclusions, and we've also shown the viral RNA is there. So you can infect them, you form these dynamic inclusions. What else do you form? Well, interestingly, about 35% of the infected cells form these strange structures that we don't see in RSV infected cells. They look similar to these extensions reaching out. Somehow they don't spread as effectively as we see in our 2D culture, but they are forming. And interestingly, when they form, if you just use those same neutralizing antibodies to prevent infection of HE cultures, they block really well. But if you try to block spread, there's only minimal block, suggesting that even in these HE cultures, cell spread may actually be a critical way that HMPV moves, and a way that a respiratory virus might have an advantage. So that just brings me kind of to our final models. We've talked about inclusions and the idea that we think these are dynamic phase separated regions, and that this is a really exciting area of ongoing research. This is what we think of as the common mechanism for going ahead and spreading to a cell. Go out and assemble a new particle, move that new particle, release it out, find a new target, and come in. And that clearly does happen with HMPV. But we also think this might be another mechanism, form an extension and form a connection between two cells. And one way, and we're still obviously looking at a lot of the details of this, but one thing you can then do is just simply move an inclusion, move the replication test. And we're not the only ones proposing this. There's been some lovely work now from Roberto Catania's lab on measles, which is suggesting something similar. And a number of work from a number of groups suggesting that this kind of process, moving multiple genomes over, would help. And if you think about this, if I move an inclusion that has multiple genomes in it and the replication status, and I move that from this infected cell to a new target, I'm not moving one genome, I'm moving hundreds. And you're essentially bypassing a lot of the early steps in the infection process and really giving yourself a head start on anything that the new target cell might mount against you. So we think this is an exciting area, this idea of moving multiple genomes. And I think it's an area that a number of virologists are now looking at from different systems. And it's something that we're going to continue to look at. And with that, I just want to thank people. I've tried within the slides to thank each person in my lab that has worked on this. I also want to thank Carol Monsman, who's a very talented microscopist. And then Rachel Ferns, who provided us with the mini genome system for HMPV and is a great collaborator. John Max Renner, who's a structural biologist at Oxford, who worked with us on the N0P construct. And then Hong and Nicole, who were at AstraZeneca, who we were working with the RSV project. And with that, I would be happy to take any questions.
Thank you, Becky. Wonderful talk, fascinating. We have a couple of questions coming in. One question is there a plan for more complex in vitro models of the lower respiratory?
Yes, sir. I mean, we're
organoid type things.
Yeah, we're looking at lung organoid or respiratory organoid systems to try to get more complex. Obviously, some of this is not easily done in any kind of animal system. But we think that some of those more complex organoid systems would be good. They also would allow us to look taking primary cells from different donors to see the effects of different changes or different illnesses in this.
Does M2 have any role in the formation of inclusion bodies, or is that only dependent on N plus P?
For HMPV, you can form what looks like an inclusion with only putting in N and P. You do not need to put in M21. Now, M21 for RSV has been suggested to play an important role, and it's one of the, it's a protein that's involved very important in the transcription replication process. So it is present in inclusions. The question is whether it's a scaffold. And it's one of those things that we'll continue to look at. So far, our data keeps supporting the idea that at least for HMPV, P is that critical first step, and it's bringing the other stuff in. And in a cell, I think P has so many binding partners that it needs something that makes it coalesce, and I think that's N. If you put P and M21 in together, you don't form an inclusion. But I do think then M21 is pulled in.
Just one or two quick questions more. Well, I recently heard a talk by Roy Parker, who was talking about RNA-RNA interaction as well as protein-protein interaction in forming liquid-liquid aggregates like this. Do you see any effects of RNA in your in vitro droplet formation?
That is a fantastic question. And one, it's an area that we're actively pursuing. Certainly just adding generic RNA doesn't have we see it go to these regions, but we don't see a major change. But there's been some really nice work from the SARS-CoV-2 system that suggests that the structure of the RNA itself may matter. So far we've only tested generic fluorescent RNAs, but what we don't know is if the structured RNAs that, for instance, form these 3 and 5' regions of the genome might play a very different role. And in fact, they've shown that for SARS-CoV-2 and the N protein, the structure of their in vitro phase separated regions look different depending on what RNA they put in. So that's actually something we're actively working on right now.
Just one last question. Well, why does P protein form droplets? Does it have a hydrophilic and hydrophobic domain? I mean, can you make any sense of why it does this?
Yeah, I at least can start. So P has a number of characteristics that make you think it could. First, as I said, multivalency. So valency refers to how many binding partners you might be able to fit in a single part. Are you able basically to pull it in? And because P is a tetramer and it has the ability to bind a wide array of things through both its N and C terminal intrinsic disorders regions, it has a very large valency. But if you look at its sequence, P has regions that alternate between positive and negative charge. Which could well be involved in PP interactions that can help drive this process. So we think that's at least a portion of why P on its own can form droplets. It is interesting, we see this clearly with HMPV. Multiple people have done the experiments, we've expressed the proteins multiple times, but it's not necessarily seen with the paramyxoviruses, for instance. So but HMPVP, even from RSVP, is quite a bit larger. So it may be that this virus brings something a little bit different.
Okay, wonderful, Becky. It's really been a pleasure. I think we need to sign off for the moment, but thank you so much.
All right, thank you.
Really enjoyed it. Okay.
So we'll be back at 1 o'clock with Richard Kuhn. So I hope most of you will be able to join us at 1 p.m. And we'll take a break right now and hope to see all of you back at 1 p.m. With Richard Kuhn, who will be our first speaker for the afternoon session. Thank you.
Okay, so let's get started. So it was an excellent first morning session, a little bit of a hitch, but overall went well under the circumstances. And it is really my pleasure to introduce Richard Kuhn to today's symposium. Richard did his PhD with Eckhart Wimmer and then did a postdoc with Jim Strauss at Caltech. Needless to say, Eckhart and Jim are legends in the positive sense RNA virus field. And then from there, Richard got a position at Purdue University. And just as Harvey had mentioned this morning, collaborations are an important part of doing great science. And he began a collaboration with Michael Rossman at Purdue as well. And it's been an amazingly productive collaboration between the two groups. Sadly, we lost Michael last year, but Michael was a real giant in the world of structural biology of viruses and viral proteins. And Richard was a big part of that interaction with Michael. And Richard's interests have been, as I said, positive sense RNA viruses, specifically looking at alpha viruses and flaviviruses. And I don't need to inform this audience, but flaviviruses do have a tendency to spike up every once in a while and spread very quickly across the planet, as we saw with Zika. And Richard has worked with Michael on a number of structures. Specifically, as of 2016, they did the first Zika virus structure. So RNA viruses and flaviviruses have been a big part of Richard's career, and he will talk a lot about that today, I'm sure. One of the really great things about Richard is his ability to understand the complexities and nuances of both structural biology and molecular virology, and does a great job going between both and explaining both, so as we will see shortly. So with that, I want to welcome Richard and thank everybody for coming back after lunch. So take over. Let's get these. Hopefully this works. Excellent.
Okay, so you see it?
Yes.
Okay, great. Still see it?
Yes.
Okay, all right. So thank you very much, Joe. I want to thank the committee. I want to thank Joe and the committee for this opportunity to talk to you today. It's really a great privilege for me to speak at the Norman Salzman Memorial Symposium. He was really a giant in terms of research, and he was a giant in terms of mentoring students. His legacy is hugely tremendous, and I'm just so pleased to be able to talk to you a little bit about some of the work that, as Joe mentioned, my colleague Michael Rossman and I have done for the last 20 years or so. So what I want to talk about is flaviviruses and really focus primarily upon dengue. And we published the cryo-EM structure of dengue virus, and I'll go through that in a bit, in 2002. And we thought at that time that we had the structure, and that's pretty much all we needed to know about what we called at that time the mature structure of dengue virus. Well, it turns out that we had a lot to learn. We started trying to understand how the virus initially is formed in the ER lumen and how it undergoes a maturation process that allows the virus to become fully infectious. So we did a lot of those studies. I'll quickly go through those things. But it turns out in retrospect that in the case of dengue viruses in particular, that they never really grow up. That is, they never fully mature, or only a very, very small fraction of those viruses mature. And so we wanted to understand what's the reason for that? What's the biological consequence of that? We also found out along the way that dengue virus and flaviviruses in general are actually dynamic. So the very static structures that we've generated using, we and others have generated using cryo-electron microscopy, is a average snapshot of the virus. And so these viruses are very dynamic, and that dynamic property probably plays a significant role in the life cycle of the virus. And in particular, we've shown that temperature can alter the state of the virus. And I'll walk through some of the experiments that we've done to show that. And then in the last part of the talk, some more recent work that we've done that suggests that maybe some of the flaviviruses, if not all of the flaviviruses, are not perfect icosahedral structures like Michael and I first proposed, rather that they are imperfect and it's a consequence of their assembly process. So my lab has been interested in over the last few years of how the virus structure, the heterogeneity, and plasticity of virus particles can influence the tropism and the disease of these viruses. And dengue is a good virus to study that, because I'll show you in a few seconds one reason why. So these are, as Joe said, these are positive strand RNA viruses. They're rather small in size, being only about 50 nanometers. They have a lipid bilayer membrane. They're found all over the place. There is at least 70 members of the genus flavivirus, which include dengue, yellow fever, West Nile, and Zika. And these viruses cause a wide range of different clinical conditions in humans. Many of them are going to be asymptomatic infections, something we're seeing very frequently with COVID-19. But again, they can also range to a full encephalitis and death. And there are three genera within the Flaviviridae family, and I'm only going to talk about the flavivirus genus itself today. So the reason why dengue is an interesting and important virus to study is because it affects so many people in the world. The estimate is that there may be up to 390 million dengue infections on a yearly basis worldwide, with maybe about 50 to 100 million apparent cases of dengue fever, and a much smaller number of the severe form of the disease, which is called dengue hemorrhagic fever. And in this form, there can be death associated with it. So dengue is probably the most important virus that is transmitted by an insect in the world. I said that the genome is a single strand of RNA. It's rather small, especially compared to COVID-19, of only about 11 kilobases. In those 11 kilobases contain all of the proteins required for replication of the viral genome, antagonism of the host immune response, and the ability to package that genome and move it to uninfected naive cells. And that's done by this virus particle that is simple in terms of its composition. It contains a lipid bilayer. On the inside is this single molecule of genome RNA together with capsid protein. There are two envelope proteins that are embedded in the lipid bilayer. One is the major surface protein, the envelope, and the other is a minor protein called M. And early on, we were able to show that the virus has icosahedral symmetry. So back in the earlier days, and 20 years ago is not the earlier days of cryo-EM, but today it seems rather ancient. But that allowed us to go to reasonable resolution in terms of a reconstruction of the virus structure. This is just a crude schematic showing what I just described to you, that M protein is a rather small protein that sits underneath the major surface protein of E. And as many of you know, and I will show, the interesting thing about the flaviviruses is that the surface glycoprotein in the mature form lies flat against the virus bilayer. Unlike most of the other viruses that we're familiar with, HIV, flu, COVID-19, in which these envelope proteins are projected outward and in a tangential organization relative to the surface of the virus. So in the genome, the three proteins that we're going to be interested in are at the N-terminus of the polyprotein, the capsid protein, a precursor to the small M protein, which is called the precursor M protein, and the major surface protein called the envelope protein. And the envelope protein is responsible for both attachment to cellular receptors as well as fusion under low pH conditions in the endosome. So this is the structure we did about nearly 20 years ago now of dengue virus. You can see that it is rather featureless on the surface, and it resembles a golf ball in appearance in that it does have little small bumps here and there. But what may not be apparent, but I will show you, it does have this symmetrical organization of its surface proteins, the E and the M protein. And we were able at that time to be able to position an atomic structure that existed at that time, which was done by Felix Ray and Steve Harrison and Franz Heinz, of another flavivirus envelope protein, which was done by crystallography. We were able to dock it in to the structure that we had determined by cryo-electron microscopy. And in so doing, we were able to show that within the repeating unit of this icosahedron, there were 3 molecules of E and 3 molecules of M. And the E was organized in the form of a dimer that was repeated 90 times on the surface of the virus particle. And here you have the fitting of the atomic structure of the E protein into the cryo-EM structure of dengue virus. And right here is a twofold axis of symmetry, and these 2 molecules of the E protein sit right on that twofold axis of symmetry. I don't have time to really talk about the implications of this organization in terms of biology, but there are interesting aspects that, because of the way these proteins are arranged, has consequences for the way that antibodies bind and neutralize, and by the way, presumably receptors are able to bind to the virus particle. This is a cross section. This is now showing Zika. Joe mentioned in the introduction that we did the structure of Zika shortly after the virus was recognized as a pandemic type of virus. And on the outer surface are the envelope protein, and then underneath is the small M protein. And we can see going across a lipid bilayer membrane, which is shown here and here, there are transmembrane regions of the E and M protein that penetrate toward the inner leaflet of the bilayer, but do not penetrate inside, and therefore they don't connect with the inner nucleocapsid core and genome RNA. So there is a gap that exists, which has been interesting for us and many other groups, and I'll have more to say about that a little later in the talk. We had done studies that showed what the maturation process of flaviviruses looked like upon formation of the virus particle as it is budding into the ER lumen. The virus actually does have a spike-like appearance, and that spike-like appearance is contributed by a heterodimer of the precursor M protein, PRM and E. 3 of those PRM-E heterodimers come together to form a spike-like projection. Now, this particle, this immature particle, which is non-infectious, is going to be secreted, move through the secretory pathway. And when it encounters the low pH that is present in the trans-Golgi, a conformational and translational change will occur in the particle, such that the heterodimers will essentially be broken, and we will now reform or form homodimers of the E protein. And these E proteins will now lie in a flat configuration. This rearrangement allows for the enzyme, the host enzyme furin, to come in and recognize the cleavage site in this folded down structure. The cleavage site is not present in this spike-like immature structure. Furin can then cleave, and under the low pH conditions, the PR domain, which is now cleaved, remains bound to the virus particle, protecting a fusion loop that's present on the E protein. And that PR is not released until the virus particle is secreted into a neutral pH environment of the extracellular milieu. Once this occurs, this is now considered to be a fully infectious virus particle. And we have structures of all of these states. This is what I had previously shown you. This is now the case of Zika virus that Michael and I did a few years ago, together with some of our colleagues. In this structure, I showed you already the fitted C alpha backbone of the E protein. This is the structure of the immature form of Zika virus, in which we have these trimeric spike-like projections. Now, interesting, in Zika virus, we were able to see in the immature virus that there might be some connectivity between the E and PRM proteins and the nucleocapsid underneath the bilayer. And I'm going to come back to that later. We also have high resolution structures now of all of these proteins. And this is just showing some of those high resolution structures. This is in the immature form. This is Zika virus once again. And this is showing with the C alpha backbone. And this is the mature virus. So you can see that there is a significant change in the organization of these proteins as they go from the immature to the mature form of the virus. So one of the things that was really surprising to us very early on, this is probably around 2008, this is a collaboration that we had with Michael Diamond and David Fremont at Washington University, as well as John Roerick, then at CDC, and Ted Pearson at NIAID, was that if we took dengue virus and we added a neutralizing monoclonal Fab fragment to it, at room temperature, we could not get binding of that Fab to the virus particle, even though it's a neutralizing antibody. But of course, neutralizing assays are done at 37 degrees and not at room temperature. And so what we then did was we incubated this dengue virus particle and the Fab antibody at 37 degrees, and we were able to demonstrate binding. And when we did the cryo-EM reconstruction of that Fab-bound virus particle, we had found that the virus had expanded and that the E protein had changed its position from lying flat to a little bit elongated and projecting upward. And our interpretation at that point was that this probably represents some aspect of breathing of the virus particle at elevated temperatures. And that at low temperatures, these sites that this one antibody bound to was not available. And so it needed to bind at 37 degrees. We also did some studies just looking at the transition between a low temperature structure, say at 22 degrees room temperature, and a virus particle at 37 degrees. And we found that the virus particle at 37 degrees is bumpy. Well, we called it bumpy, or Ted called it bumpy. And here's a little schematic of what we could imagine might be going on with the virus particle as different E proteins are moving around on the surface. Now, I want to come back. This is data from many years ago. When we first did the reconstruction of dengue virus, we found that there were a lot of what we said were damaged particles. If I can point out, for example, this red arrow or these particles here, you can see that they're not homogeneous like this one right here. And at that time, we just decided and thought that these were damaged virus particles, and we actually threw them out of the reconstruction, and we only used these nice homogeneous particles. Well, in retrospect, it turns out that the majority, in the case of dengue 2, lesser for the other dengues, but still they're variable, these viruses are not homogeneous when they come out of a cell. In fact, they are a mixture of a few mature virus particles, a lot of partially mature virus particles, and a few immature virus particles. The interpretation is that this cleavage by host furin is not a very efficient process. And so what we see is a continuum of particles of variable maturity, and that's because of this inefficient cleavage. The cleavage is required for a particle to be infectious. This immature virus particle has been shown to be non-infectious. These partially mature particles are probably infectious, but to varying degrees. The presence of PRM on a virus particle probably does influence its ability to interact with receptors and antibodies. In fact, antibodies against PRM have neutralization activity and have the potential for this antibody-dependent enhancement that is a significant concern for the dengue viruses. And then finally, patients who have the more severe form of dengue hemorrhagic fever, seem to have elevated levels of antibodies against PRM protein. So that clearly in patients, they too are seeing virus particles that contain the PRM protein. And so that might be an important aspect of the virus biology. We set out, this was work by Devika Sarohi, at that time a graduate student, to quantify using a parallel approach to our visual imaging that we were doing using cryo-electron microscopy. She established a mass spectrometry method of selected reaction monitoring in which she was able to then measure the amounts of PRM in various flaviviruses. And we've looked at a variety of flaviviruses. I failed to tell you that there are distinct dengue viruses. Each of those dengue viruses are able to cause a similar type disease. And antibodies, of course, against one serotype do not protect against the other serotype, although they may actually bind to the virus and be responsible for the antibody-dependent enhancement to the more severe disease form. Other flaviviruses, such as Kuhn, which is a relative, a very close relative, an attenuated relative of West Nile virus, and Langet, which is a tick-borne virus, do not show such heterogeneity, and their cleavage of the PRM protein is far more efficient than that we see in the dengue viruses. And this is just comparing dengue 2 and dengue 4. Dengue 4 is better in terms of PRM cleavage. I won't go into the details too far, but when we measured using mass spectrometry, we found that a significant fraction of the dengue 2 preparations contain partially immature and immature virus particles, more so than the amounts that we saw for dengue 4. And if we put it on this, this graph here, we could see that for a number of different strains, I'm only showing one strain here, but we've done it on multiple strains of dengue 4, that we get a cleavage efficiency that is probably about 80 to 85% of most dengue 4 strains. On the other hand, when we look at dengue 2, we can find our laboratory, our common laboratory strain, this one here, 16681, is less than 50% cleaved. That means that particles coming out will likely have significant fractions of PRM and therefore are partially immature virus particles. And so this PRM content, this bumpiness that we can see as we elevate temperature, which is related very closely to the concept of viral breathing, clearly has a significant play in terms of heterogeneity of the dengue viruses. They're far less homogeneous than the other flaviviruses. And we really like to understand What's the reason for that? So why does dengue maintain this inefficient cleavage? I'll just say right now that I don't have the answer for that. Oops, sorry, I want to just go back here a second. If I can. I guess I can't go back. Oh, there you go. Furin host enzyme, it has a preference for basic residues in the P1, P2, 3, 4, 5 positions. And if we look at the flavivirus PRM cleavage site, we see that in most of the flaviviruses, they do not have a basic amino acid that resides at the P3 position for the furin cleavage site. Not only is that true, but the dengue, the 4 dengue serotypes, in fact, they don't even have these nonpolar amino acids. They have acidic residues that sit in the P3 position. So it's clear that this is one of the determinants for the inefficient cleavage of PRM in the flaviviruses relative to the other flavivirus. If you change, well, I should say it's an RNA virus, and so mutation and selection, goes hand in hand, and the error frequency is very high in these RNA viruses. And so if we just had an amino acid that was a bad amino acid for cleavage, we could very easily select for viruses that had a better cleavage site. That doesn't happen. The flaviviruses, dengue in particular, maintains the acidic residue at that position. And so that's a requirement for some reason in terms of the biology. Viruses that you mutate to a basic residue, which have a higher efficiency PRM cleavage, do not grow as well in cell culture as those like the wild type virus. So I'm just going to summarize this part of the talk right now and just say that PRM content is going to influence the structure and the dynamics. If you have now PRM mixed into this surface of packed E proteins, it's going to influence the ability of those proteins to move around. And it may influence the half-life of the virus. It certainly is going to affect the infectivity of the virus. It may also have an effect on the entry of the virus particle into the host cell. It certainly will affect the immune response, and that will then affect disease and pathogenesis. And so let me now turn to the last part of the talk and talk about imperfect icosahedral symmetry. A student of mine a few years ago, Matt Thurkelson, wanted to understand what was the process by which we went from an immature virus particle to a mature virus particle. What was the pathway of the E and PRM and M proteins as they underwent this transition between the immature state and the mature state? And what Matt wanted to do was to come up with a strategy for blocking the transition, hopefully somewhere in between the immature and mature states. And what he decided to do was to pick an antibody, which we had previously characterized. This was an antibody from Michael Diamond's lab at WashU, which was called E16. And we knew that the Fab fragment from E16 should only bind to the mature virus because the epitope is not present on the surface of the immature E protein. So Matt decided to use this antibody to trap some aspect of the transition state. And so this is, these are 2D class averages. This is just looking, Matt decided to use Kuhn rather than West Nile because of the BSL-3 constraints around West Nile. That E16 still binds to Kuhn. We had done work with Ted and Mike on an antibody against PRM in dengue virus This is work by Devika Surohi. And in that work, we showed that we were able to bind one Fab at the very tip of PRM to the surface of the virus particle. And this is what those 2D class average particles look like. But when Matt did the same type of experiment with Kunjin and took immature Kunjin and mixed Fab E16 to it, he could not get a good 2D class average. The particles were somehow affected, and he could never come up with a reconstruction of this virus particle in complex with E16. Now, of course, this is not, E16 is not supposed to bind to the immature Kunjin virus particle. And so this was very strange to us because it was very specific to the addition of the E16 Fab. So what Matt did was, rather than use symmetry, icosahedral symmetry, to generate the structure of the virus, he decided to look using asymmetrical reconstruction. And so he took Kunjin virus, and instead of doing a icosahedral refinement, he did an asymmetric one in which we don't use the 60-fold symmetry that we have from the icosahedron. And when he did that, he found that the virus particles were correct on one side, but somehow defective on another side of the virus particle. And you can see this area right down here seems blurred out. And so he decided to look at that a little bit more carefully and do the reconstruction. Now, the icosahedral reconstruction is much higher resolution than the asymmetric reconstruction because we imposed 60-fold symmetry. Same data is used in the top set of images here as the bottom set. It's simply the way the reconstruction was done. Without imposing symmetry, what he found was in these immature Kunjin virus particles, the surface of the particle was fine on one side of the virus and deformed on the opposite side of the virus. We called one the proximal pole, the other the distal pole. The proximal pole, it turns out that when you look in cross-section like we're doing here, the nucleocapsid has attached to the glycoproteins on this proximal pole. So it's really packing up against the lipid bilayer on this proximal pole. And there's a significant gap that exists on the opposite side on the so-called distal pole. And you can see that reflected in these diagrams here. And so there's the clear density. This is the wild type virus. I shouldn't say the wild type, I should say the one using icosahedral reconstruction. And now this is the one that doesn't use icosahedral reconstruction. You can see that a large fraction of the virus surface is the same, and yet we have a region here that is deformed. This is a morph movie that shows the transition between the icosahedral reconstruction and, oops, I messed that one up. The icosahedral reconstruction and the non-icosahedral reconstruction, that there's a region right around here that is distorted. You can see that here. And it's not exactly at the 5-fold axis, it's slightly off the 5-fold axis, which we thought is a little bit interesting. Our interpretation is that we don't have exactly the right number of E and PRM subunits to have an icosahedral structure. So that means we are missing perhaps 3 to 5 E proteins and PRM proteins. And that causes a dimple, if you will, on the surface of the virus particle in this region right here. Was that some kind of artifact with Kunjin virus? We did it again with Zika virus, and we see the same type of asymmetry in the immature virus particle. We then asked, what about the mature virus particle? Is the mature virus particle saved? Does it now have a perfect icosahedral symmetry? So once again, this is using icosahedral symmetry to impose averaging. And now this is the asymmetric process. And in the asymmetric process, again, there is a there is a proximal pole that is good, and on the opposite side, there is a defect in the glycoprotein organization of these viruses. Well, we don't know the answer to this yet, but here's a model that I propose for you all to consider. The flaviviruses are going to be forming on the surface of the ER lumen, and they are going to be wrapping. The glycoproteins are in the luminal surface. We have the RNA and nucleocapsid forming on the inside surface, and they're going to begin associating and wrapping around that nucleocapsid core. And everything is fine until you get close to the end and you have a near formed virus particle. And now you're going to have steric clashing between these spike proteins, these PRME spike proteins. And perhaps because of this steric collision in the budding process, as we go to pinch the neck, we don't pinch the right number, the exact number of E proteins. And so we wind up with a distal pole that has a deficit, and that deficit is continued as we go through to the mature virus particle. So let me just conclude. I just told you that the core is eccentrically positioned in the immature flavivirus. It reorients itself in the mature viruses. There are deviations in icosahedral symmetry. These asymmetric features are possible consequences of assembly and budding. This is what we're looking at. And we propose that we have fewer than 180 copies of glycoproteins. That is the result of the imperfect icosahedron. This imperfection allows for increased flexibility and may contribute to the ability of these particles to be dynamic. And lest I miss something that Michael was very keen about, he thought of the analogy with the bacteriophages. And he thought that this dimple or this distal pole defect may actually facilitate entry and or fusion, or may have some other biological role. So with that, I want to thank you for your attention. This, of course, all the work that I talked about here was a long-term collaboration with Michael. Michael and I collaborated for about 27 years before he passed away in May of last year. We had lots of interactions during those periods. I just want to mention Devika Sarohi, who did the Zika virus structure and also looked at the PRM content using mass spectrometry. Andy Miller also participated in some of the reconstructions, and Matt Thurkelson did the last part of the talk that I gave. Thomas Close has been one of our great experts in cryo-electron microscopy with Madhu Zavana, who is a research scientist in the group who's also contributed. I should say, I didn't talk about Baldeep Kaur's work, but Baldeep has a structure of a Tsutu virus, another flavivirus, below 2.5 angstrom resolution. This has been a long-term collaboration with Michael Diamond and David Fremont at WashU, and with Ted Pearson, my friend and colleague from NIAID, and with Kim Dowd, who's been there all the way. So thank you.
Okay, thank you, Richard, for a really interesting talk. We do have a few questions that people put in the chat. So let's see, I can read them to you. I'm not sure if you can see them. So Adam Zlotnick asks, Richard, your partially mature dengue appears to have mature and immature phases. Is this an artifact of the reconstruction approach, or is there a processive maturation process indicative of communication between adjacent envelope proteins?
Yeah, that's a tough question, Adam. I think, you know, we've looked a lot at the artifact question, and it doesn't seem to be an artifact of reconstruction. Is it a processive maturation process? You know, that was work that we published many years ago, in that we think that there is progressive maturation, and that it starts at a single point and wraps around. I think that that's true, but I think we need to do more experiments to actually validate that that statement. So yeah, I don't think we're absolutely sure on that yet.
Okay. Another question, does the heterogeneity of viruses identified differentially by host antibodies, whether immature viruses are facilitated by ADE to enter cells while mature are sufficient to bind the receptors independently? So 2 questions there.
Yeah, so does the heterogeneity of viruses identified differently by host antibodies? Clearly there are sites that are present in mature virus that may not be present in PRM containing viruses. And there are also sites that are cryptic until they're exposed. So the example that I gave you was with this 1A1D2. This captures a state of breathing of the virus particle and then presumably traps the virus particle in that state. There is heterogeneity and antibodies will detect that heterogeneity. Small differences in the breathing and the surface features will and have been shown by different antibodies to bind differently. So that's certainly true. Whether immature are facilitated by ADE, this is an open question right now. There has been work that was done by Yolanda Smith's group and others who have suggested that immature viruses can get into cells and be cleaved once they enter cells.
Okay, we have a question from Stuart Ray. Should references to, quote, cell culture, quote, host cell immune response, and other contextual comments about dengue virus include explicit recognition of the additional complexity imposed by invertebrate vector, the invertebrate vector?
Yeah, so, you know, we and others, when we've done our structure studies, have used a mix really of both vertebrate cells and invertebrate mosquito cells to grow virus. And in terms of the furin cleavage, so the amount of maturation, even though there are different proteins really that are cleaving at different versions of furin, the amounts of cleaved versus uncleaved appear to be pretty similar. And so we think that the determinant for the cleavage efficiency is mostly held at the virus level. Clearly, there are differences in terms of the glycosylation, but again, the experiments that we and others have done doesn't show that the level and type of glycosylation makes significant difference in terms of infectivity of the viruses. So, you know, when we first came up with this breathing and this 22 versus 37 degree observation, we were pretty excited because we thought, oh, you know, the mosquito-derived virus has one configuration. The virus, once it enters an infected host, is in a different configuration. And of course, the host may come up with a fever, raising the temperature further. But it turns out in the dengue viruses, only dengue 2 shows that particular behavior of the 22 to 37 degree switch. The other ones are still breathing. We have data from that, but they don't have this elevated structure that's present at 37 degrees.
Okay, one more question from Ziming Zhang. Is there viral RNA in the immature particle?
Yes, the viral RNA gets encapsulated by the capsid protein and is there when the virus particle buds as an immature particle into the ER lumen. We have never really gotten a very clear structure from that core particle, but it's clear that the RNA is in the vast majority of those virus particles.
So, okay, well, in the app, you know, we should probably move on. So thank Richard virtually again for a very nice talk, and we'll move on to the next speaker. Thank you, Richard.
Thank you, Richard. I believe Mrs. Salzman will introduce the next speaker, our next award winner.
Yes. Thank you. Welcome back after a very exciting and stimulating morning and a good beginning with Richard Kuhn's talk. I had introduced Victoria Avanzato this morning, and she is the first graduate student award recipient for a Salzman Award. And I will not redo what I said this morning. I'm going to turn it over to Victoria now and let her have the forum for her talk. Victoria, welcome.
Thank you. Awesome. So I'm Victoria Avanzato. I'm a fourth-year grad student in the OxCam program. I'm really excited to be here today. Thank you for the opportunity to share my work with everybody. I'm working with Vincent Munster at Rocky Mountain Labs and Thomas Bowden at the University of Oxford. And today I'm going to talk about some of my work about the neutralization of Nipah virus via antibody-mediated targeting of the fusion glycoprotein. So an introduction to Nipah viruses. The prototypic Nipah viruses are Nipah and Hendra virus. Both of these are highly pathogenic paramyxoviruses that cause severe respiratory and neurologic disease in humans. This is often highly fatal, and outbreaks have fatality rates anywhere between 40 and 100%, depending on the outbreak. So Hendra virus first emerged in Australia in 1994. This was associated with horses and the trainers, and continues to cause sporadic spillover events to this day. Nipah virus had an initial emergence in Malaysia in 1998 and caused a large outbreak of encephalitis. This was associated with pigs and pig farm workers and resulted in over 100 deaths. And it has not since reemerged in Malaysia, but continues to cause near annual outbreaks in Bangladesh and India. And there was quite a large severe outbreak in India in 2018. So both of these viruses circulate in host Pteropus bat species. So these are fruit bats. And the transmission to humans occurs either through intermediate hosts, which was the case with Hendra virus in Australia was through the horses, and Nipah in Malaysia was through pigs into people, or direct transmission from bats to humans, primarily through the consumption of contaminated date palm sap. In the more recent outbreaks, human-to-human transmission has been observed, which is concerning. And currently there are no licensed vaccines or therapeutics against either of these viruses, which highlights the need for more research to develop countermeasures. The paramyxovirus surface is densely populated with the surface glycoproteins. So there are two, there's the receptor binding protein and the fusion glycoprotein. So on Nipah virus, this is G, which binds the host cell ephrin receptors, and the F protein, which then mediates membrane fusion. As these are surface exposed and thus accessible to the host immune system, they make attractive targets for the development of vaccines and therapeutics. And a lot of the vaccine candidates in development aim to elicit antibody responses targeting the glycoproteins. And also there have been several studies showing monoclonals or passive serum transfer of antibodies targeting these proteins does protect against disease in several animal models. So it does show that a neutralizing antibody response against these glycoproteins is beneficial in combating these viruses. So the project that I've been focusing on for my thesis has been working to characterize a panel of monoclonal antibodies that were generated against Nipah G and Nipah F. These were actually made by Ben-Hur Lee at Mount Sinai through the vaccination of rabbits with a combination of DNA and soluble glycoprotein vaccination. And then they produced the hybridomas, and we were able to sequence them to get the variable regions to produce these antibodies for our studies. So the one that I'm going to focus on today is called MAB66, and this one targets the fusion glycoprotein and has activity against both Nipah and Hendra. So the project goals were, we wanted to define the epitope using X-ray crystallography. So we aimed to get a structure of the antibody in complex with the Nipah fusion protein, and this would allow us to identify the key interactions and elucidate how the antibody neutralizes this virus. And this would also allow us to identify vulnerable targets on the virus surface as kind of the broader goal. So as I mentioned, this both of my labs are heavily involved with this work, but it was also a collaboration with Ben-Hur Lee and his grad student Sope. So together we all set out on a structure function characterization of 66. So the target is the pre-fusion conformation of the NEPA fusion protein. And this is a class 1 fusion protein that is displayed as a trimer on the virus surface. 3 functional domains, domain 1, domain 2, and domain 3, which are color-coded in this diagram, with domain 3 being the pink one, which is the most membrane distal, and then domain 1 and 2 are yellow and red. It also has a cleavage site and a fusion peptide, which are required for the activation and fusion function. So during the production of the F protein, it goes under processing in the host cell, and it needs to be cleaved to allow the release of the fusion peptide for insertion into the host cell membrane during fusion. There are also 2 heptad repeat domains that are important for the fusion function. So HRA is within the D3 domain, and HRB is the blue region there. It's the membrane proximal stalk. So when the virus undergoes fusion, the fusion protein goes under a series of conformational rearrangements, and these heptad repeat domains come together and drive the fusion of the virus and host cell membrane. So this was the target of the antibody we're going to be talking about today. So the first thing I did here was determine the crystal structure of the Fab region of this neutralizing mAb in complex with the Nipah virus fusion protein trimer. So the crystal structure revealed an epitope at the membrane distal apex of the trimer. And we see that there are 3 Fabs bound to the trimer apex. There's 1 Fab bound per protomer, which are shown in different shades of blue here. And I will note that there are we did not observe any major conformational changes between the aligned structures of the Nipah fusion protein with the antibody bound and the unbound form. So we don't believe that this antibody induced any major rearrangements here. So taking a closer look at the epitope, looking at the contributions of the CDR loops, we can see that based on the interface area that is contributed by each of them, the interaction is actually dominated by CDR3 on the light chain, which is a bit different than the canonical heavy chain CDR3 domination. So we see that the loop is actually inserted kind of into the shallow depression on the Nipah surface in between these 2 helices and an apical loop. And then looking more closely at the epitope, we can see that there are several key tyrosine residues on that loop that form important interactions with several residues on the Nipah surface. Obviously, there are other important contributions from the other CDR loops as well that we can see in the background, particularly the heavy chain CDR3, which is shown in pink, and the other light chain CDR loops, which are seen in the background here. What was interesting about this epitope actually is we noticed some glycosylation at the periphery of the epitope. So the Nipah fusion protein has 5 end-linked glycosylation sites in the sequence, and previous work has shown that 4 out of the 5 are occupied. And consistent with this, in our crystal structure, we noticed electron density supporting the occupancy of the same 4 out of the 5 sites. The structure, I did deglycosylate the protein to aid with crystallization. So we have the first N-acetylglucosamine residue modeled here. And interestingly, we noticed that the second glycosylation site, which is referred to as F2 throughout this presentation, this was at the periphery of the MAB66 binding interface. And we can see in that second panel, the 2 light chain CDR loops both interact with it, and both the asparagine and the glycan itself contribute to this interaction. As I said, this protein was deglycosylated, so that raised the question of whether a full-length glycan at this site would inhibit the antibody binding, as there's plenty of literature suggesting that glycosylation can shield viruses from neutralization. So what I did is I was able to model a full-length complex glycan on both the Nipah fusion protein and also was able to model the Hendra fusion protein based on the structure information available. And I was able to model a full-length glycan into this site, and glycans are fairly flexible, which I've kind of noted with this arrow here, but based on the modeling, it doesn't look like the presence of the fully processed glycan at this site would inhibit the antibody binding on either that corresponding protomer or the neighboring protomer. The CDR loops approach from the side, and it's likely that they're able to still access their epitope despite the presence of the carbohydrate. To confirm this, we did a functional assay. So this was a cellular-based binding assay where we expressed the wild-type fusion protein for both Nipah and Hendra, and also created a mutant in which this F2 glycan site was removed, and tested MAB66 binding to both. And we did not observe any significant difference in binding between the wild type protein or the protein without this glycan site. So we don't believe that the glycosylation here inhibits this antibody binding. Also, previous work has shown, and our binding data as well, has shown that MAB66, it has activity against both Nipah and Hendra. However, it is about 10 times more potent against Nipah. So looking more closely at the epitope to compare these two viruses here, I have the epitope mapped onto the fusion protein with the contributions of the light chain and the heavy chain. And looking at the sequence alignment, the epitope is very conserved through all the Nipah sequences, but there are two differences in the residues in the epitope between Nipah and Hendra that may be contributing to this difference. So the residues at position 70 and 74 differ between the two viruses, and there is a lysine substitution at position 70 and a threonine substitution at position 74, which I have noted in red on the above structure. So both of these residues form extensive contacts with MAB66. So the top two panels show the antibody bound to Nipah, and we see these two residues. So at position 70, we see that side chain interaction with both heavy chain and the light chain. And at position 74, we see the interactions with the heavy chain. So we were able to model this onto the Hendra fusion protein structure to see if there were any structure rationale as to why it would be binding this virus less. And we can see that the substitution at position 74 doesn't look like it would introduce a major steric inhibition to the binding. However, the lysine substitution at position 70, certain conformations may be accommodated, but certain side chain conformations, this is a larger side chain, so certain conformations may be less favorable for MAB66 binding to Hendra, particularly with the positioning of that heavy chain CDR loop we see in pink. To confirm this, we created reciprocal mutants. So we mutated residue 70 and 74 in the fusion protein and replaced the Nipah fusion protein with those Hendra residues, and then put the Nipah residues into the Hendra fusion protein, and tested MAB66 binding to these reciprocal mutants with the same cellular-based binding assay, and also with a VSV-based neutralization assay. And quite remarkably, we noticed that introducing the Hendra residue in binding by MAB66, but introducing the Nipah residues back into the Hendra fusion protein actually enhanced the binding and neutralization by MAB66. So it's possible that other local differences on the fusion protein surface may be contributing to the difference in binding, but this does support that these sequence differences at position 70 and 74 do contribute to the binding and neutralization differences that we observe in vitro for this antibody. As I mentioned before, the surface of the virus is densely populated with a heterogeneous mixture of the receptor binding and fusion proteins. And that led us to believe that perhaps the most membrane distal regions were the most accessible to the host immune system. And consistent with this, we do notice that our FAB66 or our MAB66 epitope is at the membrane distal apex. So we wanted to evaluate if this region of the fusion protein was under selective pressure by the host immune system. So to do this, we performed a series of DNDS analysis on the fusion protein. So to do this, we looked at the entire coding region of the protein, but also divided the protein up into 4 groups based on the functional domains and the membrane proximity. So group 1, it would be our domain 3, which contains that apex and our MAB66 epitope. Group 2 has domain 1 and domain 2, and then group 3 and group 4 are membrane proximal stalk and transmembrane domain. So there was no evidence of positive selection acting on Nipah-F for the whole protein or for any of the functional groups we defined. So one potential weakness of this analysis, though, was the relatively small amount of Nipah sequences available at the time we did this. So to add more power to the analysis, we performed the same analysis also on the measles fusion protein and the parainfluenza 5 fusion protein, which are other paramyxoviral fusion proteins, but they had more sequences available. And we also found no evidence of positive selection acting on measles or PIV5 either. So this does suggest the presence of functional and structural constraints on the paramyxovirus fusion protein, which may make it an attractive target for therapeutics. So also we wanted to speculate on a potential mechanism for this antibody. So I mentioned that the protein goes under significant conformational rearrangements during the process of membrane fusion. And a lot of these rearrangements happen within the D3 domain, which is where our MAB66 epitope is, which I've colored red on this structure. So there is no post-fusion structure available of NIPA-F, but there is a post-fusion structure available of parainfluenza 3 fusion protein. So I was able to map the equivalent residues of the MAB66 epitope on this post-fusion virus structure. And we noticed that the MAB66 epitope becomes disrupted in the post-fusion conformation. So more experiments would be needed to actually confirm this mechanism, but it's possible that antibodies binding this region of the fusion protein may prevent the rearrangements that are necessary for membrane fusion to occur. And then finally, since we had this hypothesis that this membrane distal region is maybe a common target of the host immune system, we tested the mutants that we made, the apex mutations, both the F2 glycan mutation and also the residue 74 reciprocal mutants against the polyclonal sera. So this polyclonal sera 835 is a polyclonal sera raised against the NIPA fusion protein. And we found that these apex mutations either disrupted or enhanced the polyclonal binding as well, similar to T6. And as the polyclonal mixture contains of thousands of antibodies, and the fact that we notice a difference in binding and neutralization here suggests that many antibodies in this polyclonal mixture target the apex region of the fusion protein, which does support this as a site of vulnerability on the virus surface. And that makes this membrane distal apex an attractive target for the development of vaccines and therapeutics. And kind of the last slide I wanted to show is a little bit of a sneak peek of the work I currently have ongoing. I'm working on characterizing further monoclonals targeting fusion protein apex, and we've started some in vivo studies with these. So this study is currently ongoing, but I was pretty excited about how it would show a slide. So we prophylactically treated Syrian golden hamsters, which is a model for infection with another targeting monoclonal, and we found that it didn't really have great effect against Hendra virus, but it protected really well against a Nipah virus challenge. So we will be continuing study with the antibodies to see how they work in animal models. So just my conclusions and future directions. This work has shown that the membrane distal apex of Nipah fusion protein is a site of vulnerability on the Nipah virus surface, and characterization of further monoclonals will reveal additional epitopes and vulnerable sites on the virus surface. We're currently working on in vivo testing, starting with individual monoclonals, and hopefully in the future we can test some monoclonal cocktails to determine how effective they are in animal models. And as I mentioned, our hamster work is currently ongoing. And together this work contributes to the blueprint for a structure-based design of vaccines and therapeutics against Nipah viruses. And obviously this project was very collaborative, so I have a lot of people to thank, mostly Vincent and Thomas, my two PIs, for all their support and taking me on as a student. Also Sope also worked very hard on this project, and everyone in the Laboratory of Virology and the division at Oxford and the OxCam program and the Alliance, and all of you for listening to my talk today. And with that, I'll take any questions.
Thank you, Victoria, for a lovely talk. Can you tell, it's a bit of a naive question, but your hamster model, how faithful is it to the human disease in terms of clinical, the pathogenesis of the virus?
Sure, so the hamsters do display signs of neurologic disease and also respiratory disease. So that is the disease phenotype that we see in humans. So. Okay.
And after the hamster model, the next step would be a non-human primate?
Yeah, so people will also use ferrets, and then we'll move these into non-human primate models to test the antibodies and vaccines. It's, we have studies going with African greens, and also other non-human primate models would be the next step, yes.
Okay, thank you. I think we can move on to the next speaker. Thank you so much again, Victoria, and congratulations on your beautiful work. Thank you.
Thank you.
So our next speaker is David Wang from Washington University in St. Louis. Pleasure to have David here. David is a virus hunter. He looks for novel viruses, and he's developed many important tools that we use today and algorithms to identify viruses, novel viruses in nature. David received his PhD at MIT and his postdoc training with Joe DeRisi at UCSF. He started his lab at WashU in 2004, where he is currently professor in the Department of Molecular Microbiology, Pathology, and Immunology. David, as I mentioned, has pioneered novel genomic approaches such as ViroChip and high-throughput sequencing-based strategies to discover new viruses. His lab has identified many new viruses in humans and animals and has also developed model systems to study these new viruses. Sort of related to this, the theme of this symposium and this year, coronavirus, David, while he was a postdoc with Joe DeRisi, was the first one to sequence and characterize the first SARS virus back in 2003 and to classify it as a novel coronavirus. So he's been at the forefront of coronavirus research for quite some time. So without further ado, let's give a warm welcome to David, and David, take it away.
Okay, I'm going to share screen.
Great.
So thanks very much, Nihal, for the introduction, and it's a pleasure to be here and to speak with everyone about some of the ongoing work in my lab. And as Nihal mentioned, we have focused over the past 15 plus years on identifying and characterizing and then exploiting novel viruses to try to learn more about both human disease and fundamental basic biology. And so what I'm going to do today is basically tell you one story where we've been able to build new experimental system from a novel virus and tell you what we've been able to learn about virus infections from that system. So as Nihal mentioned, in my lab, work focuses initially on trying to find new viruses, and we've done this from a huge variety of different specimens, not just diseased and healthy humans, but animals, insects, environmental samples, sewage, etc. And again, as she mentioned, years ago when I was a postdoctoral fellow, I used this approach in the development of the first pan-viral microarray, and I was able to use this successfully in collaboration with CDC and others to help identify the original SARS virus as a novel coronavirus. I am actually not going to talk about any coronavirology today. Subsequent to the microarray, we utilized high-throughput sequencing-based approaches, first using high-throughput Sanger sequencing, and of course, with the advent of next-generation sequencing platforms, adaptation to next-generation sequencing to do high-throughput mass sequencing with bioinformatics to identify novel viruses. And the premise of all this hasn't changed in almost 20 years. The idea is if you start randomly sequencing nucleic acid molecules present in a particular sample, you can computationally identify whether these come from the host or ideally whether they are viral in nature. And if they are viral, are they very similar to known viruses? Are they highly divergent? And our interest has been on trying to find highly divergent viruses with the idea that viruses have taught us so much about basic biology that the more novel and different the virus, maybe it utilizes very different pathways or has other things about biology to teach us. And so for today, I'm going to focus on this organism up here in the top right, which is the nematode C. elegans. And so just to make sure everyone's on the same page, so C. elegans is a classic model organism. It's been widely used in neuroscience and developmental biology for about 50 years. It's highly genetically tractable. It has a very rapid reproductive lifespan. So in moving from being an egg born to being sexually mature, it takes about 3 days. So this makes genetics, classic genetics and crossing highly amenable. So there are 4 genetics through chemical mutagenesis screens. There's whole genome reverse genetics that's possible. Importantly, the animals are transparent. And so this is enables facile live imaging of GFP tagged proteins. And it's a potentially very robust system to define host-virus interactions, in part because of its simplicity. So there's no known professional immune cells and no adaptive immunity that we're aware of. So there's no B cells or T cells. There's no macrophages. in terms of innate immunity, there's also no interferon, which doesn't appear until much later in evolution. And in terms of toll-like receptors, there's only one TLR homolog. So it's a very relatively primitive system. And of course, as a model, about 50% of its genes are conserved in humans. And many fundamental discoveries in biology have been made in this system, notably the discovery of RNA interference. Now, labs for about 20 years have been using this to study bacterial pathogenesis, looking at pathogens like Pseudomonas and Salmonella. And many people have proposed in the past that this would be a great system to look at host-virus interactions. But there was a major challenge or limitation, and that was that, first of all, no mammalian virus could people get to actually infect or replicate in C. elegans, and there was no natural viruses of C. elegans that had ever been described. So what are the reasons for this? It could be twofold. One is that people who had looked for viruses in the past may not have had very sophisticated or robust methods. And of course, we had spent a long time developing what we thought were the most sophisticated methods in existence to find viruses. So we felt that we had that aspect under control. The other possibility is that maybe people didn't look in the right places. And so the laboratory strain of C. elegans was first brought into the lab by Sidney Brenner in the 1960s. And it's subsequently been propagated in the lab from petri dish to petri dish for generations. But of course, if you think about a worm in a petri dish, that's not a great place to be exposed to natural worm pathogens. In a way, you can think about this as a massive shelter in place for worms for 50 years and not being exposed to any external viruses. So what we wanted to do was find the high-risk worms that were infected. And we wanted to look in the wild for wild populations in their natural habitats. And ideally, they would be sick, because this is where we would start and increase our chances of finding a novel virus. And so we wanted to look for viruses in sick wild strains. And in order to do this, we engaged in a collaboration with Marie-Anne Felix in Paris, who was a worm ecologist. And what she found early in her career is that she could find lots of wild strains of C. elegans on rotting fruits in fruit orchards. And the reason this is that, of course, decomposing fruit is due to a lot of microbial activity. There's lots of bacteria growing on the fruit. And C. elegans is a bacteriivore. And so wherever there's a lot of bacteria, you can find a lot of C. elegans. And so what Marie-Anne does is she goes all around the world collecting rotting fruits from all over the place and brings it back into the lab. Once into the lab, she'll put the rotting fruit, as you can see here, around the perimeter of a petri dish. And in the middle, she puts a spot of E. coli, pure E. coli. And so what happens is that over time, the worms will crawl out from the edges of these rotting fruits to the middle. And under the microscope, you can observe the individual worms. She can pluck off individual worms and transfer them to their own individual wells with E. coli and no more rotting fruit. And then because the worms conveniently have a hermaphroditic lifestyle, they can reproduce. And so what you can obtain are clonal lineages of individual isolated worms. So she's done this for thousands and thousands of worms from many parts of the world. And what she also noticed with her keen eye is that under the microscope, some of these worms looked sick. Okay. So I'm not going into the details of what the sick worms look like. They had some observable phenotypes under the microscope to a worm expert. But so then what she did is she did what most good clinicians would do. They tried to treat it with antibiotics. And some of these worms got better and suggesting they had a bacterial infection, but some of them did not. And so focusing on those that did not respond to antibiotic treatment, she homogenized these worms, passed them through a 0.2 micron filter, added them to naive worms, and found that she could recapitulate some of this phenotype and suggesting that there was an unknown virus causing this morphological disease. And so the bottom line is we use next generation sequencing to identify from some number of worms multiple novel viruses. So we found 3 novel viruses, one from a wild strain of C. elegans from an orchard in Orsay, France. And so that's called Orsay virus. And then 2 viruses in a related species of worm C. briggsii called Santoy and LeBlanc. So what are they? These are viruses that are most closely related to the family of notaviruses, which are positive sense RNA viruses with 2 genome segments. There are 2 known genera of these viruses of this family. The most well-known ones are in the alpha notavirus family, including flockhouse virus. There are beta notaviruses of fish. And so here's a phylogenetic tree showing up here in the top, a cluster of these fish notaviruses, insect notaviruses, and here are these nematode nota-like viruses. Now, I'll note that these viruses seem to be species specific, meaning that the Orsay virus only infects C. elegans, it cannot infect C. briggsii, and the two C. briggsii viruses we found cannot infect C. elegans. So then we went ahead, we sequenced the whole genomes of these viruses, and just a simple note on their genome organization. So the classic notaviruses have 2 segments encoding 3 open reading frames. Likewise, we have 2 genome segments. They're slightly larger. They also encode 3 open reading frames, one which is a totally novel one absent in all other notaviruses. I won't go into the details of this, but this, in early studies we did, this is a protein that's expressed by ribosomal frame shifting as a fusion of the capsid, and we know that this is incorporated into the viral particle. So we have these novel viruses that have some similarity to notaviruses as well as some unique properties. So one question that we wanted to ask about these viruses is, well, what is their tropism? Because what we found these by homogenization of the whole worm, and so that would be the equivalent of trying to homogenize an entire human, let's say, post-autopsy, sequencing the viruses and finding a virus and not knowing whether it's a GI virus or a respiratory virus or a CNS virus. Now, of course, we had a hint based on the morphology of these intestinal cells that was disturbed that this was likely an intestinal infection. And I'm going to give away the punchline because I need to make sure everyone understands the physiology of the worm. And so here's a cartoon of the C. elegans. C. elegans has about 1,000 somatic cells in the adult worm. And what you can see here is that it has actually exactly 20 intestinal cells. They form in 10 pairs of 2 cells that together define the entire intestine, and the hollow opening in between is the intestinal lumen. So with these new virus, what we did is we raised antibodies against viral peptides of the polymerase and the capsid, and we did immunostaining to try to define the tropism. And so what you can see here are immunostains against the viral polymerase. And strikingly, what you can see in these different images is that there's exactly 1 cell that we can see with potent staining. That's shown here, different worms. You can see maybe a small dark circle in the middle, and that is the nucleus. So these are like the other notaviruses, cytoplasmic viruses. Many of the worms had 1 cell that we could see positive. Some we could see had 2. Here are 2 adjacent cells. Here are 2 that face each other across the lumen. Here's a worm with 3. And what we've been able to see is as many as 6 cells staining positive in some of these worms, and they're clearly in the intestine. And so this suggests this is a new model for studying enteric virology. Here's some more staining with a capsid antibody with a marker, an antibody that binds to an apical surface protein in the worm. And you can see that there's this very distinct strong staining along the apical surface of the cells, as well as some additional large foci intracellularly. And this suggested that there was a lot of virus particle or virus capsid along the apical surface of the intestinal cell. So to corroborate this, we decided to do some electron microscopy. And so here now what you're seeing is a cross section of the worm. You can imagine the worm is coming out at you through the plane of your computer screen. And focusing in on the red rectangle that I've highlighted is the intestinal lumen. I'm going to zoom in and you can see like the clear villi structures, and in the middle is the lumen. And I hope you can appreciate there's a lot of electron density around the apical surface. And if you zoom in, you can even see crystalline lattices of viral particles. And so these are Orsay virus particles lined up at the apical surface of the intestinal lumen. Obviously, this is a static picture, so we don't know what they're doing there. Are they just assembling there? Did they transit there? Our preferred hypothesis is that these are virus particles lining up, preparing to egress to be shed from this animal as part of the virus life cycle. Okay, so knowing where the virus is now, we've done a lot of other work, and what I wanted to tell you today is some genetic studies that we've done to try to find host factors that are critical for, say, virus infection. And as I mentioned before, our reasons for doing this was that this is a model system. Hopefully, the findings we would find in this Orsay virus C. elegans systems could be extrapolated to a mammalian context. So to do this, we set up a genetic screen. And the critical part of the screen is what is the assay, what is the reporter? And what we settled on is a transcriptionally activated fluorescent GFP reporter. The bottom line is we did some expression profiling to find genes that are rapidly induced, robustly induced by virus infection. We found a gene identified as PALS5 that's induced about 1,000-fold by virus infection. And through a collaboration with Emily Tremel's lab, who is at UCSD, and she studies microsporidial infection of C. elegans, it turns out that microsporidia and Orsay virus both activate strong transcriptional induction of this PALS5. And so if you put clone GFP behind this promoter, integrated into the worm, you get a reporter worm that turns green upon virus infection. So with that as our tool, we then set out to use the classical tools of chemical mutagenesis. And so we could take this reporter strain, which normally is GFP off, mutagenize it by EMS treatment, challenge with Orsay virus, and the vast majority of the worms still turn green, and a very small number of worms, less than 1%, fail to turn green. And these are the worms we're interested in because we presume they're mutated in some gene that's critical either for virus infection or for responding to the infection to turn on the GFP. So we can isolate these mutants, amplify them, and using standard mapping and sequencing type approaches, we can identify the gene, we can quantify, and we assume they should have some defect in Orsay virus infection. So the mutants that we found either are defective in a gene required for virus infection or replication, or they're defective in one of the genes responsible for signaling. And so we think that through this, we could also find, for example, the equivalent of RIG-I or the downstream transcriptional factors necessary to activate host responses. So we did an initial preliminary screen, and from that, we identified 3 of these mutants that we refer to as VIRO for virus-induced reporter off. And so here's what that looks like. So here's a worm. In this first column are the control worms. So in the absence of infection, they're off. After virus infection, you can see GFP. And in these mutants, there's no GFP after virus infection. We can also challenge them with the microsporidia. And you can see in all cases with the microsporidia, they still turn on GFP. So these mutants aren't due to some trivial loss of your GFP reporter cassette. They actually have a virus-specific defect in GFP signaling. So we next then took these worms and we quantified their virus load. And what you can see here is here's the parent. And here 2 of the 3 have very low virus loads, more than 6 log reduction. This other mutant here appears to have near wild type levels. And we think this is probably one of the sensor type mutants, but I'm not going to talk about that anymore today. Instead, I'm going to focus on these genes here, these mutants here. And so then we tried to map the mutations in this VIRO1 strain. And our leading candidate was a gene called SID3. SID3 is a gene for which at that point in time, there was 1 paper in the C. elegans literature describing it. It encodes a non-receptor tyrosine kinase. That's a cytoplasmic protein. And it was thought to be important for importation of small RNAs via some kind of endosomal trafficking. So with this as our leading candidate, we demonstrated by a number of different approaches that SID3 is the causal gene. I'm just going to show you one example of how we do that in worms. So we have this mutant. We think it has a premature stop in SID3. So to try and prove that this is the gene, we can try to rescue this by supplementing it with wild type SID3 in trans. So much like you would do this in a cell culture by transfection of a plasmid, we can introduce genes on plasmids or in phosmids into worms by microinjection. And so that's what you see here, is that we have this VIRO1 strain to which we've ectopically introduced the wild type form of SID3. And in this line now, they're in the absence of infection, they're off, but now they can turn green after infection. Whereas before in the VIRO1, it fails to turn green. By viral load, we can also see by qPCR that this can rescue the viral replication numbers. So this was with wild type SID3. Since I told you SID3 is a kinase, we made a version of the kinase that should be catalytically inactive. So we made a point mutation. And when we introduced that, we failed to restore GFP. We also failed to restore the virus load by qPCR. And so this suggests that the SID3 kinase activity is important for its function in promoting Orsay virus infection. Okay, so this was great. We had a gene. Now, remember, the question is, can this be extended to mammalian systems? And about half the genes in worms have human orthologs. And fortunately for us, SID3 has a human ortholog. And that gene is identified as ACK1 or TNK2. This stands for activated CDC42 associated kinase. And this gene has been well studied in many contexts. but not in the context of virus infection. So there's no literature linking this to a role in virus infection. And this was really important because remember, I told you we wanted to study these esoteric, highly divergent viruses because we thought they could teach us something new and different about biology that the conventional approaches couldn't. So this gene has been studied in cancer. It's a kinase. Many of its substrates have been identified biochemically, and it's a wide range of substrates. And so what we wanted to do next was to see, well, what happens in virus infection if we knock this gene out in mammalian cell lines. And so we knocked it out in a couple of different cell lines using CRISPR. And then we tested a wide range of mammalian viruses. And we had to do this because the Orsay virus is unable to infect mammalian systems. And the notaviruses, there's no thought that any of them are bona fide mammalian viruses. There is one virus, notamuravirus, that can infect murine cells, but most of the notaviruses have more limited tropisms to invertebrate-derived cells. So we tested a wide range of viruses, and in the end, the virus family that seemed to have an impact was picornaviruses. And so here you can see that here we generated independent lines of knockout cells. This is in A549 lung epithelial cells, and using encephalomyocardial virus, which is a murine picornavirus, you can see at early time points after infection, we can get as much as 90% reduction in viral protein in infected cells. In multi-step growth curves, we can see at 24, 36, 48 hours, we can get up to 4 logs, almost 4 logs of reduction in viral load in the knockout. And this is true for multiple viruses in the family of picornaviruses. So we tested Coxsackie-B3, polio, NROD68, and all of them had some dependency on TNK2. There was some variation in the quantitative nature of this, but clearly multiple viruses that are mammalian pathogens utilize TNK2 in some way to optimize their infection. So what this told us is that the C. elegans system can be utilized to find evolutionarily conserved functions in supporting virus infection. And again, just to remind you, there was no studies that previously had found that TNK2 was important in supporting roles of virus infection. Okay, so this was great. This is in vitro cell culture work. The next thing we did then is we generated a knockout mouse lacking TNK2, and we challenged that with EMCV. And what you can see here is that the TNK2 knockout is more resistant, has increased survival compared to the wild type. And this is similar to what happens in the worms, where you have much lower virus load in the TNK2 knockout compared to the wild type. And so this tells us that this pro-virus function of SID3/TNK2 in vivo is evolutionarily conserved from nematodes to mammals. Okay, so this was super exciting. So this was our first candidate gene. Remember now, from this initial screen, we had a second gene called Viro2 at that point. We mapped this. It came up with this gene locus name, which means that nobody has studied this in C. elegans, but annotation-wise, it was an ortholog of the human Wiskott-Aldrich syndrome protein, which is known to be a mediator of actin polymerization. I'm not going to go through any of the details. All this is published, but we validated through multiple means that this is the causal gene. And the thing that was most interesting to us is that if you recall, I told you that TNK2 is a kinase with many defined substrates in mammalian contexts. But quite amazingly, we found that, this gene, WASP, the worm gene is an ortholog of one of the known mammalian substrates. So in mammalian cells, TNK2 phosphorylates WASP, and what we found are the worm orthologs of these two genes. And so just to make sure that's totally clear, in humans, what's known is that TNK2 phosphorylates WASP. This increases its actin polymerization potential. What we identified in C. elegans is that SID3 is important for virus infection. We know that its kinase activity is important. We do not know if it specifically phosphorylates this WASP ortholog, but we know that both of these in worms are critical for virus infection. I showed you that TNK2 is important in mammalian cells. So the next question is WASP important? And so similarly, we made knockout cells in A549. You can see significant reductions at both early time points and in multi-step growth curves. And so what this tells us is that not only does WASP have this evolutionarily conserved function, but that this is a pathway that has evolutionarily been conserved from, again, nematodes to mammals. So this was really exciting, and that was a small scale screen that we started with where we found only a couple of mutants. We've subsequently done much larger scale screen. We found actually identified many more mutants, and of those mutants, we've characterized some of them. And so here's a table. I didn't have time to tell you, we identified NCK1 as another gene. This is actually a protein that's known to bind to both TNK2 and to WASP, and so that's clearly part of that pathway. There's a kinase, another putative kinase DRL1, and then we still have actually quite a few additional mutants, even more than shown here, that we've yet to identify. But I'm going to just tell you quickly about our most recent studies where we did identify the causal gene responsible for another one of these mutants. And that led us to the identification of a gene in worms called HIPR1, which stands for Huntington Interacting Protein Related. So this is a protein that has known clathrin and actin binding. It's thought to be a connector protein that sort of connects these pathways of endocytosis with actin biology. The gene in C. elegans has not been extensively studied. There are a couple of papers, but the yeast ortholog has been very well studied. And in humans, there's two orthologs of this gene, HIP1 and HIP1R. So here's the C. elegans gene. There's a human HIP1R here and a human HIP1 here. So I'm not going to show you any of the data that demonstrated that this is the causal gene in worms. Again, this is all published already. We did the same thing as before. We knocked out these genes by CRISPR in mammalian cell lines. And again, here in A549, you can see HIP1 alone knockout, you have a clear reduction. HIP1R knockout, you have a reduction. If you look at a double mutant, you still get similar amounts of reduction. And so again, here's a gene that's evolutionarily conserved in terms of promoting virus infection in worms and in mammalian cells. So what do we know about this protein? So this protein has a number of different domains. There's a N-terminal anth domain that's involved in binding of phospholipids. There's a coiled domain, presumably for interactions, and also this is involved in clathrin binding. And then there's this Thatch domain, which is involved in actin binding. Again, I'm not going to tell you the details. We did some dissection to determine which domains of these are important. And it turns out that both the anth and the Thatch domains are critical for its function. The structure or the organization of these genes is conserved in humans. And so what we tried to do is we tried to rescue the human HIP1 deficient cell lines with this worm HIPR1 and vice versa. We tried to put human HIP1 and HIP1R into C. elegans. Though none of those experiments worked, but there's a lot of reasons why those might fail. So then what we did next is we then made a series of chimeric proteins where we only swap domains. And so I'm going to just show you data for this one construct where in the C. elegans backbone, we put in the human HIP1 anth domain, and then we tried to see if this could rescue the HIPR1 defect in C. elegans. And in fact, it does. And so here you can see, here's the mutant line. And when you add back in this fusion protein, this chimeric that has the human anth domain, you can see that you restore GFP expression. And again, by qPCR, you can also see that you can rescue this. And so what this tells us is that the human HIP1 anth domain has conserved function such that it can actually function in C. elegans to rescue the virus infection. All right, so we've also done a large number of other studies trying to dissect more of the mechanism. For the sake of time, I'm just going to summarize in a cartoon what we think we know. And so that cartoon is shown here. In a number of studies, again, that I don't have time to tell you, but are published, we know that these genes are important in worms and in mammalian cells at an early stage of virus infection, pre-replication. And in some studies that Hongbin Jiang, a very talented postdoc in my lab, did, he determined that TNK2 deficient cells have a partial defect in virus internalization. We also know that TNK2 binds to NCK, which binds to WASP, and this leads to this phosphorylation event. And all of these appear to be critical in some aspect of this early life cycle. We also know that in C. elegans, the HIPR1 is important at this similar type of stage. And we don't quite know exactly the linkage of this, the HIP1 and HIP1R genes to this pathway, but a lot of the literature links these to endocytosis. And so we have this model where we're identifying a large number of genes that are involved in facilitating and promoting virus entry and virus endocytic pathways. And so with that, I'm going to conclude by telling you that I described today this new model system. It's not really that new. We've been working on this for 10 years now. A new C. elegans or say virus model system to study host virus interactions. And I've shown you that we've identified a large number of hits just from one single genetic screen. One can envision many other types of genetic screens to look for different kinds of host gene functions. And so one example is in collaboration with Eric Miska's lab, a large genomic screen for antiviral genes was done. And likewise, from that screen, we found a novel gene that is important for antiviral resistance, both in worms and that's evolutionarily conserved and has function in mammals. I told you today specifically about novel proviral functions of T and K2 and WAS-L, and that the premise here is that novel genes, and not just genes, but actually entire pathways that are evolutionarily conserved between nematodes and mammals can be identified using this very simplistic and primitive virus infection system. And so with that, I have to thank the people in the lab who have done the work. Most of the work I talked about today is done by these people in blue, highlighted by, again, as I mentioned, Hongbing Zhang, an undergraduate, Christian Leung, a recent graduate student, Louis Sandoval, and another student, Stephen Tahan. I have a lot of collaborators who I've tried to mention along the way. The EMs were taken by Wang Beatty, funding from different sources. If this type of work interests anyone, I'm looking for postdocs. And with that, I'm happy to take any questions. I'll stop sharing and open it up.
Thanks, David. Very interesting talk. I'm just going to read some of the questions. First one is from Nazarena. Is there any evidence of significant role of viruses in human pathogenic nematodes?
So that's a great question. I know a lot of people have been thinking about this. And surprisingly, the number of viruses in nematodes, period, that people have found is remarkably low. When we identified these viruses in C. elegans and C. briggsii about 10 years ago, they were actually the very first molecularly characterized viruses of any type of nematode. Subsequently, with NGS, people found things in other plant pathogens, so soybean cyst nematodes and other kinds of more soil nematodes. And I know people have talked about looking in human parasitic nematodes, but I haven't seen any convincing studies so far. So, although I have no doubt that some of them have viruses. The flip side is that people have thought about whether you could use viruses as some kind of, again, like phage therapy, so viral therapy to try to treat pathogenic nematodes. And again, that's just the things I think people are thinking about.
Okay. Is your assay designed mostly to pick up host factors that are on the entry stage?
So that's a great question. So we actually didn't know originally what exactly our screen could pick up. All we knew was initially was that this gene was transcriptionally activated by infection. Okay. And then as sort of concurrent with doing the screen, what we discovered is that virus replication alone from a replicon is sufficient to activate the GFP transcription. And so what that tells us is that we cannot find things that are post-replication because GFP will already be on. So this screen is in fact biased to find things that up to and including replication. So we can't find any assembly or egress defects with this particular reporter, but there are other ways that we think we can do that. And that would be exactly, as I said, a different screen that we could do to try to find late life cycle host factors.
And one final question, are there any coronaviruses that infect C. elegans?
So that's a great question. There are no coronaviruses that anyone has found in any nematode. So actually in C. elegans, the Orsay virus is still the one and only natural virus that anyone has found. Okay. People have found other relatives of other pathogenic viruses in other related nematodes, like there is a flavivirus of the soybean cyst nematode. And we've actually identified Bunya-like viruses. So far, no one has found any corona-like viruses. Interestingly, we're going to do some experiments to try and see if by transgenically adding ACE2 into and TMPRSS2 into worms, whether we can get SARS-CoV-2 to infect worms, but we don't know the answers to that yet.
And just sort of following up on that, when you say corona-like viruses, what would you look for in a virus to be able to say it's corona-like, as opposed to say flavy-like or some other? Is there any specific things that you would?
Right. Well, of course, the first hint comes from the sequence conservation, typically of the RDP, and then, but once you have that, if you have the whole genome sequenced, you would have, the gene complement, the genome organization.
Okay. So you would look for general sequence similarities and organization.
Sequence similarity and then gene organization, right? I mean, if we found a 30 kb-ish genome in the worms, it would probably be very unlikely it's a flavivirus, right?
Okay. The size is one thing. Okay.
Size, polyprotein, order of genes, etc.
And one final question came in. What's the basis for activation of the reporter?
The basis for the activation. So what we know is that this promoter is responsive. We have identified one host protein, a gene called DRH1, that seems to be necessary for the signaling. That's actually worm ortholog, distant ortholog of dicer-related, it stands for dicer-related helicase. It's a distant relative to RIG-I. So it seems potentially to be a sensor, but we do not yet know the rest of that pathway or the transcriptional factors that are critical for activating this particular promoter. But that's an area that we and others are investigating.
Okay, thank you, David, for a very interesting talk. And I pass it to Gabriel to introduce our final speaker.
Thanks very much.
Thank you, Nicole. Well, I will introduce the final, as you say, but not the least. I will also introduce the second bloom from this symposium, Jesse, which is son of Marshall Bloom. And full disclosure, when we were putting together the panelists at the meeting, we didn't know on their relationship, so we learned after the fact. But Jesse was invited because he's a young, very successful virologist. He did his PhD in Caltech under the supervision of Francis Arnold, and his postdoc also at Caltech, but under the supervision of David Baltimore. As you know, both are Nobel laureates. Then he moved in 2011 to Fred Hutch in Seattle, where he has his own lab. He is now associated professor from the Basic Science Division and Public Health Science Division. He is a Howard Hughes Medical Institute investigator. Jesse studies evolution using viruses and proteins as models, specifically focusing on influenza. He uses computational biology and real-world data to understand the constraints of protein evolution. Today, we'll hear about his work on SARS-CoV-2. Thank you, Jesse, and all yours.
Okay, thank you. And hopefully you can see my slides. If people can't see my slides, someone please unmute themselves and interrupt me and let me know.
We can see your slides.
You can see them? Okay. Yeah. So thanks for the kind invitation. It's a real honor to speak here today. And today I'm going to be talking about the evolutionary potential of the SARS coronavirus 2 receptor binding domain, or RBD. And the slides I'm presenting right now are available at this link if you'd like to follow along. So I'll wait 1 second if anybody wants to copy down that link before I get started. Okay, I'll have the link at the end too. So as I'm sure most of you know by now, SARS coronavirus 2 virions are decorated by spike proteins shown here in red that mediate viral entry into target cells. And one key portion of the spike is the receptor binding domain, shown here in purple, which is an autonomously folding subdomain of spike. And the function of the receptor binding domain is to bind to ACE2, which is a protein that is expressed on the surface of target cells and serves as the virus's receptor. And so the binding of the receptor binding domain, or RBD, to ACE2 is the first step in the process of cellular infection. In addition, we now know that neutralizing antibodies against SARS-CoV-2 often bind to the RBD, and in many cases block its interaction with ACE2. So for instance, there have been several recent papers that show that anywhere between 75 and 98% of the neutralizing activity in convalescent sera is mediated by antibodies that bind to the receptor binding domain. So for these two reasons, mapping how mutations affect the RBD is really crucial for understanding the virus's evolution. It's both crucial for understanding how at some point in the past, this virus acquired the ability to effectively bind human ACE2 and then be able to infect humans. And it's also really crucial to understand whether any of these mutations might affect the efficacy of antibody or vaccine mediated polyclonal antibodies that are sort of our best hope for combating this pandemic. So in biology and virology, we're very used to the idea of mapping the sequence of a protein to its structure. So for instance, I just showed you the cryo-EM structure of the spike, and that was done by an experiment which took the sequence of the protein and figured out what structure it folded to. Today, I'm not going to talk about mapping the sequence of these proteins to their structures. Rather, I'm going to be talking about mapping changes in the sequence of the protein to changes in various biochemical phenotypes. And in particular, I'm going to be focusing on biochemical phenotypes of the SARS-CoV-2 receptor binding domain, which we think are relevant to viral fitness. So those include the ability of this protein to properly fold, its ability to bind with high affinity to human ACE2, and then its ability to be recognized by antiviral antibodies. And so even though all the experiments I'll discuss are directly mapping changes in sequence to changes in these biochemical phenotypes, the structure of the protein still matters a lot as it sort of implicitly determines this mapping from mutation to phenotype. So my talk is going to be divided into three parts. First, I'm going to simply talk about how we build a map of how all mutations to the RBD affect its ability to fold and bind to ACE2. Then I'm going to discuss how we can map how all mutations to the RBD affect the binding by antiviral antibodies. And then finally, I'm going to discuss how these maps can be used to predict sort of if and how the virus can escape from antibodies. And I'll also kind of put in some vignettes related to some mutations that are observed in this recent ferret cases of SARS-CoV-2 that I think will also illustrate the utility of these biochemical maps of the effects of mutations. So first, I'd simply like to discuss how we can map how all mutations to the RBD affect these biochemical properties of folding and ACE2 binding. So there are quite a few possible mutations you can make to the SARS coronavirus 2 receptor binding domain. It's 201 residues long, and each of those can be mutated to 19 other things. So that's 3,819 possible amino acid mutations. So it would obviously take a long time to characterize all of these individually using sort of conventional biochemical approaches. So we instead used a high throughput biochemical approach that involves yeast. So basically what we did is we took advantage of the fact that the RBD is autonomously folding, and we expressed it on the surface of yeast, where we can then label the RBD with a fluorescent tag to see how much properly folded protein is on the yeast surface. And we can also incubate the yeast with fluorescent ACE2 to see how much ACE2 this RBD is capable of binding. And importantly, we didn't just do this for some reference strain of the SARS-CoV-2 RBD. We actually built a library of yeast, with each yeast expressing a different RBD mutant, and then the yeast carrying a 16 nucleotide barcode that tells us which particular RBD mutant they have on their surface. And I'm not going to go into the details of exactly how we made these libraries in this talk. You can click here if you want to see details of that. So for a single yeast variant, it's actually pretty easy to see how you could use these yeasts combined with a flow cytometer to actually make real biochemical measurements. So you could take your yeast and you could incubate them with fluorescently labeled ACE2. And then if you did this at various ACE2 concentrations and ran the yeast in a flow cytometer, you'd see something like this. When you don't have any fluorescent ACE2, there's very little fluorescence in the yeast. And then as you add ACE2, more and more of the fluorescent ACE2 binds to the yeast, so they become more and more fluorescent. And then at some point, this binding is saturated. So if you just sort of turn these flow cytometry histograms on their side, so we're plotting fluorescent signal versus ACE2 concentration, what you end up with is a biochemical binding curve shown here, which you can use to do things like determine the dissociation constant for the interaction of this receptor binding domain with ACE2. So importantly, we don't just have one yeast variant, we actually have a whole library of yeast variants. But we can repeat that same experiment where we have all of the thousands, actually there are hundreds of thousands of different RBD mutants in this library. We incubate them with ACE2 and we run them on the flow cytometer. Now you can see that the flow cytometry histograms don't look nearly as sharp as before because we're seeing sort of a smear of the effect of all of these different mutations. But any given RBD mutant is still sort of transitioning from left to right here according to the binding curve governed by its dissociation constant. So rather than just reading out the flow cytometry signal, we instead sort these yeast into bins and then use deep sequencing to determine how each RBD variant is sort of migrating in fluorescent signal as we increase the ACE2 concentration. And so that deep sequencing allows us to then infer these biochemical dissociation curves for all of these mutants of the RBD at once. And then we can sort of use a similar approach where we're just looking at the tag on the RBD to measure the expression of folded protein for each RBD mutant. And so the result of this work are these big phenotypic maps of how mutations affect RBD folding and ACE2 binding. So I'd like to show you what these maps look like. So here's an example of one of these maps. And I'd like you to sort of think of these maybe in the same way you'd think of a crystal structure. So a crystal structure allows you to determine, sort of maps the amino acids to three-dimensional space. And here we're mapping the amino acids to their biochemical phenotypes. So at the top here is the phenotype of the receptor binding domain binding to ACE2, so the log dissociation constant. And here is the level of expression of properly folded RBD. And then each cell here is a different mutation. So for instance, if we go right here, we can see that mutation S349Y is fairly bad for ACE2 binding. So red means bad for that property, and is quite bad for expression of properly folded RBD. And if we just look through this map, you can see there are some sites where there's mostly white, so mutations are pretty well tolerated. There are occasional sites where mutations are even beneficial for these biochemical phenotypes. And then there are a lot of red sites where mutations are deleterious. And so this is just kind of a lot of information to look at if you don't really care about the receptor binding domain. But right now, a lot of people care about the receptor binding domain. So it's really useful to have this map. So for instance, some of you may have read in the news that there is this, these strains of SARS-CoV-2 that have been isolated from these mink outbreaks in Denmark. And so for example, many of those viral strains coming from the mink have a particular mutation, Y453F. So we can just go look at our map and we can see what does that particular mutation do. And you can see that this Y453F mutation that's found in these ferret isolates actually increases the affinity of the SARS-CoV-2 receptor binding domain for human ACE2. And in fact, it was probably selected in ferrets, or I mean in mink, to better bind to mink ACE2, which has a few differences at the interface. And sort of an incidental side effect of that is that mutant now has a higher biochemical affinity for human ACE2. And then there's lots of other mutations that have, sort of popped up in these viral strains, and you can use these maps to go see what those mutations do. So, okay, so to go back to my talk, we can also sort of look at these phenotypic maps projected onto the structure of the receptor binding domain. So for instance, here is the ACE2 molecule. This is a crystal structure. And here's the receptor binding domain. And you might want to then see how does any given mutation affect this particular interaction and where is that mutation found. So again, if we go look for Y453F, our favorite example, you can see it's right here. It's sort of near the interface with ACE2, and you can see which are the mutations that are preferred at this site for binding to ACE2. And you can actually see that the wild type residue of R binds to ACE2 less well than this mutant F, which is is popped up in these mink outbreaks. Okay, so there I've just kind of told you how we can generate these big maps of information and hopefully given you a little bit of illustration why they might be of use if you really care about the evolution of the RBD. So that was the first section of my talk. I'd then like to tell you about sort of more large-scale data generation before I really tell you how we can use these data to do interesting things. So as I mentioned, a second reason why we care about the receptor binding domain is that it's the major target of neutralizing antibodies to SARS coronavirus 2. So we were also interested in mapping sort of a third biochemical phenotype, which is namely how do mutations to the receptor binding domain affect recognition by antiviral antibodies. So to do that, we take our population of yeast, we simply incubate them with a fluorescently labeled antibody, and then we use flow cytometry to identify those yeast that are binding less antibody than most of the rest of the yeast. And then we just use deep sequencing to figure out which receptor binding domain mutations were present initially and which ones are present in this antibody escape population. And I'm going to represent these phenotypic maps as shown here, where I draw a tall letter when a mutation is very good at allowing the virus to escape from binding by the antibody. So basically, when I show these data, remember, if you see a tall letter, it means it's a mutation that can escape from the antibody. So for instance, here's what this escape map looks like just for one particular monoclonal antibody, not maybe of tremendous interest from a therapeutic perspective, called COV22499. So there are just a few sites in the receptor binding domain, which I'm showing here. I'm only showing the important sites where mutations can escape from this antibody. These mutations are clustered in around 440 and around 490 in the primary sequence of the receptor binding domain. And as I discussed, the phenotypic effects of mutations are generally determined by the protein structure. So if we were to project this map of phenotypic effects of mutations onto the protein structure, we can see that very clearly. So here's, again, there's no crystal structure for this particular antibody, but here's ACE2 in the dark color, and here's the receptor binding domain in sort of the white color. And then here, this line plot is showing all of the mutations such as 443, 445, 446, 496, et cetera, that escape binding from the antibody. So if we just look and see where those mutations are, we can see that they all cluster in a coherent region on the protein structure, which must be where the antibody binds. So this makes sense. Why the escape mutations would cluster in one region. This is actually a very rapid way, probably a lot faster than structural biology, to determine the epitopes of antibodies, and quite nicely, it can also just be applied to polyclonal sera in a way that's difficult to do for structural biology needs. So we can do this for lots of other antibodies. So for instance, here's, I'm going to show a few more antibodies. To orient the results, I'm going to sort of categorize the SARS coronavirus 2 receptor binding domain into several structural regions. I'm going to show sites in orange if they're in the core of the receptor binding domain. I'll show them in light blue if they're in the receptor binding motif. So these are the loops that sort of stick out of the receptor binding domain and contact ACE2. And then I'll show them in dark blue if they're residues that are actually in direct contact with ACE2. So you can see this first antibody I discussed, is primarily escaped by mutations in the receptor binding motif, often at ACE2 contact residues. So here are just two other antibodies that also are mostly escaped by mutations at ACE2 contact residues. But I hope you can see that even though these antibodies contact sort of similar regions of the receptor binding domain, they often have distinct mutations. So for instance, COV22832 in the middle here has an entirely orthogonal set of escape mutations from COV22499 here, even though they both target the receptor binding motif. COV22165 and COV22832 share some common escape mutations, such as site 487, but there are also some escape mutations, such as the ones here or the ones here, that are unique to one or the other of these antibodies. And importantly, this sort of high throughput mapping validates quite well in pseudovirus neutralization assays. I'm not going to show those data right now, but if you click on this link, you can take a look. So as we generate more and more of these maps, so at the time we were making this preprint, we had them for 10 antibodies. We now have these maps for I don't know, must be something like 30 antibodies and dozens of human sera, it becomes useful to be able to sort of organize these high dimensional data in a way that allows us to understand the relationships among these antibodies in the space of viral escape. So here I'm showing a multi-dimensional scaling plot. So for instance, if anybody's familiar with single-cell RNA-seq, you may have seen TS&E plots, which project high-dimensional transcriptomic information into 2 dimensions. So a multi-dimensional scaling plot is algorithmically generated somewhat differently than a TS&E plot, but conceptually it does the same thing. It puts antibodies close together or far apart, depending on their similarity. So here I've colored all of these antibodies based on what regions of the receptor binding domain they select escape mutations, and then the antibodies are placed on the map based on how similar escape mutations they have. So for instance, 2165 and 2832, which are shown at the top and the middle here, share some sites of escape, such as site 487, so they're pretty close together. But 2499 at the bottom of these logo stacks has no common escape mutations with these other 2, and so it's far apart from them. And so I think it becomes very useful when you're thinking about sort of understanding large sets of antibodies to be able to project them down in this way that allows you to understand how similar they are from the perspective of viral evolution. Okay, so hopefully you've been able to bear with me through the first 2 parts of this talk, where I've basically simply discussed how we can use this yeast system to generate a large amount of information on how mutations affect the key biochemical phenotypes of the SARS coronavirus 2 receptor binding domain. And for the last part of the talk, I want to discuss how we can use these maps to understand if and how the virus can escape from antiviral antibodies. So we first simply wanted to test if these measurements of biochemical effects of mutations could predict what arises when the virus actually grows in the presence of antibody. So to do this, we didn't use live SARS coronavirus 2 for a variety of reasons, mostly ease and biosafety. We used a spike expressing VSV. We used the particular one developed by Sean Whalen's lab. And I'm not going to go into a ton of detail on the technical aspects of that. Again, you can see this link here for more details. But the idea is that we're growing a virus which needs the SARS coronavirus 2 spike to enter cells in the presence of a neutralizing antibody and seeing what, if any, escape mutations come out. So for instance, if we do these experiments with the antibody COV22499, we did this 16 times. In 11 of those 16 times, the antibody sort of controlled the virus for the duration of the experiment. But in 5 of those 16 times, the virus became resistant to the antibody. And these are the mutations it acquired. 3 times it acquired G446D, and 2 times it acquired Q498R. So if we go to our map of the biochemical effects of mutations on antibody binding, we can see that G446D and Q498R indeed have tall letters, so they escape antibody binding. But it's not so obvious why out of this very large space of mutations that aggregate antibody binding, these 2 are the ones that arose repeatedly. But we can add on some additional information we have. So first of all, we were mapping the effects of all amino acid mutations, but we know that the virus evolves primarily by single nucleotide changes. So now I've subsetted this map of escape mutations to just those mutations that are accessible to the virus by changing 1 nucleotide. Now you can see that there are fewer escape mutations accessible to the virus, and the 2 that actually emerge are again among those. Importantly, the virus doesn't just have to get away from the antibody, it also has to maintain these biochemical properties, such as binding to ACE2 with high affinity. And as I described to you, we've also measured how all mutations to the RBD affect its dissociation constant for binding ACE2. So now I've colored each letter in this map of binding escape by how well that mutation allows the virus to still bind to ACE2. So yellow or light yellow means very poor binding, and brown means very strong binding. So you can now see out of this whole set of possible escape mutations, there are only a handful of mutations that both escape the antibody and retain good ACE2 affinity. And the mutations that actually emerge in the virus are among those handful of mutations. So another way to represent these same data is as shown here. So now I've drawn a point for every possible mutation to the SARS coronavirus 2 receptor binding domain. And I've put the points over here to the right on the x-axis if they escape antibody binding, and I've put them high up here on the y-axis if they maintain good ACE2 affinity. So if we were to speak teleologically from the virus's perspective, as you're never supposed to do in evolutionary biology, the virus would sort of want to escape by getting mutations that are here in the upper right of this plot, because those are the mutations that get away from the antibody without messing up ACE2 binding. And you can see that once we gray out all of those mutations that are not accessible to the virus by single nucleotide changes, there are only a handful of sort of good escape mutations from the virus's perspective. And the ones that actually emerge in our experiments are among that small subset. This is a general finding. So if we look at other antibodies, we consistently find that the mutations that actually arise in the virus are sort of the mutations that are as far in the upper right as it's sort of possible for the virus to get, given the biochemical constraints on the receptor binding domain. We can also understand why the virus is unable to escape from some antibodies. So for instance, despite trying 56 times, we were never able to select any viral mutants that escaped from antibody CoV2-2165. So if you go to our maps of the biochemical effects of mutations, you can see that there are in fact 3 sites where mutations enable the virus to escape binding by this antibody. And even when we subset just to those that can be accessed by a single nucleotide change, it should still appear that there are 4 mutations that have the biochemical property of abrogating antibody binding. But then when we project on our other biochemical phenotypes, we can understand why the virus is unable to escape from this antibody. At 2 of the sites, the escape mutations disrupt expression of properly folded RBD protein, here at 420 and here at 475. So these are not really viable escape mutations for the virus. And then over here at 487, the mutations disrupt binding of the receptor binding domain to ACE2. So these are also sort of not viable escape options for the virus. As a result, the virus can't escape from that antibody. And so I think these phenotypic maps that are really just built at a biochemical level can actually explain to a large extent if and how the virus is able to escape from antibodies. And then finally, I'd like to just point out that that was an initial proof of principle study, but we think it's also going to be quite valuable to build up these maps of escape from the types of antibodies that are in current clinical use. And so we're working on that right now, and I'll just give one example. So here's the map of escape mutations for REGN10933, which is one of the 2 antibodies in the Regeneron antibody cocktail. So these are all of the mutations that we mapped, the ones that are accessible by single nucleotide changes and abrogate binding of this antibody to the RBD. And in their paper describing this antibody, Regeneron did a very large set of viral escape selections using live SARS-CoV-2. And in red here are the mutations that they selected. And so you can see there's a high degree of concordance. Our biochemical mapping is showing the same escape mutations that they select. The nice thing is our mapping is really comprehensive. I mean, even though Regeneron put a lot of work into this, you're only able to do so many repetitions of growing SARS-CoV-2 in the presence of antibody. So it's hard to say whether they found all the escape mutations. And in fact, we can see that they didn't. There's a lot of other mutations which they didn't yet uncover, which also escape this antibody. And so the nice thing is that our approach maps the mutations that they are able to select in the viral escape, plus sort of everything else that escapes from this antibody. And we think it's really useful to have these complete maps for surveillance of emerging viral mutations. So for instance, if we go back to our Danish mink, I discussed how they have this Y453F mutation, and we can look here and we can see that Y453F actually indeed escapes from this antibody in the Regeneron cocktail. So that particular finding was already known from the Regeneron paper. It's sort of buried in the supplement of their paper. But the nice thing is that we think it'll be possible for us to very rapidly build up these maps for lots and lots of antibodies. So then as mutations are observed through mink or whatever other processes are going to happen over the next few years in the evolution of this virus, we'll be able to quickly look up what those mutations do. So just to conclude, I've told you how we can create phenotypic maps of how, at the biochemical level, mutations to the SARS coronavirus 2 receptor binding domain affect key properties like folding, function, and antigenicity. I've shown you how these maps are useful for understanding evolution of the virus under antibody pressure. And I hope I've sort of alluded to how I think having these maps could be useful for viral surveillance in terms of interpreting the effects of mutations that are observed in nature. Finally, I'd like to end with some thanks. This work was all started and really pioneered by Tyler Starr, who's a really brilliant postdoc in my lab, who sort of started doing all of this work in February and has made amazing progress. Ali Greeny is a really excellent MD PhD student in my lab who also put aside her original project and has been working closely with Tyler in this work and now leading the antibody parts of it. And then Sarah Hilton's a former grad student in my lab who built a lot of the interactive visualizations I was showing. This work was a close collaboration with the Crow Lab for the second part on antibodies, in particular Seth and Pavlo in the Crow Lab. And then I'd like to thank a number of other people in my lab and key collaborators in the Crow Lab, Sean Whelan's lab, Neil King's lab, and David Feaster's lab. Finally, I'd like to thank my funding sources. The slides that I presented are available at this link if you'd like to look at them. Thank you for the invitation to speak here, and I'd be happy to take any questions.
Thank you, Jesse. That was a fantastic presentation. There are a couple of questions. I will try to go in order. The first one raising his virtual hand was Alex Compton. Alex, Janelle, can you unmute? There you go. Thank you.
Yeah, so I had a question about whether you think it was justified, the massive culling of the minks based on the identification of this mutation.
So whether it was justified from a public policy perspective to cull the mink is really something that I think is beyond my expertise to comment on. I mean, certainly some of the viruses in these mink were developing a mutation with antigenic consequence. On the other hand, I doubt that that's going to be a feature unique to mink. I think the mink are just offering us one example of the types of things we can expect to happen over the coming years as this virus evolves. So I'm not really able to comment on the, sort of the public policy cost benefit of whether culling the mink was the right decision.
Thanks.
Nazarina? Nazarina, you want to unmute?
Yes, sorry, I wasn't able to unmute. Thank you. One small question on how this approach might be employed to study the potential emergence of new coronaviruses or other viruses for that matter. Thank you.
Yeah, that's a great question. So actually, that's what Tyler in my lab is currently working on. So you can imagine that in addition to sort of plugging in all of the mutants of the SARS coronavirus 2 receptor binding domain into this system, it basically has the capability of looking at, probably 10 to 100,000 different mutants all at once. So what Tyler's done is he's put into this system sort of all of the known sarbecovirus RBDs plus all of the sort of ancestors. If you go back on the phylogenetic tree and you figure out what ancestral sequences are, and honestly, if there were more sarbecovirus sequences out there, we could put even more in there. And then we can simply measure how all of those bind to human ACE2 and potentially ACE2 of, sort of any other species for which you can prepare ACE2 protein to use as a FACS reagent. So I think what this would allow you to do is you could look at phylogenetic trees and see exactly how ACE2 binding affinity was sort of diffusing along these phylogenetic trees. And so that could be useful for understanding what clades of these viruses are going to be able to bind to what ACE2s and with what type of affinity. But that seems to be working, but we don't have really final results from any of it yet.
Jesse, there is another question. If it was possible to determine if there was any or many mutations in the RDV on those cases with reinfection, or probably if you look at the sera from some of those reinfection cases?
Yeah, we haven't looked at that directly. My understanding, I sort of follow the literature as much as I can on the cases of reinfection. I think, and this is not based on any experimental work, so this is not my work at all, the cases of reinfection for which I've seen sort of actual papers. As you know, many of the cases of reinfection are sort of announced on CNN rather than in papers. The reinfecting virus has not had really dramatic mutations in the RBD that seem likely to be of large antigenic consequence, at least in the particular studies that I'm familiar with. I haven't seen any studies of reinfection for which they had sera prior to the reinfection. So I think it's difficult to say whether these reinfections indicate the virus was able to still infect in the presence of neutralizing antibodies, or whether the initial infection elicited a very sort of weak antibody response, and that's what enabled the second infection. That's my understanding, but I can't be certain that I'm 100% up to speed on all of the literature that's come out in that area.
Jesse, I have a question on my own. So in your biochemical map of AC2, you show that there are some mutations that actually increase the affinity or the binding. Have you looked at if any of those mutations are actually reported to be present in escape mutants from any other antibodies that you haven't shown here?
Yeah, that's a great question. So as you noted, there are actually quite a few mutations, including some that are accessible by single nucleotide changes, that further increase the affinity of the SARS-CoV-2 receptor binding domain for human ACE2. And we see no evidence that those mutations are being selected during the virus's evolution. So the way I'd interpret that is the virus sort of already binds human ACE2 well enough, and there's either no benefit or possibly even a detriment to binding more strongly. So by the way, I think this is sort of an interesting contrast between SARS-CoV-1 and SARS-CoV-2. As many people probably know, during the SARS-CoV-1 epidemic, there was evidence of mutations that were fixing or arising in the RBD that made it bind to human ACE2 even better. We haven't seen that at all in SARS-CoV-2, and it's one of the several strange things that we don't understand about the origins of this virus, but it certainly indicates that by the time the first viral sequences were obtained, the virus already bound to human ACE2 well enough, and it wasn't under pressure to bind stronger. The mutation in mink, which I think is this Y453F mutation, which is the mutation that I've seen out there among natural strains that increases the affinity the most. That mutation, I think, was incidentally selected in mink because I think it probably improved the affinity for mink ACE2, and then it also happens to improve the affinity for human ACE2. And then I think it's sort of incidental that it also happens to escape from Regeneron 10933. I do think the fact that there are a lot of mutations that increase the affinity for ACE2 could be bad news in terms of the potential for viral antigenic drift, because one of the things that sort of we've hoped is that because many of the antibodies target sort of the receptor binding region, maybe mutations there will be relatively constrained because if the virus gets mutations in these antibody epitopes, it will decrease its receptor affinity. However, there's also a slew of mutations out there that sort of re-increase that receptor affinity. You could easily get sort of compensatory mutations where you get an escape mutation that is then compensated, that decreases ACE2 affinity and then is compensated by a second site mutation that restores that affinity. And though we haven't looked at it from a biochemical standpoint, I think in a few of these cases that have recently been reported of chronic infection, where there's lots of mutations that fix, at least in one of these cases, in chronically infected humans, there may be some evidence of that sort of compensation happening.
Well, thank you, Jesse. Nicole? Oh,
Thank you, Jesse, for a lovely talk. I think Paolo is going to be giving the closing remarks.
Yes, so I guess I have the privilege of keeping you tied to your screens for an additional minute. And I guess with Dr. Bloom's lecture, we conclude the 2020 Salzman Symposium. And I believe I'm interpreting the sentiments of everyone that this has been really an outstanding symposium, despite the virtual format that we were forced to adopt this year. And I wish to thank the large number of attendees from different parts of the world who participated in this event. I want to thank our exceptional speakers who have given really superb and inspiring lectures, and particularly our 2020 Nobel laureate, Dr. Harvey Alter. It was a real honor to have you with us today. I also would like to let you know that the entire conference has been recorded, and you can find the link to the recording on the symposium website. Then I want to congratulate again the two winners of this year's award, Dr. Corbett and Dr. Avanzato, for their great work. Well deserved. A special thank goes to Mrs. Lenore Salzman and the Salzman Foundation for their support and their generosity in expanding the scope of the Salzman Award with the addition of the Postbac Award, which was an absolute novelty of this year's edition. Thanks to the FNIH for serving as our host, and especially to Janelle Lewis and Kathy Robinson for their outstanding logistic support. Have to say that without them, we would have never been able to set up this almost flawless event, and particularly with this year's extra challenges. And last but not least, I want to invite everyone to join us again next year, hopefully in person here at the NIH, and hopefully after the world has taken control of the COVID-19 pandemic, thanks to the extraordinary work of virologists like Norman Salzman. So see you next year.